I would like to call this meeting of the Molecular and Clinical Genetics Panel to order. I am Andrea Ferreira Gonzalez. I'm the chairperson of this panel. I am a professor and chair of the Division of Molecular Diagnostics at Virginia Commonwealth University in Richmond, Virginia. I note for the record that the members present constitute a quorum as required by 21 CFR Part 14. I would also like to add that the panel members participating in today's meeting have received training in FDA device law and regulations. For today's agenda, the panel will discuss, make recommendations, and vote on information regarding the pre-market approval application, PMA, for the Shield test by Guardant Health, Inc. Before we begin, I would like to ask our distinguished committee members and FDA attendants to introduce themselves. When I call your name, please state your affiliation, your position, and area of expertise. William Bruce.
Good morning. I'm William Brugge at Mass General Hospital. I'm a gastroenterologist interested in malignancy of the GI tract.
Carla.
Hi, I'm Carla Ballman. I'm a professor of biostatistics at Mayo Clinic, and I have interest in early detection and prevention study designs.
Marielle McLeod.
I am Marielle McLeod, Director of Programs and Advocacy for Cancer Hub Network, and I'm an AYA colorectal cancer survivor.
Thank you. Stephen Hewitt.
I'm Stephen Hewitt, Captain in the United States Public Health Service, stationed at the National Institutes of Health, National Cancer Institute, Laboratory of Pathology. My background is anatomic pathology, molecular pathology, and assay development.
Nathan Winslow.
Hi, my name is Nathan Winslow. I'm the industry representative. I am the global head of regulatory affairs for Roche Diagnostics.
Mark Gilbert.
Hi, my name is Mark Gilbert. I am a pediatrician and pediatric gastroenterologist and professor emeritus at Baylor College of Medicine in Houston, Texas.
Donna Roscoe.
Hi, I'm Donna Roscoe. I'm the acting division director for the Division of Molecular Genetics and Pathology in the Office of In Vitro Diagnostics here at FDA.
Charity Morgan.
Good morning. I'm Charity Morgan. I'm a professor of biostatistics at the University of Alabama at Birmingham, and I specialize in clinical trial design.
Edward Lofgren. You're muted, sir.
I am Dr. Edward Lofgren, a retired dentist, but a Crohn's colitis patient, and I'm on the consumer panel. I'm also on the board of the Crohn's Colitis Foundation and the United Ostomy Association.
Alexander Borowski.
Go by Sandy Borowski. I'm a professor of pathology and laboratory medicine at UC Davis, where I'm also director of the molecular diagnostics lab. I have expertise primarily in breast cancer, and I'm part of the executive leadership team of the Wisdom breast cancer screening trial.
Vikesh Singh.
Good morning, I'm Vikesh Singh. I'm a professor of medicine at the Johns Hopkins University School of Medicine. I also direct our pancreatic disease program as well as our endoscopy unit across our health system. My areas of focus are acute and chronic pancreatitis as well as chronic abdominal pain.
Padma Trigadapol.
Yeah, hi, good morning. My name is Padma Sheila Rajagopal. I'm a physician scientist, early investigator in the Center for Cancer Research at the NCI. My areas of focus are germline and somatic genetics, how they're measured from a clinical standpoint and how they're applied to patients, and the intersection across different types of assays.
Thank you. Sivana Tesak.
Hi, Giovanna Tejak. I am a branch chief of molecular genetics branch at the FDA.
Amanda Ratnak.
Yes, hi, I'm Anand Pathak. I'm a medical officer in the FDA's Division of Molecular Genetics and Pathology. I am trained as an MD PhD, and I have training in internal medicine and board certified in preventive medicine.
Thank you. Sean Spencer.
Hi, good morning. I'm Sean Spencer. I'm a physician scientist at Stanford University, general gastroenterologist, as well as a neurogastroenterologist. I'm interested in molecular diagnostics, particularly as they pertain to GI disease. Thank you.
Yuhan.
Hi, my name is Yihan. I'm a scientific reviewer in the division of the molecular and genetics pathology, and I'm the leader reviewer for the Gardant Shield test.
Elisa Garcia.
Hello, good morning. My name is Alicia Garcia. I am a mathematical statistician with the FDA at their Center for Devices and Radiological Health and with their Division of Biostatistics, and I was the statistical reviewer for this mission.
Thank you all for the introductions. At this time, Mr. James Swink, the designated federal officer for today's molecular and clinical genetics panel meeting, will provide the of interest statement. James?
Thank you. Good morning, everyone. I will now read the conflict of interest statement. The Food and Drug Administration is convening today's meeting of the Molecular and Clinical Genetics Panel of the Medical Devices Advisory Committee under the authority of the Federal Advisory Committee Act of 1972. With the exception of the industry representative, all members and consultants of the panel are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the statuses of this panel's compliance with federal ethics and conflict of interest laws covered by, but not limited to, those found at 18 U.S.C. sections 208 are being provided to participants in today's meeting and to the public. FDA has determined that members and consultants of this panel are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees or regular federal employees who have financial conflicts when it has determined that the agency's need for particular individuals' services outweighs his or her potential financial conflict of interest. Related to the discussions of today's meeting, members and consultants of this panel who are special government employees or regular federal employees have been screened for potential financial conflicts of their own, as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, credas, teaching, speaking, writing, patents, royalties, and primary employment. For today's agenda, the panel will discuss, make recommendations, and vote on information regarding the pre-market approval application for the Shield test by Guardant Health Incorporated. The proposed indication for use statement is as follows. The Shield test is a qualitative in vitro diagnostic test intended to detect colorectal cancer, derived alterations in cell-free DNA from blood collected in the Guardant Blood Collection Kit. Shield is intended for colorectal cancer screening in individuals at average risk of the disease, age 45 years or older. Patients with an abnormal signal detected may have colorectal cancer or advanced adenomas and should be referred for colonoscopy evaluation. Shield is not a replacement for a diagnostic colonoscopy or for surveillance colonoscopy in high-risk individuals. The test is performed at Guardant Health Incorporated. Based on the agenda for today's meeting and all financials interests reported by the panel members and consultants, no conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208. Mr. Nathan Winslow is serving as the industry representative, acting on behalf of all related industry. Mr. Wilson is employed by Rose Diagnostic Solutions. We would like to remind members and consultants that if the discussions involve any other products or firms not already on the agenda, which the FDA participant has a personal or imputed financial interest, that participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the panel of any financial relationships they may have with any firms at issue. A copy of the statement will be available for review and will be included as part of the official transcript. Thank you. I will now read the deputization memo. Pursuant to the authority granted under the Medical Device Advisory Committee Charter of the Center for Devices and Radiological Health dated October 27, 1990, and as amended August 18, 2006, I appoint the following individuals as voting members of the Molecular and Clinical Genetics panel for the duration of this meeting on May 23, 2024. Dr. Carla Baumann, Dr. William Bruget, Dr. Mark Gilger, Dr. Stephen Hewitt, Dr. Charity Morgan, Dr. Padma Rajagopal, Dr. Vikesh Singh, and Dr. Sean Spencer. For the record, these individuals are special government employees or regular government employees who have undergone the customary conflict of interest review and have reviewed the material to be considered at this meeting. This was signed by Dr. Jeff Shuren, Director of the Center for Devices and Radiological Health on April 23, 2024. At this time, I will turn the meeting back over to Dr. Gonzalez. Thank you.
Thank you. We will now proceed to the Guardant Health presentation. I would like to invite the Guardant Health representative to begin. I will remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel chair. In order to help the transcriber, please identify who's speaking. Please be sure to identify yourself each time and every time that you speak. I also want to note that the FDA has received 14 written comments, which have been reviewed by the panel and the FDA staff. Guardant Health, you have 75 minutes to present. You may now begin.
Good morning. Thank you to the chair, members of the advisory committee, and FDA for the opportunity to introduce SHIELD, a blood-based colorectal cancer screening test for average-risk adults. My name is Amir Ali Talasaz, Co-Chief Executive Officer at Guardant Health. We are pleased to be here to share the data supporting a positive benefit-risk profile of SHIELD. Over the course of the presentation today, you will hear from my colleagues and experts across the field of cancer screening about the current opportunity to raise the rate of colorectal cancer screening and reduce preventable CRC deaths. We look forward to sharing more with you today. Colorectal cancer is the second leading cause of cancer-related death in the United States. Consistent evidence supports that early detection of colorectal cancer significantly improves patient survival and reduces preventable CRC deaths. But CRC detection requires that individuals are adherent and complete their screening. Unfortunately, despite the availability of multiple screening options, millions of eligible adults remain unscreened, and screening rates continue to fall short of guideline-recommended targets. New choices are needed to improve CRC screening. There are several guideline-recommended primary CRC screening methods, including invasive options such as colonoscopy and non-invasive stool tests. Colonoscopy is the most accurate and prioritized option given its ability to both screen and intervene when abnormal lesions are identified. In this regard, the National Colon Cancer Roundtable, or NCCRT guideline, highlights that colonoscopy has been shown to reduce death and can also prevent cancer. Because of this, all individuals willing to discuss CRC screening should be offered colonoscopy. For individuals who choose not to undergo the procedure, non-invasive options play a critical role. These stool tests offer a broad range of device performance and the NCCRT guidelines recognize that they all reduce CRC deaths. Today, millions of people are getting screened by each one of these options. There is an FDA-approved blood test, methylceptin-9, which was approved as a second-line option, but was not recommended by guidelines because the status and poor device performance. This hindered patient access and adoption of this screening method, which ultimately led to its removal from the market. Evidence we will present today support that Shield is a novel non-invasive blood-based screening test that should be offered as a choice for patients alongside other non-invasive stool tests. Clinical evidence has shown the value of offering choices to patients in improving CRC screening. As a result, different CRC screening guidelines have recommended offering multiple choices and highlight the role of patient preference. Now, in terms of the performance of Shield, results from our pivotal Eclipse study were recently published in the New England Journal of Medicine. Eclipse demonstrated that Shield has CRC sensitivity of 83.1% and advanced neoplasia specificity of 89.6% compared to the reference standard colonoscopy. With these results, this study met both pre-specified co-primary endpoints, establishing Shield as an effective screening option. To help contextualize the screening landscape, presented here is the one-time testing performance of non-invasive CRC screening options, with CRC sensitivity on the top and AN specificity on the bottom. Shield, shown in dark blue, is the first blood-based test with CRC sensitivity and AN specificity in range with guideline-recommended non-invasive screening options. The testing interval for Shield has not been established yet, and Shield may need to be repeated every 1 to every 3 years, similar to stool-based tests. The Eclipse study also demonstrated that Shield has limited sensitivity for detection of precancerous lesions, and as such has limited ability to prevent CRC. Advanced adenoma sensitivity with Shield was 13.2%, and 22.6% of advanced adenomas with high-grade dysplasia were detected. Shield's AA sensitivity is on the lower end of the range of widely used non-invasive CRC screening options. It is important to recognize that colonoscopy is the most accurate test for AA detection with AA sensitivity of 95%. Even the widely used tests that are currently recommended as primary choices. Based on these findings, screening for advanced adenoma is not a proposed indication for use of Shield. Here is our proposed indication. Shield is intended to detect colorectal cancer in individuals at average risk of disease, age 45 years or older. As shown in this table, Shield's performance for CRC sensitivity of 83%, AN specificity of 90%, and advanced adenoma sensitivity of 13% are in range of stool tests. In addition, adherence to complete Shield may be much higher than stool tests. The evidence from real-world clinical use of the laboratory-developed test, or LDT implementation of Shield, consistently demonstrates adherence exceeding 90% in over 10,000 prescribed blood tests. We look forward to discussing with the panel today about the safety and effectiveness of Shield in detecting CRC in unscreened patients, and use of the Shield test as a primary non-invasive screening choice alongside and on equal footing relative to stool tests. Here is the agenda for today's presentation. Dr. Peter Leong will describe the benefits of CRC screening and the need for additional screening options. Dr. Daria Chudova of Guardant Health will then present an overview of the Shield development program. Dr. Daniel Chong will describe the Eclipse study design and review the effectiveness and safety results. Dr. Mani Singleton will provide his clinical perspective. And finally, Dr. Craig Eagle of Guardant Health will conclude the presentation. We also have an additional expert here today who's available to address questions from the advisory committee. All outside experts have been compensated for their time and travel to today's meeting. Thank you. I will now turn the presentation over to Dr. Leong.
Thank you and good morning. My name is Peter Leong. I'm a gastroenterologist, researcher who focuses on colorectal cancer prevention and health disparities, and an assistant professor at NYU Grossman School of Medicine. I am pleased to be here to discuss the benefits of colorectal cancer screening and the need for additional screening options. Colorectal cancer is a major public health challenge in the United States. It currently ranks as the 4th most diagnosed cancer and the 2nd leading cause of cancer-related death. It's estimated that in 2024, more than 150,000 adults will receive a colorectal cancer diagnosis, and more than 53,000 will die from this disease. Despite these staggering statistics, a substantial proportion of eligible individuals are not up to date with colorectal cancer screening and remain at increased risk. In fact, more than 3/4 of colorectal cancer deaths, or 76%, occur in individuals who are not up to date with their screening. Colorectal cancer is well suited to screening due to the natural progression of the disease. Colorectal cancer primarily arises from precursor lesions known as adenomatous polyps, which can grow into malignant lesions that can either be detected preclinically through screening or can present symptomatically as clinical colorectal cancer. The development of colorectal cancer is not as rapid as for many other cancers. It is a multi-step process that progresses slowly, with an estimated total dwell time ranging from 17 to 29 years. This estimate is based on data used by the U.S. Preventive Services Task Force, or USPSTF, to inform guidelines for colorectal cancer screening. In addition, evidence suggests that less than 5% of adenomas grow into preclinical cancer on an annual basis, with an adenoma dwell time ranging from 13 to 25 years. Early detection of colorectal cancer significantly improves patient survival. Data from National Cancer Institute's SEER database show that the 5-year survival rate among individuals diagnosed with localized colorectal cancer between 2014 and 2020 was 91%. 5-year survival plummets to only 16% if the cancer has metastasized at the time of diagnosis. Therefore, the goal of colorectal cancer screening is to detect the cancer as early as possible to allow for early treatment. Despite this clear link between early detection and survival, still today, when we look at the percent of cases by stage at diagnosis as shown above the figure, 23% of people already have metastatic disease by the time they are diagnosed. Recognizing the significant benefit of screening, guidelines from leading organizations, including the USPSTF and the American Cancer Society, now recommend that screening begin at age 45 for all adults at average risk for colorectal cancer. Guideline recommended screening options are shown in the top blue row of this table and include direct visualization with colonoscopy and non-invasive stool-based tests. Importantly, USPSTF acknowledges that there is no one-size-fits-all approach to colorectal cancer screening and seeks to provide clinicians and patients with the best possible evidence about the various screening methods to enable informed individual decision-making. Despite current screening modalities, the proportion of patients who are up to date with colorectal cancer screening is well below the guideline recommended target, further highlighting the pressing need for additional options. Results from the National Health Interview Survey in 2021 found that 58% of eligible U.S. adults between the ages of 45 to 75 years are up to date with colorectal cancer screening, which is well below the target of 80% set by leading healthcare organizations. Based on the 2020 U.S. Census, this leaves approximately 50 million screening-eligible Americans who are not up to date with colorectal cancer screening. Furthermore, evidence supports that screening rates are lower among racial and ethnic minority populations and among lower socioeconomic groups, highlighting systemic inequalities in colorectal cancer screening access and uptake. When looking more closely at the primary non-invasive colorectal cancer screening tests, evidence supports their ability to effectively detect colorectal cancer with sensitivity ranging from 67 to 92%. On the other hand, the ability to detect advanced adenomas varies across current stool-based options, ranging between 11 and 42%. Because of this, colonoscopy remains the reference standard for the prevention of colorectal cancer, given its ability to detect and remove advanced adenomas. However, the benefit of a colorectal cancer screening test is not only dependent on its ability to detect colorectal cancer or advanced adenomas. The problem with current standard of care screening options isn't efficacy, it's adherence. Published adherence rates to stool-based screening tests range between 28 to 71%. Of note, adherence has been consistently defined across studies of colorectal cancer screening tests as the proportion of individuals who were offered a screening test and elected to complete the test. Importantly, for blood-based tests, adherence ranges from 83 to 99%. Studies have consistently shown that adherence is impacted by barriers associated with current screening modalities. For the stool-based home tests, people have communicated an aversion to handling stool and have expressed concerns about the challenges in performing the test given the complex multi-step process required. When assessing the benefit of a colorectal cancer screening option, adherence matters as much as sensitivity. To illustrate this, let's consider a test that has 100% sensitivity. If no one completes the test, it's useless in detecting colorectal cancer. If we have 50% adherence, assuming all individuals with a positive test receive a diagnostic colonoscopy, we will identify half of those with colorectal cancer. Thus, the accuracy of a test and patients' willingness to undergo it are both critically important in assessing potential benefits of a new screening option. When applying this methodology to clinical practice, we can see the clear impact of adherence on the detection capabilities of current screening options. Presented here is the table I shared earlier with colorectal cancer sensitivity and adherence for stool-based screening tests. When factoring in adherence, the estimated colorectal cancer detection probability is drastically reduced. The issue of adherence becomes more pronounced when we consider that the benefits of colorectal cancer screening require a person to be adherent to a screening test at regular intervals over 3 decades. Making sure individuals are rescreened according to guideline-recommended intervals is a critical factor when evaluating the clinical utility of a colorectal cancer screening option. For example, the recommended screening interval for non-invasive stool-based tests ranges from every 1 to 3 years. This corresponds to 11 to 31 tests throughout a patient's lifetime. Therefore, the barriers to current screening options may be amplified at each screening interval over a patient's lifetime, further impacting longitudinal adherence. In conclusion, despite our best efforts in using currently available screening tests, millions of adults are not up to date with colorectal cancer screening, and colorectal cancer remains the second leading cause of cancer-related death in the United States. In my experience, many patients who choose not to undergo a colonoscopy are willing to consider a non-invasive option. When we discuss what an ideal screening method would look like, they emphasize the importance of accuracy, non-invasiveness, and convenience. As we will hear today, an effective blood-based screening alternative could bridge this crucial gap, which would enhance patient access and increase the number of individuals up to date with screening, with a goal of reducing preventable colorectal cancer deaths by increasing the number of individuals who are up to date with colorectal cancer screening. Thank you. I will now turn the presentation over to Dr. Chudova.
Good morning. I'm Daria Chudova, Chief Technology Officer at Guardant Health. In my presentation today, I will discuss Shield operating principles and briefly review device development. The scientific principle of Shield is based on the identification of DNA fragments originating from the tumor and found in circulation. As both normal and tumor cells turn over, their DNA is released from inside the cells and digested into smaller fragments known as cell-free DNA, or cfDNA. Unlike normal cells, tumor genomes are known to harbor a significant number of genomic and epigenomic alterations, and tumor-derived cfDNA fragments carry these alterations into the bloodstream, providing a source of cancer-specific markers. Over the past 10 years, measuring tumor-derived alterations from blood has become a well-established methodology. In fact, the Guardant 360 CDx test was the first comprehensive liquid biopsy approved by the FDA for therapy selection and tumor profiling in advanced stage cancers. In developing SHIELD, we have expanded the capability of our Guardant 360 technology to be able to identify earlier stage disease present in the asymptomatic individuals. This extension was enabled by assessing the methylation state of cfDNA, as genome-wide aberrant methylation is one of the key alteration types known to arise early in colorectal neoplasia. The figure in this slide depicts typical methylation signals observed in cfDNA across selected genomic regions. Here, 50 individuals with CRC are shown on the top panel, and 50 individuals without CRC are shown in the bottom panel. Darker colors in the heat map represent higher levels of methylation within the specific region and individual. Increasing level of methylation across selected regions is evident in cases with CRC relative to that observed in controls. Differential methylation across the genome forms the basis for the CRC detection capability of the SHIELD assay. During development, the assay was optimized for efficient cfDNA molecule capture and selection of highly informative genomic regions. Next, SHIELD classification modules were developed using large, independent development cohorts, including more than 3,800 samples, representing individual CRC cases across all cancer stages and relevant controls. Using this data, the statistical models were trained to optimally separate the profiles of individuals with and without CRC. Prior to proceeding with clinical validation, the performance of SHIELD was assessed on an independent verification cohort with more than 1,000 CRC cases. This data provided strong evidence for the CRC detection capability of this device, as shown in the relationship of CRC sensitivity against specificity. The specificity target for this device was established based on the clinical risk-benefit considerations for a blood-based CRC screening test, where individuals with a positive result would be referred for colonoscopy evaluation. As such, a specificity target of 90% was established for the assay during development. And in this targeted range of 90% specificity, we observed a high sensitivity of over 85% CRC detection. In conclusion, SHIELD relies on well-established principles of cfDNA carrying tumor-associated DNA alterations into circulation. Strong CRC detection capability was demonstrated using more than 1,000 independent CRC cases in a verification study conducted prior to clinical validation. Analytical performance was evaluated using more than 15,000 sample test events, and all analytical studies achieved their pre-specified objectives. Thank you. I'll now turn the presentation over to Dr. Daniel Chung.
Thank you and good morning. My name is Daniel Chung, and I'm Director of the High Risk GI Cancer Clinic at Mass General Hospital and Professor of Medicine at Harvard Medical School. I'm a gastroenterologist and physician investigator who has focused on colon cancer my entire career. Specifically, I've been interested in the genetics of colon cancer, risk assessment for colon cancer, and screening for colon cancer. My clinical practice has revolved primarily around cancer screening since I became a gastroenterologist some 30 years ago. With respect to the Eclipse study, I have been involved in its development and execution, and it's a pleasure for me to have this opportunity to share the study with you. I will present the design and results from the Eclipse study, demonstrating that Shield is a safe and effective blood-based screening test for patients eligible for average-risk CRC screening. Eclipse was a prospective, U.S.-based multi-center study designed to evaluate the performance of Shield to detect colorectal cancer in average-risk individuals. The study enrolled participants from October 2019 to September 2022. Following enrollment, participants underwent a study-specific blood draw prior to any medical preparation for colonoscopy. Blood samples were then processed to plasma at a central laboratory before being shipped to Guardant Health for testing. Within 6 months of enrollment, participants underwent colonoscopy per standard clinical practice. All abnormal colonoscopy findings were confirmed by central histopathological review. All clinical data analyses were conducted by an independent CRO. Follow-up is ongoing and will continue for 2 years following colonoscopy. Eclipse enrolled participants 45 to 84 years of age at average risk for colorectal cancer who were undergoing routine screening with colonoscopy. Individuals were excluded from the study if they had a prior history of cancer, inflammatory bowel disease, a hereditary predisposition to CRC, or history of CRC in a first-degree relative. In addition, individuals with a recent colonoscopy or other non-invasive screening test were excluded from the study. Eclipse enrolled individuals from 265 sites across the United States. Trial sites included academic and community centers. A mix of primary care settings and endoscopy sites were included to ensure a broad demographic representation in the study cohort. There were 2 co-primary objectives to evaluate the performance of Shield compared to the reference standard colonoscopy. The first co-primary endpoint was CRC's sensitivity. Sensitivity was measured relative to colonoscopy, and the goal was for the lower bound of the 95% Wilson confidence interval to exceed the performance goal of 65%. The second co-primary objective was the specificity of Shield for advanced neoplasia, defined as CRC or advanced adenoma relative to colonoscopy. Specificity was considered acceptable if the lower bound of the two-sided 95% confidence interval exceeded the performance goal of 85%. The performance goals used in the Eclipse study were based on precedent for approved CRC screening tests. Secondary and key exploratory objectives included sensitivity for advanced adenomas, positive and negative predictive values for Shield, device performance by demographics, specificity for the absence of any neoplastic findings, and assessment of malignancies detected in follow-up. Eclipse was powered for the co-primary endpoints, but the sample size was driven by the number of CRC events. Assuming a true sensitivity for Shield of 80.7%, 68 individuals with colorectal cancer are necessary to provide 85% power for the lower bound of the two-sided 95% confidence interval to be greater than the sensitivity goal of 65%. A sample size of 7,000 individuals negative for advanced neoplasia would provide greater than 85% power to achieve the specificity goal of 85%, assuming a true specificity of 86.3%. Overall, 22,877 individuals enrolled in Eclipse were included in the clinical validation cohort. 10,179 participants were not selected through pre-specified random downsampling and were not tested with the device. 2,440 were used for the specificity interim futility analysis. Eclipse passed the pre-specified interim futility analysis, and thus the study proceeded. This left 10,258 selected participants from the clinical validation cohort. Of these, 7,861 were eligible for analysis. The primary reason for exclusion was an incomplete or invalid colonoscopy, which is not surprising given what is known about colonoscopy completion rates. Out of the evaluable population, 65 had colorectal cancer and 1,116 had advanced adenoma. This slide shows the demographics for the evaluable cohort of patients with both colonoscopy and Shield results, whom we use to calculate sensitivity and specificity. On average, participants were 60 years old. 70% of participants were 50 to 69 years of age, and 22% were over the age of 70. Importantly, the racial and ethnic demographics of study participants were generally comparable to that of the U.S. population. 12% of participants were Black, 7% were Asian, and approximately 13% of the participants were Hispanic or Latino. This is important given that access to screening disproportionately affects minority populations. Turning now to the primary objective results. Shield met the first co-primary objective, demonstrating CRC sensitivity of 83.1% when compared to colonoscopy. Shield detected 54 of the 65 cancers. The lower bound of the two-sided 95% confidence interval was 72%, which exceeded the performance goal of 65%. Shield also met the co-primary objective, demonstrating 89.6% specificity for the absence of advanced neoplasia compared to colonoscopy. The lower confidence bound of 88.8% exceeded the performance goal of 85%. The secondary endpoint of Shield sensitivity for advanced adenoma was 13%. Higher sensitivity was observed in lesions of greatest malignant potential based on size and pathology features such as high-grade dysplasia. There was no unexpected performance variability among subgroups. Presented here are the CRC sensitivity and advanced neoplasia specificity by baseline demographics. While subgroup analysis of CRC sensitivity is difficult due to the small number of cases, overall sensitivity was consistent regardless of age, sex, race, or ethnicity. Similar effects were seen with specificity for advanced neoplasia, with the exception of age, where an inverse correlation was observed. As expected, Shield sensitivity was correlated with CRC stage and also correlated with lesion size. Shield detected tumors from all locations throughout the colon. With more advanced disease as measured by increasing tumor size and tumor stage, there were trends toward greater sensitivity. This is seen by the 55% CRC sensitivity for stage 1 disease and 100% CRC sensitivity for stage 2, 3, and 4 disease. Next, we evaluated Shield's prevalence adjusted positive and negative predictive values for CRC. Using the 0.41% prevalence observed in Eclipse, the positive predictive value was 3.03%. As expected for a CRC screening test, this is influenced by the low prevalence of CRC in the study population. The negative predictive value was 99.9% for CRC. These results further support the benefit of Shield as an effective CRC screening option with performance that is in range with other screening tests. The final key observational objective was specificity for absence of any neoplastic findings. SHIELD demonstrated 89.9% specificity in individuals without any neoplastic findings identified on a colonoscopy. Next, I'll review safety considerations. In terms of safety, the risks associated with the SHIELD device can be categorized into direct and indirect risks. Direct risks include known risks associated with the required blood test. Indirect risks include the consequences of a false positive or false negative for colorectal cancer. SHIELD has a low direct risk. Individuals are only required to undergo a routine blood draw, consistent with other blood-based diagnostic tests. There were no unanticipated adverse device effects observed among the 22,877 enrolled participants. Of the 43 adverse events reported in the Eclipse study, 30 were related to phlebotomy. 13 were unrelated to the study or device, including 2 serious adverse events. All adverse events were reviewed by an independent medical monitor. In terms of indirect risks, as with any screening tests, we'd be concerned about an individual receiving an inaccurate result, either a false positive or a false negative. With SHIELD, a false positive could lead to a colonoscopy and its associated risks. However, colonoscopy is the recommended standard of care in this population, so that risk is considered minimal. Another potential risk of a false positive result is that it may represent a non-colorectal cancer. Data from Eclipse demonstrate that the rate of non-CRC cancers is not increased in individuals with a false positive SHIELD result. As shown in the table, of participants in Eclipse who completed 1 year of follow-up, 0.8% of individuals with a false positive result developed a non-colorectal cancer. This rate is similar to those with a true negative SHIELD test, as 0.9% of patients with true negatives also developed a non-colorectal cancer. The Eclipse study is ongoing and will continue to gather 1 and 2 year clinical outcomes in individuals who had a positive SHIELD test that was determined to be a false positive based on colonoscopy results. In terms of risks associated with a false negative result, this could lead a person with cancer to forego other recommended screening procedures, such as a colonoscopy. For context, the false negative rate of 17% with SHIELD is in range with other CRC screening tests, ranging from 8 to 33%. In addition, SHIELD had 100% sensitivity for detecting stage 2, 3, and 4 colorectal cancers in Eclipse. Over half of stage 1 cancers are also detected, and the sensitivity for stage 1 cancers is in range with other non-invasive screening tests such as FIT. Furthermore, when evaluating the clinical impact of a missed CRC diagnosis, it's important to consider that the natural progression of CRC provides multiple opportunities to intervene through longitudinal testing, as depicted in the figure by the check marks. Colon tumors develop relatively slowly. Advanced adenomas are reported to progress to cancer at a rate of 2 to 5% per year, and the sojourn time for a preclinical colorectal cancer to the diagnosis of a clinical colorectal cancer is estimated at 3.6 to 4.7 years. For these reasons, SHIELD's detection capability for advanced adenomas can be offset by longitudinal testing, and a blood-based approach can improve adherence to this longitudinal testing. The totality of evidence from the Eclipse study supports SHIELD as a safe and effective blood-based screening test for patients eligible for average-risk CRC screening. Shield met the pre-specified acceptance criteria for both co-primary endpoints of CRC sensitivity and advanced neoplasia specificity. CRC sensitivity and advanced neoplasia specificity were consistent across a diverse patient population with respect to sex, race, and ethnicity. However, we did observe an increase in CRC sensitivity with CRC stage and lesion size, and a lower advanced neoplasia specificity among older participants. The secondary endpoint of AA sensitivity showed that Shield has limited detection capabilities for precancerous lesions. Finally, there were no unanticipated adverse device effects observed across all participants enrolled in the Eclipse study. In conclusion, Shield's proven level of performance for CRC detection, combined with the acceptable safety profile, supports its clinical utility as a primary screening option that should be offered alongside other screening modalities. Thank you, and I'll now turn the presentation over to Dr. Singleton.
Thank you, Dr. Chung. Good morning. My name is Dr. Monty Singleton, and I'm the Medical Director of Singleton Health Center and the Medical Center of Santee in Orangeburg, South Carolina. For 8 years, I served on the National Advisory Committee for Rural Health and served as a board member of the National Rural Health Association. I am a National Health Service Corps scholar, and my entire 40-year medical career is focused on serving rural and marginalized populations and on the treatment and prevention of chronic illnesses, including colorectal cancer. I am pleased to be here to share my clinical perspective on the data presented today and how I utilize the multiple CRC screening options. Colorectal cancer screening and the early detection of tumors improves survival. As you've heard today, the challenge we are faced with is not test accuracy. It's the willingness of our patients to complete the prescribed screening tests at each recommended interval. Patients and providers need additional CRC screening options that are convenient, non-invasive, and accurate. An effective blood-based screening option could enhance patient access and adherence to screening recommendations, with the goal of reducing CRC preventable deaths by increasing the number of individuals who are up to date with screening. Today, you will be asked to discuss whether Shield should be offered alongside guideline-recommended non-invasive stool-based tests, or whether access should be limited to patients who decline other CRC screening tests. As a primary care physician, I take great responsibility in facilitating the shared decision-making process, which begins with educating patients on the benefits and limitations of current screening options. Once a patient understands the importance of CRC screening and is activated, there is a discussion of options. For patients who prefer non-invasive screening, we are currently limited to offering only stool-based tests. Once a patient selects a stool-based screening modality, an order is placed and the test is provided to them in the office or mailed to their home. I know that there is a significant risk that some patients will not complete the stool-based test, even though they acknowledge the importance of screening. Many unscreened patients do not decline stool-based tests during the decision-making encounter. Instead, they leave the office and never complete the screenings. This noncompliance underscores the importance of approving Shield alongside other non-invasive screening tests. Restricting access to a blood-based option creates missed opportunities to complete CRC screening increasing the burden of care for accurate tracking and follow-up. For example, if a blood-based screening test was only offered after failing to complete a stool-based test, tracking screening completion would become cumbersome and would create unnecessary management challenges in busy primary care settings. Typically, it requires institutional infrastructure that is oftentimes unavailable in small independent practices, particularly those that care for minority and marginalized populations, where CRC screening rates lag behind national rates. We must recognize that colorectal cancer screening is a partnership between the patient and the physician. This partnership is aimed at making informed decisions about healthcare interventions with the goal of maximizing screening follow-through by selecting an option that is likely to be completed. In this regard, data suggests that screening interventions are higher among patients who are offered tests that align with their preferences, and that adherence to the selected test is increased when multiple tests are offered at the same time a patient is activated. These decisions include test performance, patient preferences, healthcare accessibility, and the frequency at which the selected test should be repeated. Once the test is offered and the patient agrees to complete it, we need to reduce the likelihood of non-adherence, which we know is highest among individuals who are prescribed a test they view as undesirable. In these instances, patients do not actively decline testing, but delay or defer testing they've previously agreed to during the provider encounter. This becomes more important as we consider that CRC screening is a journey, one that requires patients to stay up to date with their screening at every step of the way. This shared decision-making process is key if we want to achieve guideline-recommended screening targets. Having access to all effective CRC screening options during these interactions will maximize screening uptake and increase the likelihood that a test is completed. Presented here is an excerpt from a National Colorectal Cancer Roundtable, or NCCRT, resource, which was developed to help primary care physicians facilitate conversations with their patients about CRC screening options. When I discuss CRC screening options with my patients, I stress that colonoscopy is the preferred modality, given its ability to reduce death and prevent cancer. I am thrilled when patients elect for colonoscopy. However, many patients choose not to undergo this option as they prefer a non-invasive alternative. Consistent with the NCCRT guidelines, when discussing non-invasive screening options, I highlight that all have the potential to reduce death from CRC. However, education at this point is critical because stool-based options vary in their ability to detect precursor lesions, and thus they have limited ability to prevent CRC. Evidence from the Eclipse study demonstrate that Shield is an effective CRC screening modality with performance in range of available non-invasive stool-based tests. While I acknowledge the concern about the low AA sensitivity, in my experience, and consistent with the NCCRT guidelines, the benefit of these screening options lie in their ability to reduce preventable CRC deaths. Based on the totality of evidence, Shield addresses a critical unmet need by offering a safe, effective, and non-invasive CRC screening test, which should be made available as a choice for patients. As stated earlier, When discussing CRC screening with my patients, I acknowledge that colonoscopy is the prioritized screening option, given its ability to both detect and prevent CRC. However, many patients prefer a non-invasive option. To achieve the guideline-recommended screening targets, it is critical that we promote shared decision-making rather than mandating and implementing barriers that restrict and/or delay access to effective screening options. Offering Shield alongside existing screening methods has the potential to increase screening, bolster adherence, and reduce preventable CRC deaths. And Shield provides a much-needed, innovative option that is convenient, easy and simple, yet is effective and accepted by patients and providers. I'd like to conclude by reinforcing the message by leading healthcare organizations, which aligns with my clinical perspective. When it comes to CRC screening, the best test is the one that gets done. Thank you. I will now turn the presentation over to Dr. Eagle.
Thank you, Dr. Singleton. Good morning. My name is Craig Eagle, and I am the Chief Medical Officer at Guardant Health. Screening is about getting a test done that is right for the individual, since not being up to date with screening is the worst outcome. Shield adds to the choice and increases the success of screening. I would like to remind the committee that the performance of Shield is within the range of screening choices. Shield's performance with CRC sensitivity of 83%, specificity of 90%, has been established with a large, well-designed study. As can be seen here, Shield's performance is within range of other non-invasive CRC screening tests and has the added benefit of being a blood test. The top graph shows the CRC sensitivity compared to other currently used non-invasive screening options. Similarly, the bottom graph shows the specificity across non-invasive devices. Of course, we need to consider additional aspects of a screening device that I would like to summarize in the next few slides. Detection of advanced adenoma as the precursor lesion of most CRCs adds an important component of CRC screening. Advanced adenoma detection and removal impacts CRC incidence. Shield's advanced adenoma sensitivity is on the lower end of the range of widely used stool options as shown in the slide. Colonoscopy is the best test for identifying advanced adenomas and therefore is the best test to truly impact CRC incidence. As a result, the proposed indication for Shield is to detect colorectal cancer. Whilst we have shown this slide previously, I wanted to remind the committee that all these screening devices are used to reduce CRC mortality, and this is across a range of advanced adenoma performance as shown here. This highlights the complexity of assessing one-time advanced adenoma sensitivity. The interplay of one-time advanced adenoma sensitivity in non-invasive tests, including Shield, can impact CRC incidence through a combination of advanced adenoma detection and adherence to repeat testing at fixed intervals. These parameters need to be considered when assessing non-invasive tests and their impact on CRC incidence. We recognize the potential that convenience of a blood-based test may lead to individuals selecting Shield based on ease over other test modalities, in particular colonoscopy. However, evidence supports that adding screening options as primary choice increase overall screening with minimal impact on current screening options like colonoscopy. Presented here are data from the National Health Interview Survey evaluating the impact of screening rates following introduction of CT colonography and MTS DNA stool test over a 15-year period. As shown by the solid blue line, throughout this time, we see a steady increase in the total number of individuals screened. In addition, this study shows that there was no change in proportion of individuals screened with established tests. In particular, there was no decline in colonoscopy rates as shown in the blue dotted line following the introduction of non-invasive tests. Further evidence, more specific to blood testing, has demonstrated an increase in overall screening rates when individuals are given additional choice. Presented on the left is a randomized controlled study which evaluated 381 average risk screening eligible individuals. All participants were contacted by letter and telephone to inform them that they were overdue for screening. Participants randomized to the control group were offered colonoscopy and FIT, and those in the intervention group were given the additional choice of a blood test. The addition of a blood test increases the number of individuals completing screening 1.8-fold, and importantly, the rate of colonoscopy and FIT in the intervention group is similar to the control group. On the right is a separate randomized controlled study. In this study, more than 2,000 average risk screening eligible participants were randomized, with one group offered FIT and colonoscopy, and the other offered the added choice of Shield. All individuals were contacted via telephone. Similar to the first study, a 2.4-fold increase in the likelihood of completing CRC screening was observed with the addition of Shield, without significant test substitution. These studies support the potential benefits of adding Shield to current screening options and suggest that through shared decision-making, test completion rates may increase. The American Cancer Society supports choice and acknowledges shared decision-making is important. I quote, "There is evidence that patients will have a preference for one type of screening test over others if provided sufficient information regarding these test attributes. Although no single test appears to consistently dominate patient preferences, supporting a strategy of offering choice. Intention to screen is also higher if the screening test ordered is consonant with the patient's preference. Therefore, the goal is to allow physicians to employ the test most appropriate for the individual and the one the individual is most likely to complete. Guardant Health is committed to patient and provider education on Shield to facilitate informed decisions. Gardant proposes a strategy that includes physician and provider education to clearly outline the benefits and limitations of Shield, including patient-friendly language on device performance, including advanced adenoma performance, implications of a false positive or a false negative result, the need for repeat testing at regular intervals in people who have a normal signal detected, and the need for diagnostic colonoscopy in those with an abnormal signal detected. Gardant has convened an independent group of communication experts to ensure accuracy and comprehension of educational material. Guardant Health will work with the FDA to define the communication channels to distribute materials to ensure they are leveraged during patient and provider discussions. In addition to facilitating informed decision-making, Guardant Health continues to build data and will complete the Eclipse long-term 1- and 2-year cancer follow-up visits. To date, 92% of participants have completed their 1-year follow-up visit. We are also committed to addressing clinical questions with additional studies and collecting data in collaboration with the FDA, guideline committees, colorectal cancer screening experts, and the community. These could include the long-term evaluation of patients who receive a false positive result, longitudinal adherence to diagnostic colonoscopy, and cumulative PPV to inform the appropriate test interval with Shield. In conclusion, our Eclipse study was designed to determine whether Shield was appropriate as a primary CRC screening test. The co-primary endpoints of CRC sensitivity and advanced neoplasia specificity were met, and Shield's CRC sensitivity is in range of the sensitivity for the current primary non-invasive screening tests. In other words, the 42% of patients eligible for CRC screening who are not up to date on screening may opt to get screened given the option of a blood test. This type of adherence greatly increases the probability of CRC detection, and therefore it has the potential to further reduce CRC mortality. Shield has clinically meaningful performance in range with guideline recommended screening tests across all stages of the disease. Given the convenience provided by a blood test, we believe that the availability of Shield will significantly increase adherence to testing and increase the opportunity to perform screening during a routine healthcare visit. Shield is a test that physicians can feel confident that patients will complete. In real-world clinical practice, patients do not decline stool testing, they just do not complete the test. monitoring completion or non-completion of tests is often infeasible. Moreover, a secondary indication and offering tests sequentially to patients would place access barriers and may generate misperceptions about the effectiveness of this choice. While giving choices are important to improve overall compliance, Guardant Health is committed to work with the FDA on appropriate labeling and provide appropriate educational materials and fact sheets to physicians and patients about the benefits and limitations of Shield. Finally, since this panel last met on CRC screening over a decade ago, we haven't seen the progress we'd hoped in terms of CRC screening rates. And alas, CRC remains the second leading cause of cancer death. Millions of people are not benefiting from the mortality reduction of early colorectal cancer detection. We know that an additional convenient CRC screening option will help us achieve that aspirational goal of 80% screened and bring benefit to individuals who are at risk today. Thank you. We would now be happy to take your questions.
Sorry.
Yes, there we go. Yes, I would like to thank the Guardant Health representatives for their presentation. Does anyone on the panel have a brief clarifying questions for the sponsor? If you can raise your hand, I would really appreciate it. And if you can state your name before speaking to make sure the transcribe picks the names of that. Let's start with Carla.
Hi, yes, this is Carla Ballman. I have like one question, maybe with two parts. It's my understanding that currently there's no data on longitudinal adherence, and adherence for the 90% that was cited was based on having had the test ordered. Did they get the test? It had nothing to do with the propensity of patients to come in on a regular interval if this needs to be done on a regular interval. And then finally, what frequency would be envisioned for this test?
Good morning, and please allow me to introduce myself. My name is Victoria Raymond. I'm the Vice President of Medical for the screening program here at Guardant Health. I've been working on the screening program for the last 5 years, and it's my honor to be able to address these questions and talk through the results of the Eclipse study with you today and the opportunity we have for blood-based testing to improve screening rates. Dr. Ballman, to address your question, when we think about adherence and the way we've described adherence today, it is for that one-time adherence. So for those individuals who have been offered a test, did they actually complete the test? And the numbers that were shown for Shield and for the other non-invasive screening tests are aligned to that one-time screening adherence. As Dr. Eagle mentioned, longitudinal adherence is one of the data gaps that we look forward to pursuing in post-approval studies. For your second question about the interval of screening tests, as we think about the interval for screening tests for the stool-based tests that are currently available, we know that interval is somewhere between 1 to 3 years. While the screening interval for Shield has not yet been defined, given that the Shield performance is within range of the other colorectal cancer screening tests that are available today, we anticipate that interval would also be between 1 to 3 years. And we look forward to working with the guideline committees to make sure we're understanding the right timeline to offer that repeat testing.
Thank you.
Thank you. Stephen Hewitt?
Yes, this is Dr. Stephen Hewitt. Can you address the specimen requirement for the assay? Currently, in the documents I've read, it says 4 tubes or 4 Streck tubes. And I'm assuming that those are 10 mil Streck tubes. Is there a boundary or a disqualification if the sample is inadequate? And how does one determine that? And what is one incurred? Because looking at the Eclipse data, a larger volume across most of the patients was drawn.
Yes, so for the Eclipse data, we did collect additional blood, and in our commercial ordering, we do request, as you mentioned, about 4 tubes of blood. To complete the assay, we actually only need about 2 to 3 tablespoons of blood, or about 4 to 8 milliliters.
Thank you. Alexander Borowski?
Yeah, just a general question. Dr. Liang presented a 23% stage 4 at detection. And I'm curious to know specifically what's known about the difference in that number in a screen compliant versus non-compliant, and or mortality rate in a screen compliant versus non-compliant. And at the root of my question is sort of the underlying knowledge that not all colon cancers are created equal. And sort of a secondary question is one that may be hard to answer, which is the mortality benefit of colonoscopy the result of harvesting many and any and all potential precancers, even though many would not progress? Or is it more in the detection and treatment of invasive carcinomas before they have that opportunity to metastasize? And again, we know that many of them actually are metastasizing at very early time points. Thank you.
Great. To address your question about what's known about stage 4 in symptomatic versus screen detected individuals, and then understanding the landscape of colorectal cancer screening benefit from incidence reduction from advanced polyp detection versus early detection of asymptomatic cancer, I'll invite Dr. Leung.
This is Dr. Peter Liang. So as you mentioned, the slide we showed here showed that of individuals who were diagnosed with metastatic cancer, the 23% are diagnosed at that stage, and the prognosis is very poor, 16%. We are not able to differentiate whether these are screen detected versus diagnosed symptomatically. So I don't have specific answers in terms of whether the prognosis is different based on kind of those how it's detected and whether symptoms were present at the time of diagnosis.
And then for the second part of your question, which is about incidence versus early detection, it's known that both of these are key aspects of any colorectal cancer screening test. And really, ultimately, the goal is a reduction in CRC-related mortality, which can be achieved either way through a reduction in incidence from adenomas or from early detection of asymptomatic cancer. It's known that if individuals are diagnosed with stage 1 or stage 2 colorectal cancer, that 5-year survival rate is upwards of 90%, as Dr. Liang showed. So it is a combination of both.
Thank you. Mark Gilbert?
This is Mark Gilbert. The presentation was very clear and very comprehensive, but thank you. My question is, in false positive detection of the shield, what are you actually detecting?
Yep. So the false positive detection of Shield, I would invite our Chief Technology Officer, Dr. Chudova, to comment.
Good morning, Daria Chudova, Guardant Health. You're raising an interesting point about what are the false positives. So for any screening program, clearly we expect to see some level of false positives. And so the question when the assay is methylation-based and detects system-level status from a blood draw, is there anything specifically we know about the nature of the false positives? So there's a couple of factors that are important in considering that. One is there evidence for co-occurring incidental non-CRC malignancies? And I believe Dr. Chang addressed that in his presentation by pointing out that in the 1-year follow-up data, if we can bring that slide back, in 1 year, thank you, follow-up data for over 6,000 individuals, we did not observe an increase in non-CRC malignancies within our false positives in comparison to true negatives. And as a reminder, the data collection is ongoing for a 2-year follow-up of those individuals. The other reason that we've discussed also in the presentation is the trend for increasing false positive rate with age, and so that's probably a reflection of the methylation as the endlight for assessment of the signs. And so these two factors probably should be taken into account when counseling patients on positive results.
Thank you very much. Charity Morgan?
Hi, Charity Morgan. You stated that when showing the participant disposition, that about 300 subjects had a Shield test result that was not valid. Can you comment on whether this rate of invalid tests is in line with other non-invasive procedures?
So when we look at the rate of invalid test results, it ends up being somewhere around 2% of samples that were invalid, and that's exactly what we're seeing in our laboratory developed test experience as well. When you look across the landscape of non-invasive colorectal cancer screening options, taking FIT as an example, it's thought to be a target of less than 5% invalid test result return is proposed for programmatic screening.
Thank you. Speaker Singh?
Good morning. I think I sort of have two clarifying questions. The first is, how did you define advanced neoplasia and advanced adenoma? I see that throughout your presentation you're using the acronyms ANAA. It may be helpful to know exactly how you made the differentiation between the two, because in the colorectal cancer literature, I know that sometimes different studies can label these as different. And the second question is, among the patients, I guess, who were screen detected to have the higher stage cancers, stage 2, 3, and 4, how many of those patients had symptoms such as abdominal pain or anemia and so forth? Because I think this then sort of underscores the fact of how well a screening test is performing versus a patient simply presenting with symptoms that leads their provider to believe that they have colorectal cancer.
Great. I'd like to invite Dr. Chung, the study PI, to address your question, defining AN and AA in the study, and then also clarifying the inclusion-exclusion criteria.
Daniel Chung. So to clarify the definition of the terms advanced neoplasia and advanced adenoma. So advanced adenoma refers specifically to those precancerous lesions that have a certain size, have certain high-grade dysplasia, or villus components. Advanced neoplasia is a term that's reserved for either an advanced adenoma or cancer. And so when we set up the study, we set it up the way that most studies in colon cancer screening have been designed in terms of defining specificity for advanced neoplasia, which includes both colorectal cancer as well as advanced adenomas. With respect to your second question about symptoms that patients may have experienced, so as part of our eligibility criteria, patients who are eligible only if they were having screening for screening colonoscopies without any symptoms. And so none of the patients that were detected, even at late stages, were individuals who were having exams done with symptoms associated with that.
Thank you for that. Dr. Padma Ragajapal?
Yeah, sure. I'm Padma Rajagopal. Thank you so much for the presentation. In the documents provided, there was an analysis that was provided of sensitivity and specificity by age across different brackets. And while the Eclipse study did include multiple racial populations, there weren't similar breakdowns that were provided that I observed about the performance of the test across multiple racial groups. Of course, there may have been some limitations in power to that end, but as it's possible that there may be differences in methylation patterns across racial groups that can reflect both environmental and ancestral contributions. Do you have any further information about test performance across these groups?
Eclipse was powered for overall CRC sensitivity and specificity, and that was what the result was. That's what the study was powered for. But we did see some interesting trends in these subgroup analyses, as you mentioned. And to comment on that, I'd like to invite Dr. Chung.
Daniel Chung, yes, so we were very interested in making sure that we enrolled individuals throughout our cohort of different ethnic backgrounds, and we were very pleased that we were able to enroll high percentage of patients who were identified as Black, African American, Asian, as well as Latino. And so we did do, I did show a slide earlier that, let me just pull this up here, where we did look at the performance for CRC sensitivity as well as advanced neoplasia specificity among the different races and ethnicities, and we found that they all performed similarly across each of these groups. As was mentioned, we didn't have the study wasn't powered to look at each group individually. However, we did not see any trends that should notice any differences among each of the ethnic groups.
Thank you. Carla Bauman?
Yes, this is Carla Ballman. Just a couple of quick questions. I'm not sure if you know the data. It's my understanding that the incidence rate of colorectal cancer from individuals with advanced adenomas is about 5% per year. Is that correct? And then secondly, what is the sojourn time from a stage 1 colorectal cancer to a stage 3?
Great. So I'll invite Dr. Liang to come and comment a bit about what we know and don't know about the sojourn time for stage 1 to stage 3 CRC. For the kind of transition rate for advanced adenomas to colorectal cancer, the best data we have shows about a 2 to 5% annual rate. So up to 5, but 2 to 5% is what the data cite.
This is Peter Liang. So this is the slide we shared earlier in the presentation that with respect to your question about sojourn time from a preclinical colorectal cancer to clinical colorectal cancer, it's estimated to be about 4 to 5 years. This does not break it down to stage specific, so I can't, based on this data, tell you what it is for stage 1 to stage 3. I want to emphasize that this data is the same data used by the U.S. Preventive Task Force to use in their modeling to guide their guideline recommendations.
Thank you. Dr. Stephen Hewitt?
Thank you. This is Stephen Hewitt. Returning to the issue of pre-analytics. In the Eclipse study, the patients were substantially selected for inclusion in the study, which is appropriate. However, as we've experienced and learned over many years, use of a new device or diagnostic in a wide spectrum requires you to gain information about the pre-analytic variables, oftentimes endogenous concerning diseases and situation of the patient that would affect the test. Has Gardant obtained any information related to the performance of this assay in, say, a woman who is pregnant, a woman who has endometriosis, or a broad spectrum of autoimmune disorders which may result in increased cellular turnover? As noted, IBD was excluded. However, rheumatoid arthritis, diseases of the liver, and any number of other autoimmune disorders result in substantial turnover. In your presentation, you only mentioned the methylation assay aspects of it and not the fragmentation aspects of the assay. Thank you.
Great. To address your questions about pre-analytical studies, I'd like to invite Dr. Chudova.
Thank you, Daria Chudova. I will start with your initial question about blood volume and related QC metrics. Just to clarify, we do have specific pre-analytical QC metrics for both blood volume and plasma volume that allow specimens to be processed. Now, in the pre-analytical world, we've conducted a significant number of studies evaluating the reactivity of the test to various endogenous and exogenous conditions. And that data is being reviewed by the agency, both for the primary device performance characteristics, as well as validation of the tube that is part of our blood collection kit. More specifically to your question about interfering substances, we evaluated a number of analytical spike-ins that are recommended in the guidelines and did not observe sensitivity of the assay to any of these parameters. And we've also evaluated performance of the device across a number of potential comorbidities. That was done using close to 3,000 patient samples from the intended use population. And we did not observe cross-reactivity with any other disease except for liver conditions, which, as you cite, is known to provide more significant cell turnover and probably releases significant cfDNA amount in the circulation. That data is part of our review process with the FDA as well, but this is a pretty comprehensive evaluation of the comorbidity conditions that we have.
Thank you. Dr. William Burge?
Thank you. It's my understanding that your assay underwent development over several years, if not many years. And I understand that there may have been a change in performance characteristics over time. Can you tell us a little bit about the different assay systems? And did all the patients and all the samples get run with one assay, or was it based on early assay systems?
Mhm. I will invite Dr. Chudova to comment on the assay systems and the testing plan.
Thank you, Daria Chudova, Guardant Health. We have performed all of the analyses that are reporting cfDNA device performance in the Eclipse study with a single version of the cfDNA part of the device. It has not changed from the time it was locked prior to sample testing to the time as we know it today, and we will go with that assay into the commercialization as well. The device change that was referred to in our briefing documents excluded an independently operated protein component, but it did not in any way, shape, or form impact the either sample testing in the wet lab or the analytical pipelines in terms of classification scores for the cfDNA component of the assay. So all of the data reviewed has one device by one assay and one set of parameters reported across all data.
Thank you.
Thank you. Dr. Lovespring?
Yes, Edward Lawspring. I am serving as a consumer representative on this panel, and there has been no mention of cost. Have there any been studies done? Because cost can be a barrier too, between the cost difference between your test and the stool sample. And that's my question. Thank you.
It is my understanding that cost is not a topic of today's discussion or within the purview of the FDA, but I defer to this topic to the FDA.
Yes, I would think at this time is not the purview of these discussions to talk about the cost of the test or the utility. We're looking at safety and efficacy of that. So at this point, we are going to take a break, 15-minute break, and we reconvene at 11:15. Thank you very much, everybody, for reconvening. We will now proceed to the FDA presentation. I would like to invite the FDA representative, Dr. Yu Han, to begin. The FDA representative will have 60 minutes to present. You may begin now your presentation.
Good morning, everyone. Welcome to today's Molecular and Clinical Genetics panel meeting for the Shield test from Guardant Health. Today, Dr. Anand Pathak and I will be summarizing the FDA's review of this pre-market application for the Shield test. I'll start with a brief introduction for myself. My name is Yu Han, and I'm a scientific reviewer in the Division of Molecular Genetics and Pathology in the Office of In Vitro Diagnostics. I'm the lead reviewer for this pre-market application, or PMA. First, I would like to acknowledge that the review of this PMA submission has involved the work of many individuals from different offices and divisions across the center. Dr. Pathak is the medical officer, and Dr. Kondratovich and Dr. Garcia are the statistical reviewers. Other areas that were reviewed include analytical studies, software, and manufacturing. Shield is the first blood-based screening device for the proposed intended use. Based on the test performance of Shield, FDA is seeking panel input on the safety and effectiveness of this first blood-based screening device. We're also seeking input on whether the benefits outweigh the risk of using this device in the context of the proposed intended use. So, as we continue through our presentation, please consider the panel discussion questions as provided in the panel package. The FDA presentation will be presented in 3 parts. In the first part, we will provide background for the Shield test. Dr. Pathak will present the background information on colorectal cancer and advanced adenomas, hereafter referred as CRC and AA respectively. I will follow up with a brief description of the device, including the proposed intended use, along with the proposed contraindications, as already introduced to you by the sponsor. I will then provide a brief overview of the device workflow and summarize the analytical studies reviewed by the FDA to support the approval of this device. In the second part, I will present the pivotal clinical study that was conducted to support the safety and effectiveness of this device for its intended use, including the clinical study design, patient accountability, primary and secondary effectiveness results, predictive values, and other statistical analysis. In the third and final part, Dr. Pathak will discuss key aspects of the clinical studies as they relate to the FDA questions for panel discussion. He will then present FDA considerations for discussion questions. Now, Dr. Pathak will start the first part of our presentation with background materials for CRC and AA in the next few slides.
Hello, my name is Anand Pathak, and I'm a medical officer at the FDA Center for Devices Division of Molecular Genetics and Pathology. It is a great honor for me to be presenting to you today. I'll be presenting some background material here first, and I will be presenting part 3 of this presentation on review considerations. Here are some points about colorectal cancer. There are about 150,000 cases annually in the US, with approximately 50,000 deaths. CRC is also the second leading cause of cancer deaths in the US annually. It is important to know that detecting CRC early may benefit the public health, as localized CRC has an approximately 90% 5-year survival rate, while metastatic CRC has only a 15% 5-year survival rate. Appropriate screening and surveillance strategies may mitigate morbidity and mortality from CRC. Finally, I would like to add that randomized control studies with sigmoidoscopy and FOBT have previously shown significant reductions in CRC mortality compared to the no intervention arms. Also, the data from the sigmoidoscopy RCT also showed significant reduction in CRC incidence. Now I present some background on advanced adenomas. First, one must note that the majority of CRCs arises from colonic adenomas, and advanced adenomas can progress to cancer at an annual rate of up to 5%. Individual factors such as age and advanced adenoma factors such as size, histology, and degree of dysplasia can influence the risk of progression to CRC. For example, large adenomas and adenomas with high-grade dysplasia or greatest villus component are more likely to progress to overt CRC. In addition, larger sessile serrated lesions and sessile serrated lesions with dysplasia are more likely to progress to CRC. Finally, detection and removal of advanced adenomas can reduce the incidence of CRC and the morbidity and mortality associated with CRC. Now in this slide, I would like to discuss the follow-up surveillance of adenomas according to the Multi-Society Task Force, or MSTF. One important aspect of follow-up after detection of adenomas is the programmatic surveillance of patients after colonoscopy. Patients are triaged into certain intervals of surveillance follow-up based on the size of the adenoma, the histology of the adenoma, the number of adenomas, and other factors. For example, patients with adenomas with high-grade dysplasia are recommended for follow-up surveillance colonoscopy 3 years later. These surveillance strategies are integral to patient management and may forestall the progression of precursor lesions to overt CRC. Next on this slide, I would like to present some key information on CRC screening in the United States. First, I must note that approximately 1/3 of screen-eligible patients do not undergo screening for CRC, which is a curable disease if the cancer is detected early. In addition, 75% of people who died from CRC were not up to date with screening. Next, I note that the target for CRC screening is 80% in the US, according to the American Cancer Society and the National Colorectal Cancer Roundtable. Thus, there is room for improvement in CRC screening update in the US. Increased screening rates may translate into a significant reduction of CRC-associated morbidity and mortality. Now I would like to discuss CRC screening guidelines according to the USPSTF. The USPSTF evaluates the benefits and risks of screening tests, and their recommendations are evidence-based. The task force recommends a variety of modalities for screening, including endoscopic procedures, stool-based methods, as well as CT colonography, and does not currently recommend serum, urine, or capsule-based methods due to limited data. Finally, the task force recommends considerations be given to the variables listed below by physicians and patients when determining which test is best for each patient. These variables listed on this slide include the frequency of screening needed, the access to screening, the risks associated with the screening procedure, the ability of the patient to complete the pre-procedure bowel preparation, the ability of the patient to undergo anesthesia or sedation, and finally, the risk of follow-up procedures for abnormal findings. Now on this slide, I continue to discuss aspects of CRC screening guidelines from the USPSTF. Mainly, I note that a variety of testing strategies are currently recommended by the USPSTF with specific time points for repeat testing. These include high sensitivity guaiac fecal occult blood tests, or fecal immunochemical test FIT every year, or stool DNA FIT every 1 to 3 years, or CT colonography every 5 years, or flexible sigmoidoscopy every 5 years, or flexible sigmoidoscopy every 10 years plus annual FIT, or colonoscopy screening every 10 years. Repeat screening at the specified time points improves the programmatic performance of these tests over the lifetime. Now I would like to discuss the USPSTF screening guidelines with respect to age. First of all, CRC screening by these guidelines are indicated for patients 45 or older who are at average risk for colorectal cancer and do not have signs or symptoms of colorectal cancer. Next, I note that the USPSTF screening recommendations have varying degrees of strength depending on the age groups. For adults aged 45 to 49 years, the task force has a grade B recommendation due to moderate net benefit. For adults aged 50 to 75, the task force has a grade A recommendation due to the substantial net benefit. For adults aged 76 to 85, the task force has a grade C recommendation due to the small net benefit. In this age group, the task force instructs to selectively screen adults for CRC, considering the patient's overall health, prior screening history, and patient preferences. These age-based distinctions in the task force recommendations are important to highlight because of age-based differences in the performance of the Shield device. Finally, to conclude this background section, I would like to present a statement from the American Cancer Society. Screening with any one of multiple options is associated with a significant reduction in CRC incidence through the detection and removal of adenomatous polyps and other precancerous lesions, and with a reduction in mortality through incidence reduction and early detection of CRC. Thus, screening and detection of both CRC and adenomatous polyps and other precancerous lesions are considered to contribute to the reduction of CRC incidence and ultimately clinical benefit through a reduction in mortality. It is important to recognize the significance of the reduction of CRC incidence as one of the outcomes of available screening options. Now, Dr. Yu Han, the lead reviewer on this submission, will continue presenting the remainder of Part 1 and Part 2 on the clinical studies.
As you have seen in the sponsor's presentation, the proposed intended use for Shield is as follows. The Shield test is a qualitative in vitro diagnostic test intended to detect colorectal cancer-derived alterations in cell-free DNA from blood collected in Guardant Blood Collection Kit. Shield is intended for colorectal cancer screening in individuals at average risk of the disease, aged 45 years or older. Patient with abnormal signal detected may have colorectal cancer or advanced adenomas and should be referred for colonoscopy evaluation. Shield is not a replacement for diagnostic colonoscopy or for surveillance colonoscopy in high-risk individuals. The test is performed at Guardant Health INC. Shield test is not suitable for everyone. The sponsor has proposed the following contraindications. The test is not indicated for an individual who has a personal history of CRC or has a family history of CRC defined as having one or more first-degree relative, or has a known hereditary germline risk of CRC, or has a known diagnosis of an inflammatory bowel disease. A number of precautions and limitations have also been proposed by the sponsor to be included in the product labeling. I will highlight some key proposed limitations as listed here. First, the Shield test should be considered alongside other CRC screening modalities, like colonoscopy, and is not a replacement for diagnostic colonoscopy or surveillance colonoscopy in high-risk individuals. Second, Shield has limited ability for the detection of advanced adenomas. Third, screening for CRC is recommended for people over 45 years old, and providers should discuss the most appropriate test to use with patients, depending on their medical history and individual circumstances. Shield is a next-generation sequencing-based qualitative test to detect genomic and epigenomic alterations in cell-free DNA isolated from blood. The Shield test workflow includes several steps. First, whole blood is collected in Gardant Shield Blood Collection Kit, which is part of the Shield test. Plasma is then isolated from whole blood collected. After that, the FDA is extracted from plasma and processed for DNA sequencing to detect methylation pattern, fragmentation, and genomic alterations. Lastly, the cfDNA data is analyzed using proprietary bioinformatics algorithms designed to detect the presence of colorectal neoplasma-associated signals. In terms of the device panel and algorithm, the Shield test integrates the signal from 3 different analyte types to predict the presence or absence of circulating tumor DNA, including somatic mutations, methylation, and fragmentation pattern. The cfDNA sequencing data generates 4 results output from 4 different colors: somatic mutation color, fragmentomics color, methylation LR color, and methylation MR color. Signals detected from these 4 colors are combined into the integrated score, and the signal from methylation corner is evaluated independently to generate MR score. The MR score and integrated score are compared to predefined cutoffs to generate positive or negative results for each cfDNA MR call and integrated call. If either of integrated call or MR call is positive, then the Shield result is positive. A negative Shield result only occurs when both the cfDNA integrated call and MR call are negative. This result classifies samples as either abnormal signal detected or normal signal detected. The analytical performance of Shield was demonstrated with the following non-clinical studies. The blood collection tube was validated for specimen collection for use with the Shield test. Analytical sensitivity was assessed to determine the limit of detection. Analytical specificity includes cross-contamination and carryover studies, in silico primer and probe specificity, as well as interference testing. Cross-reactivity with non-colorectal cancers and diseases were also evaluated. Several studies, including repeatability, reproducibility, plasma isolation equivalence, reagent lot-to-lot interchangeability, were performed to assess the precision of the Shield test. Robustness studies evaluated acceptable tolerance ranges for critical parameters and different failure modes. Additional studies included sample stability, reagent stability, instrument evaluation, and software. The data from above-mentioned studies was provided and reviewed by FDA. The analytical data will not be discussed during the panel meeting. Now I will discuss the clinical study conducted by sponsor to support the safety and effectiveness of Shield test. In the second part of our presentation, I will be presenting the clinical study design, patient accountability, primary effectiveness result, age-adjusted device performance, and additional statistical analysis that were conducted to evaluate the performance of Shield. The name of the Eclipse study refers to evaluation of ctDNA lunar assay in an average patient screening encounter. This study was a registrational study to evaluate the performance of the Shield test to detect colorectal cancer in average-risk adults. We would like to note that the test was originally named Lunar 2 at the time of the clinical study and was renamed to Shield at the time of the PMA submission. This Eclipse study began enrollment in October 2019, and the data cutoff date is September 2022. A total of 24,876 subjects were enrolled from 265 sites across the U.S. Note that the enrollment was enriched with patients aged 60 to 84 years old, which account for 63.6% of patients in the study. The study was designed to collect cross-sectional data, meaning that data was collected from population at one specific point in time. Patients were required to have colonoscopy within 183 days of blood sample collection. Blood collection was performed prospectively from all enrolled subjects prior to the patient undergoing colonoscopy and were processed and analyzed at Guardant Health. performance of the Shield test was compared against the colonoscopy results. Here are the inclusion criteria for Eclipse. Since the sponsor has previously summarized inclusion and exclusion criteria, I will just highlight some key aspects. This study only enrolled patients that are at average risk for CRC, aged 45 to 84 years old. I would also like to note that the time window between blood draw and undergoing colonoscopy was extended to 6 months due to the pandemic. These slides listed the abbreviated exclusion criteria. I will highlight some of the key exclusion criteria here. Basically, patients were excluded if they had any conditions that were considered by a physician or healthcare provider as being of high risk for CRC, with family history of CRC, with personal history of any malignancy or any high-risk conditions for colorectal cancer. Also, patients that had undergone CRC screening using other recommended screening methodologies within specific time window were excluded as well. As I previously mentioned, colonoscopy results were used to determine disease status of the study subjects. The lesion of greatest clinical significance was used to classify each subject into one of the histopathological categories listed in the table below. Category 1 is for colorectal cancer stage 1 through 4. Category 2 is for advanced adenomas. Categories 3 through 5 are for non-advanced adenomas. And category 6 for negative or no neoplastic findings. The sponsor considered category 1 as CRC and the category 2 as AA. Subjects in category 3 through 6 were considered as non-advanced adenomas, also called non-AN. Non-ANs were included in the specificity analysis. To evaluate the performance of the clinical study, the sponsor pre-specified the following primary and secondary objectives. The first primary objective is CRC sensitivity, and the lower bound of the two-sided 95% confidence interval for the Shield CRC sensitivity shall be above 65%. The second primary objective is AN specificity, and the lower bound of the two-sided 95% confidence interval for the Shield AN specificity shall be over 85%. The secondary objective was to establish the sensitivity of the Shield test in the detection of advanced adenomas in average risk patients. Now I will discuss the Eclipse study population. A total of 24,876 subjects were enrolled in the Eclipse study, and a number of subjects were excluded from the primary effectiveness population. First, 1,999 subjects from a pre-specified enrollment time window were used towards the device development and were excluded. Second, 10,179 subjects were randomly selected not to be screened with the Shield test. The remaining 12,698 subjects for clinical validation included all CRC subjects and a proportion of non-CRC subjects selected through random downsampling to match U.S. Census age distribution. Third, of the 12,698 remaining subjects, 2,401 subjects that did not meet study inclusion-exclusion criteria or did not have valid colonoscopy within 183 days or did not have valid Shield result were excluded. Lastly, of the remaining 10,297 subjects, 2,436 that were randomly selected for interim specificity analysis and the cutoff selection were excluded from the pivotal clinical validation dataset. The total number of patients in the final clinical validation evaluable dataset consisted of 7,861 subjects with valid colonoscopy and valid Shield test results. In the 7,861 subjects, there were 65 CRC, 1,116 AA, and 6,680 non-AN. Considering the exclusion of subjects due to interim analysis and cutoff selection, FDA performed subset analysis to evaluate the potential for bias, which could have been introduced due to either the device modification that were made during the clinical study or assignment of subject to different datasets. FDA concluded that the sensitivity and specificity data presented here did not create favorable bias to the performance. As we mentioned previously, the CRC sensitivity, AA sensitivity, and AN specificity were evaluated in the clinical study. CRC sensitivity was calculated as the proportion of patients in histological category 1 who test positive. AA sensitivity was calculated as the proportion of patients in histological category 2 who test positive. And the AN specificity was calculated as a proportion of patients in histological categories 3 to 6 that had a negative test result. The clinical effectiveness data can be understood in the following way as shown in bottom table. Basically, the Shield test result were compared to the clinical truth of the patient to evaluate the ability of the test to correctly classify diseased and non-diseased subjects. Results in the primary effectiveness population are summarized in this contingency table. In the table, the Shield binary test result is cross-classified by histopathological categories of CRC, AA, and non-AN. For the primary performance measure of CRC sensitivity, the estimate is 83.1%. The two-sided 95% lower confidence bound was 72.2%, which is greater than the Gardant's performance goal of 65%. For the primary performance measure of AN specificity, the estimate was 89.6%. The two-sided 95% lower confidence bound was 88.8%, which is greater than Gardant performance goal of 85%. The estimate of the secondary performance measure of AA sensitivity is 13.2% with a lower bound of 11.3%. Now I will present the performance of the Shield test with regards to age. This table shows the CRC sensitivity, AA sensitivity, and AN specificity in 5 different age groups. This data indicates that the AA sensitivity increases with age, while AN specificity decreases with age. AA sensitivity is 3.6% in the 45 to 49 age group and increases to 33.3% in the over 80 age group. The specificity decreases from 95.5% in the 45 to 49 age group to 75.5% in the over 80 age group. The performance estimates presented in this slide are not precise for specific age groups due to small sample size. For example, there are only 4 CRC cases in the 45 to 49 age group and only 1 CRC in the 80 plus age group. Because of a small sample size in the low and high age groups, 3 age categories were considered when evaluating potential differences in the Shield test performance with regards to age. Group 1 is for patients between 45 to 59 years old. Group 2 is for patients between 60 to 69 years old. And Group 3 is for patients aged over 70. For CRC sensitivity as shown in the top table, differences in sensitivity were not statistically significant as 95% confidence intervals are overlapping between age groups. For AA sensitivity as shown in the middle table, it appears there is a trend of increasing the sensitivity of AA with increasing age. Sensitivities increased from 7.9% to 15.1% between group 1 and 2, and then 95% confidence intervals are not overlapping. For AN specificity as shown in the bottom table, there appears to be a tendency of decreasing the AN specificity with an increase in age. The decrease in specificity was statistically significant as all three 95% confidence intervals are not overlapping. Since the performance of the Shield test is different for three age groups, the age distribution should be considered in the calculation of the overall sensitivity for CRC, overall sensitivity for AA, and overall specificity for AN. As shown in this slide, the age-adjusted overall performance was calculated based on age distribution in U.S. Census population in 2020. After age adjustment, CRC sensitivity is 80.8%, AA sensitivity is 12.9%, and the specificity for AN is 89.5% as shown in the right column in the red box. The conclusions are the same as for the unadjusted estimates. Shield performance was also evaluated through its predictive values. The positive predictive value, PPV, for CRC indicates the fraction of patients with CRC among the patients with positive Shield test result. The PPV for AA indicates the fraction of patients with AA among the patients with positive Shield test results. The negative predictive value, NPV, for CRC indicates the fraction of patients without CRC among the patients with negative Shield result. The NPV for AN indicates the fraction of patients without CRC or AA among the patients with negative Shield test results. This table lists the prevalence of CRC, prevalence of AA, percent positive Shield results, PPV for CRC, PPV for AA, NPV for CRC, and NPV for AN in all 5 age groups. It appears that the prevalence of CRC is increasing with an increase in age from 0.24% in the 45 to 49 age group to 0.96% in the 80+ age group. Also, the percent of positive Shield result is increasing with an increase in age from 4.58% in the 45 to 49 age group to 25.81% in the 80+ age group. PPVs for CRC in different age groups range from 2.21% to 3.93%, and PPVs for AA in different age groups range from 5.76% to 14.65%. Please also note NPVs for CRC are above 99.8% for all age groups. In other words, the percent of subjects with CRC among subjects with negative Shield result is ranged from 0.06% to 0.16%. This analysis was conducted by FDA and was also provided by sponsor upon FDA request. Additional analyses were performed to evaluate the Shield test performance within subgroups, including subgroups defined by disease stages and lesion characteristics. However, the subgroup analysis should be interpreted with caution, since the pivotal study was not designed to evaluate the performance of the test in subgroups. Although no attempt was made to adjust for multiplicity, these analyses are useful to consider. This slide shows the CRC sensitivity stratified by cancer stage. the detection of stage 1 CRC is 54.5%, where the detection of CRC in later stages, stage 2, 3, and 4, is 100%. I would like to note that there are 5 malignant polyps that are not fully staged in the stage 1 calculation. Therefore, stage 1 sensitivity may be summarized as 64.7% when excluding those 5 patients. When calculating CRC sensitivity by lesion size, it appears that the Shield test failed to detect CRC lesions that are less than 10 millimeters. And there is a trend that the detection of CRC increases with increasing lesion size. This slide presents the assessment of AA sensitivity by lesion size. Similarly, a trend of increasing the sensitivity of AA was observed with increasing lesion size, from 0% in AA less than 5 millimeters to 23.6% in AA over 30 millimeters. AA sensitivity stratified by histopathological subcategories was also evaluated. Data in this table shows that the detection of AA in different histopathological subcategories varies between 0% for advanced adenomas, carcinoma in situ of any size, to 22.6% in high-grade dysplasia. AN specificity was also analyzed by different histopathological subcategories. It appears that the point estimate of AN specificity was slightly higher in category 6 compared to categories 3 through 5. Now, Dr. Patek will present part 3, review considerations.
I will now be presenting summative points around FDA review considerations and the discussion questions. First, I present key summative points related to CRC detection by the Shield device. Shield can detect 83% of CRCs from a non-invasive blood test. However, it will miss 17% of CRCs. Shield has a 17% false negativity for CRC, and saliently, all CRCs missed were stage 1. The sensitivity for stage 1 CRC was 54.5%. Of note, stage 1 sensitivity may be summarized as 11 over 17, or 64.7%, excluding those 5 CRC patients in the stage 1 calculation that were not completely staged. Also, the Shield test failed to detect all CRCs that were less than 10 millimeters in size. It appears that the Shield test cannot detect small CRCs, though the numbers here were small, only 6 CRCs in this category. However, I must note that the Shield sensitivity for stage 2, 3, and 4 CRC was 100%. Now I present the summative points for advanced adenoma performance. The Shield test detected approximately 13% of advanced adenomas, and this test missed approximately 87% of advanced adenomas. Also, the detection of advanced adenomas varied between 0 to 22.6% in different histopathological subcategories. Of note, Shield detects 22.6% of advanced adenomas with high-grade dysplasia and 17.9% of advanced adenomas with a villus component. These histologies represent more aggressive types of advanced adenomas that are more likely to progress onto CRC. Now I present summative key points regarding the PPV and the NPV of the Shield device. First, the PPVs for CRC in different age groups range from 2.21% to 3.93%, and was 3.10% overall. Next, the PPVs for advanced adenomas in different age groups ranged from 5.76% to 14.65%, and it was 12.04% overall. Next, it is important to note that the overall NPV for CRC is 99.92%. Thus, at the population level, this test can reassure the majority of patients testing negative that they do not have CRC. Also, the overall NPV for advanced neoplasia is 89.86%. So 1 out of 10 patients will be falsely reassured that they're negative for advanced adenoma. However, only 1 out of 1,000 patients will be falsely reassured that they're negative for CRC. Now I will present the FDA review considerations as they relate to the discussion questions. Here I present the top line review considerations for discussion questions 1, 2, and 3. For discussion question 1, related to Guardant's proposed claims, I note that the CRC sensitivity was 83.1%, the advanced adenoma sensitivity was 13.2%, and the advanced neoplasia specificity was 89.6%. For discussion question 2, related to advanced adenoma performance and potential mitigations, I note that the advanced adenoma sensitivity was only 13.2%. And for discussion question 3, related to the needs of a post-approval study, I consider what are the benefits and risks of programmatic CRC screening with repeated testing over certain intervals. For question 1, related to the benefits and risks of the SHIELD test and considering the appropriate population for the use of this test, I will present background on the performance of approved non-invasive CRC screening tests, the performance of recommended CRC screening tests by guidelines, the performance of the Guardant SHIELD test, and finally, a discussion of the impact of adherence rates will be presented. In this slide, I present background on the CRC in vitro diagnostic landscape. There are several existing FDA-approved devices for CRC screening in the average risk population for developing colorectal cancer, for example, Cologuard and EpiProColon. Some in vitro diagnostic CRC screening tests, such as the exact Cologuard test, may be considered first line, which are indicated as primary screening options for individuals at average risk for CRC who are typical candidates for CRC screening. EpiProColon, however, has a different claim that may be considered second line and is indicated for individuals at average risk for CRC who decline recommended screening methods such as colonoscopy or other first-line CRC screening tests. FIT tests are authorized by the FDA for the detection of hemoglobin in stool and do not explicitly have FDA authorization for CRC screening. Some clinical practice guidelines, such as from the USPSTF, recommend use of FIT tests for CRC screening. Performance of several FDA-approved devices for CRC screening, namely Cologuard and EpiProColon, are considered on this slide. In addition, the published performance of FIT and high sensitivity FOBT are considered on this slide. Estimates of sensitivity for CRC and advanced adenoma, along with two-sided 95% confidence intervals, are provided in this table. Specificity estimates for patients without CRC or AA, along with two-sided 95% confidence intervals, are also provided. Cologuard, a stool-based test, had a sensitivity of 92.3% for CRC and a sensitivity of 42.4% for advanced adenoma, with a specificity of 86.6% for patients without CRC or AA. EpiProColon, a blood-based test, had a 68.2% sensitivity for CRC and a 22% sensitivity for advanced adenoma, with a specificity of 78.8% in patients without CRC or AA. The performance of FIT tests for CRC screening has been reported in multiple publications. For example, in a meta-analysis of 19 studies with one-time FIT screening, in the asymptomatic average risk populations, authors reported that the pooled sensitivity of FIT was 79% for CRC with a specificity of 94%. Similar results were reported in a study by Imperial et al. In New England Journal of Medicine publication 2014, in which FIT sensitivity for detecting colorectal cancer was 73.8% and specificity was 94.9%. Also, advanced adenoma sensitivity of FIT was 23.8% in this study. We also note the performance of high sensitivity FOBT. High sensitivity FOBT has a CRC sensitivity of between 50 and 75%, and AA sensitivity of between 7 and 21%, and a specificity of between 96 and 99%. Gardant's proposed indication for the SHIELD test is for colorectal cancer screening in individuals at average risk of the disease, most similar to a first-line claim. Here, I present the SHIELD performance for patients 45 or older and for patients 50 or older, since prior original FDA approvals for CRC screening devices were for patients that were 50 or older. In the 50 or older age group, the CRC sensitivity is 83.6%, the AA sensitivity is 13.7%, and the non-AN specificity is 89.0%. The key summative points about the SHIELD test are presented on this slide, including sensitivity, specificity, PPV, and NPV, which have already been discussed. In addition, I note that the PPV for CRC and advanced adenoma is 15.14%, given that the specificity of the test is greater than 89%, and the balance of the false positives to the true positives for this test is 5.6 to 1. I will now comment on adherence rates, which can affect the probable success of a screening strategy. On 10,000 clinical orders, the SHIELD LDT showed a 96% adherence rate. This high adherence rate may have been influenced because of early adopters opting for this test. A real-world estimate of adherence has yet to be demonstrated. Regardless, the adherence to the SHIELD test is likely to be higher than for colonoscopy and other recommended CRC screening tests. Given this information, this test may fill an unmet need for patients who are non-compliant with CRC screening, but willing to take a non-invasive blood test. The consequences on other patient populations preferentially opting for the SHIELD test over other screening alternatives should also be considered. Now I present additional background for discussion question 2. In terms of advanced adenoma background, we note that the majority of CRCs arise from colonic adenomas. Advanced adenomas can progress to cancer at an annual rate of up to 5%, depending on a variety of factors. Detection and removal of advanced adenomas can reduce the incidence of CRC and reduce the morbidity and mortality associated with CRC. Detection of advanced adenoma by the Gardant SHIELD test is 13.2%. The detection of advanced adenoma vary between 0 and 22.6% in different histopathological subcategories. SHIELD detects 22.6% of advanced adenomas with high-grade dysplasia and 17.9% of advanced adenomas with a villus component. These histologies are more aggressive types of AA that are more likely to develop into overt CRC. What are the considerations for advanced adenoma detection performance for a blood-based CRC screening test? Finally, now, I present some points and background related to discussion question 3 related to the need for a post-approval study. First, the SHIELD test missed 17% of CRCs, and all CRCs missed were stage 1. Approximately 45% of stage 1 CRCs were missed, and all CRCs smaller than 10 millimeters were missed. Next, the NPV for CRC was 99.92%. So 1 out of 1,000 patients testing negative with Shield would be falsely reassured that they're negative for CRC. However, I note that the NPV for advanced neoplasia, CRC or AA, was 89.86%. So 1 out of 10 patients testing negative by this test would be falsely reassured that they do not have CRC or AA. The patients falsely reassured would be predominantly patients with advanced adenoma. In addition, for reference, the following are examples of the currently recommended USPSTF repeat testing intervals for various test types. FOBT or FIT is recommended every year. Stool DNA FIT is recommended every 1 to 3 years. Flexible sigmoidoscopy is recommended every 5 years. And colonoscopy screening is recommended every 10 years. Establishing repeat testing schedules increases the cumulative sensitivity of these screening tests. For Shield, the one-time performance is 83% sensitivity for CRC, 13% sensitivity for advanced adenoma, and 89% specificity. This concludes Section 3 of the FDA presentation on substantive points around FDA review considerations and the discussion questions. I would like to thank you for your attention. We are now open for questions. Thank you.
I would like to thank the FDA for their presentation. Does anybody on the panel have a brief clarification questions for the FDA? And please state your name again for transcription purposes. Dr. Morgan?
Hi, Charity Morgan. I have a question about the use of the word trend on slides 39 and 40. You were talking about there being a trend towards increased sensitivity or trend towards decreased specificity. Was a statistical test performed to determine whether there was a trend, or are you speaking more generally about there being a pattern that was noticed?
Hi, I'm Donna Roscoe. I'm the acting division director. The trend refers to a pattern, although we do consider the statistical significance of the overlapping confidence intervals in the various situations.
Dr. Gilger?
Just a simple question, point of clarification. On slide 51, the EpiPro is currently FDA approved or not?
It is.
Very good. Thank you.
Dr. Bruce?
Yes, just a follow-up question on the EpiPro. It was FDA approved, but I understand that it's been withdrawn from the market, so it's no longer available. Does the FDA have any insight as to why that happened?
I'm not able to offer any insight into the status of the EpiPro test.
But it's FDA approved.
It is FDA approved. And the point of the slides that Dr. Pathak made was that it is approved for patients who are non-compliant with other screening modalities. They must first, in conjunction with their physician, choose to forego those other tests.
And that's also true of Shields.
That is not true with Shield at this time. It is part of the panel discussion questions.
And this is one of the questions that they have been posed by the FDA.
Right.
Dr. Morgan?
Yes, Charity Morgan again. I'm not sure if this is something you can comment on, but can you provide some guidance on to, I guess, some clarification about why EpiPro was approved as second line versus first line? Was that decision made primarily due to the performance or other characteristics? Is that something that you can comment on?
Yes, EpiPro was approved as a second line indication because the specificity was inferior to FIT, and the sensitivity was determined to be non-inferior to FIT, but had 4 percentage points increased sensitivity. So the sensitivity of the fit in the study was determined to be 68% and 72% for epiprocolon, but the specificity was inferior to fit.
So inferior specificity, non-inferior sensitivity.
Right.
Thank you.
Dr. Bauman?
Hi, Carla Ballman. Just a question of clarification. So the FDA, when evaluating approval, only looks at the performances such as sensitivity, specificity as given, and does not weigh in terms of the frequency of what the test schedule should be for surveillance.
So you generally do not consider the longitudinal analysis in the original PMA review, but do consider it and ask the sponsor to develop studies for it in the post-market setting.
And for what I remember, there was not long-term study. There was only 1 year follow-up from the patients that they were enrolled, correct?
Gardant is currently conducting study data across out to 2 years, I believe. Okay. They presented that in their slides.
But for clarification, they're not being retested. It's just following these patients for 2 years, right?
I would like to defer to Gardant on the specificity of the study design, but they're testing them again at 1 year and 2 year intervals.
The Representative Gardant, could you clarify?
Yes, so if I can share a slide, please. So we are following patients observationally for 2 years following their blood draw. They are not retested again as part of the Eclipse study, but we are following that 1 and 2 year clinical outcomes.
Okay, thank you. That's very helpful.
Thank you.
Dr. Hewitt?
Yes, this is Stephen Hewitt. With reference to the second-line EpiPro assay test, was there documentation required from the patient when they were deferring first-line screening? Was that placed in their medical record, or was this just a discussion with the physician?
Can you repeat that question?
Was documentation required at the time when patients refused first-line screening and decided to pursue second-line screening based on the EpiPro?
So for the epiprocolon study, this decision to have a second-line claim was after the study was completed and based on the data. So they were not required to do that during the study.
For patients to use the second line, was there documentation that they had refused first-line screening?
So the process for epiprocolon is that physicians may need to document.
Thank you.
Dr. Spencer?
Hi, this is Sean Spencer from Stanford. I had a question whether the FDA can comment on the coverage analysis that the Center for Medicare and Medicaid Services made in regards to blood-based biomarker testing. This was referenced to in the executive summary, but not discussed today. And I was wondering if the FDA has made any analysis of that or whether the panel should consider it when thinking about the questions.
CMS made an announcement about their reimbursement policies for blood-based testing performance, and they indicated that sensitivity should have a minimum of 74% and specificity should have a minimum of 90%. FDA does not consider reimbursement in the review of the diagnostic devices.
And the reimbursement or even the cost should not be part of in our discussions.
Right.
That's a great question. Thank you because it's a very good clarification to steer our discussions more into the analytical and the clinical performance of the test. Any other questions for the FDA? Dr. Rajapal?
Yeah, Padma Rajagopal. I was just wondering if the FDA can comment on if there were with any of the other prior tests that have been FDA approved, any specifications that were made in the context of stage 1 cancers, if there was any limitation in term or performance or decrease in performance that were noted for stage 1 cancers, if there was any specification made for other tests in the past.
So Are you asking what the performance for stage 1 cancer was in the other approved answer?
I'm asking if there was any labeling or something provided by the FDA
No,
for those other tests.
there's no limitation regarding the stage 1 performance. However, the labeling does indicate the performance in stages. FDA also has a summary of our review called the Summary of Safety and Effectiveness, and that data is in the SSED.
Thank you.
Any other questions for the FDA? Well, thank you very much to the FDA for all the presentation and the answering the questions. We will now break for lunch. I will ask panel members, please do not discuss the meeting topic amongst yourself or with any other members of the audience during the lunch. We will resume with open public hearing sessions at 45 minutes from now. So at 1.
Okay, we're clear.
Thank you.
Hello again. It is now 1:05 p.m. and I would like to call this meeting back to order. At this time, we will proceed with the open public hearing portion of the meeting. Public attendees, we are given an opportunity to address the panel to present data, information, or views relevant to the meeting agenda. Mr. Swink will now read the open public hearing disclosure process statement.
Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency during this open public hearing session of the Advisory Committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement to advise the committee of any financial relationships that you may have with any company or group that may be affected by the topic of this meeting. For example, this financial information may include a company's or a group's payment of your travel, lodging, or other expenses in connection with your attendance at this meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. Thank you.
Thank you, Mr. Swin. Prior to the final date published in the Federal Register, the FDA received 17 requests to speak. We will begin with the presentations with Girish Pucha from Precision Medicine and Diagnostics, LLC. Mr. Pucha?
Yes, thank you very much. Let's go full screen. Okay, good afternoon and thank you for this opportunity. My name is Girish Pucha and I'm a molecular genetic pathologist by training. Here are my disclosures, but the opinions expressed here are my own. First, I want to address the concept of average risk. CMS defines this in NCD 20-210.3 as an individual with no personal history of adenomatous polyps or colorectal cancer. And USPSTF defines it as those who do not have symptoms of CRC and do not have increased risk factors for the disease, including a prior diagnosis of CRC or adenomatous polyps. In Eclipse, participants were only excluded for a personal history of CRC, but included if they were still somehow considered by a physician or healthcare provider as being of average risk. One, but not the only indicator that this and other inclusion and exclusion criteria materially increased the risk profile of participants is that there was a 21% increase in stage 3-4 CRC versus deep C. While some of this may certainly be attributable to stage shift created or related to the delayed screening due to the pandemic, this still represents a 17% increase in stage 3-4 CRC over CRC Prevent, another pivotal study performed during the pandemic. Going forward, I'd ask for greater transparency, consistency, and rigor in defining this population among the FDA, CMS, professional societies, and clinical guideline bodies, as well as characterization of the clinical performance in the actual intended use population. Next, I'm perplexed to say the least about the complete absence of hyperplastic polyps in the summaries from either the sponsor or the FDA, especially since these are clearly considered to be clinically significant lesions by guideline bodies such as MSTF and are more clearly delineated in other pivotal studies, such as this example from Blue Sea on the right. Since the categorization of these lesions could impact not only AA sensitivity, but also non-AN specificity, I again request greater transparency, consistency, and rigor in defining such clinically important subtypes within non-CRC categories, as this would improve comparability among studies and is critical for various end users, such as modelers, clinical guideline bodies, and so forth. Finally, I want to address the tiering of tests. For reasons that are unclear to me, though I'm sure we can all speculate, clinical guideline bodies, most notably USPSTF, have recently avoided ranking tests. Indeed, the most recent such ranking I could find is this one from 2017 from MSTF. This has led to the cliche that the best test is the one that gets done, even though this clearly flies in the face of CMS's non-coverage of epiprocolon and its lack of recommendation in any clinical guideline body to my knowledge. Regardless, I just wanted to highlight for the panel and the agency 2 recent modeling papers that seek to address the effectiveness and cost-effectiveness of blood-based CRC screening tests. Since I know cost-effectiveness sits outside the agency's purview, I will focus on conclusions related to effectiveness alone. Both papers modeled a blood test meeting the minimum performance criteria stipulated by CMS in its NCD, but also did various sensitivity analyses, especially related to adherence and adenoma sensitivity. Like all models, these have their limitations, but can still be useful, especially when they're consistent. They both found that adherence and AA sensitivity materially impacted test effectiveness far more than CRC sensitivity. Perhaps the most useful to the questions being posed to the panel are these figures from the papers, which show the effectiveness of different screening strategies, including blood tests. Based on benefits alone, both models show the same hierarchy of tests. But when one considers both risks and benefits, with risks measured as it usually is in these models by the number of lifetime colonoscopies and benefits in terms of life years gained, the hierarchy differs based on which model is used, which reinforces why accurate apples-to-apples assumptions for clinical performance in the intended use population and adherence in that same population are so critical. Thank you.
Thank you. We will now proceed with the pre-recorded OPH speakers. The first speaker is Dennis Barnes.
My name is Dennis Barnes. I am 55 years old. I live in Raleigh, North Carolina, and I am a husband, a father, a son, a nephew, a cousin, and a friend. I have worked with Guardant Health in the past on patient education efforts, but I am not being paid for my time here today. I wanted to share my story with you because I believe that it is fairly common. As a professional with a time-consuming job, I found it difficult to fit a colonoscopy into my schedule. Although my primary care provider pleaded with me to get screened around my 50th birthday, for the next 3 years, I consistently failed to get it done. Part of the issue was my schedule, but frankly, part of the issue was the unpleasantness of the experience. I just didn't want to be bothered with everything that comes with having a colonoscopy. And the home kit process also requires some unpleasant steps. For 3 straight years during my annual health checkup, my primary care physician asked me whether I had completed a colon screening, and each year I had to admit that I hadn't done it. At that point, he told me about a clinical trial that was being conducted where I could be screened for colon cancer with a blood test. He gave me the details, and when he was done, I knew that this made sense for me. I could finally get this task completed right then and there by simply giving blood. Screening for all types of cancer is extremely important because we know that cancer is largely asymptomatic for years, and by the time most people exhibit physical manifestations of the disease, it's already too late. This is particularly challenging in communities of color because we participate in preventative health care at much lower rates than others. We tend to wait until we are actually sick before we see a doctor. So I have made this part of my life's mission to get the word out that we must screen for cancer in as many ways as are available. And it is important that there are methods of accomplishing this task that are easy and convenient. There is no question in my mind that screening for cancer will save lives. The more screening that can be accomplished, the greater the number of lives that will be saved.
Good morning. My name is Dr. Andrew Albert, and I am a practicing gastroenterologist who also holds an advanced public health degree in health promotion and disease prevention. Imagine looking the patient in the eye after performing a colonoscopy and having to give bad news that they have colon cancer, a disease process that could have been avoided. And then imagine having their follow-up question, which is, how did this happen? And the inside part that you can't say out loud is that this could have been prevented if you had only been checked sooner. This happens every single day. Early detection of colorectal cancer plays a huge role in preventing further disease and improving patient survival. Our current testing strategies aren't working. You talk about a poop test, and unfortunately patients shut down the conversation. You talk about colonoscopy, and people get overwhelmed by taking a day off from work or having to think about the associated sedation with the procedure. Imagine a new world of testing for colon cancer through a blood test. Now we're cooking with gas in the world of preventative medicine. No procedure, no stool test to perform, but a test that makes patient compliance even easier. We can even do this test outside of the office, but you can't do a poop test or a colonoscopy in this manner. Guardant Health's Shield test has a clinically meaningful performance and a high compliance rate, which is exactly what we need to offer our patients. 50 million Americans remain unscreened today. Even if I opened up my hours to do colonoscopy every day of the week from 9 to 5, I could not accommodate the need of all these people to get screened for colon cancer. The Shield test can be administered in various settings and allows better access, and we need to offer this option to our patients. I envision this test being as useful as a routine cholesterol test. Cholesterol won't kill my patients tomorrow, but colon cancer will. Additionally, this test will allow me to scope smarter and dedicate my resources to those who need the procedure most. Imagine my doing a colonoscopy on a Shield test that's positive versus the same patient over and over again looking for hyperplastic polyps that would never be an issue. This is where it's so practical and so useful. Typically, the wait times for colonoscopy are 6 to 9 months. Having the Shield test available will make this wait time much shorter. I hope you will support gastroenterologists like me who want to save lives using a test that we can all be comfortable with, using a test that will have amazing implications for the years to come and save the lives of those who are at risk for colorectal cancer. Thank you so much for your time.
Hi, my name is Candace Henley, and I'm the founder of the Blue Hat Foundation. I want to address a critical aspect of public health that affects us all, but notably impacts our communities of color: colorectal cancer screening. This disease is a major cause of mortality, yet it is largely preventable with timely and consistent screening. However, it is deeply concerning that access to these life-saving screenings is not uniform across all communities. African Americans face significantly lower screenings, with about 38% undergoing routine colonoscopies. This disparity is a glaring indicator of the broader social determinants of health that many communities contend with, issues that we at the Blue Hat Foundation are deeply committed to addressing. Our engagement with the community has brought to light the various barriers that prevent individuals from participating in CRC screenings. These barriers range from cultural barriers to stigma associated with procedures like colonoscopies, to practical challenges such as transportation difficulties, limited availabilities of appointments, and the absence of a consistent medical home. These are not just logistical issues, they are profound reflections of inequalities that pervade our healthcare system. Recognizing these challenges, we advocate for more inclusive and accessible screening options such as blood-based screening. This innovative approach can be integrated into a routine blood draw, making it a less invasive and more convenient option for those who might otherwise remain unscreened. It's a method that respects individuals' preferences and circumstances, facilitating a pathway to screening that many may find more acceptable and approachable. Blood-based screening means that it is seamlessly fitting into the lives of those who we serve. It enables us to reach individuals right where they are in a manner that respects their time and their needs. This aligns perfectly with our mission at the Blue Hat Foundation, where we strive not only to listen to the community, but to act where we hear, breaking down the barriers to access, enhancing the understanding of the importance of CRC screening. The beauty of this method lies in its simplicity and its ability to meet people where they are. By integrating the screening into a routine blood draw, we can significantly increase participation rates. The principle is clear. The best test is the one that gets done. If a screening method is a convenient and accessible way for more people to likely use it, then it in turn empowers healthcare providers to advocate for a more vigorously screening option to improve the overall screening rates and consequently improve health outcomes. While traditional methods like the colonoscopy continue to be the gold standard in CRC prevention, it is crucial to support and adopt additional screening options that cater to the diverse needs of our population. Blood-based screening is not just an alternative, it is an imperative step towards equity in healthcare, ensuring that every individual, irrespective of their background or circumstances, has access to the necessary preventive measures. Let us embrace this opportunity to make a significant impact on public health and move towards a future where colorectal cancer is no longer or can no longer claim lives prematurely due to underscreening. Thank you so much.
Hello, my name is Mary Beth Kropp, and I really appreciate you taking a few moments to listen to my video. My husband Michael passed away from colorectal cancer in December 2021. The date is still so close for me, and I apologize right up front if I will become emotional during this video. I want to share a picture of him with you. This is Michael right here. Actually, on the day that we met the team from Guardant Health. When my husband was re-diagnosed, we decided that we needed to be more educated about colorectal cancer, so we dove in headfirst and we opened up an opportunity in California to organize the Climb for the Cure from Fight CRC, an organization that I've grown to love and trust. But see, the problem is not very many people understand about colorectal cancer. So without folks like CRC, I never would have met the folks at Gardant. Because they came to support our event at Mount Tamalpais in San Rafael, California, not even ever meeting our family, we were truly impressed by their commitment to really help everyone understand the complexities of colorectal cancer. I do believe the Shield test is a game changer. I'm sorry, here it comes again. So just please understand that from the bottom of my heart, we have to do something about colorectal cancer. Our organization, Big Mike's Bottom Line, is now just committed to educating people before they have to go through what my family went through. And a test like the Shield test would be such a game changer for everyone, because many people would opt for a blood test versus a colonoscopy or another kind of invasive test, just like my husband did. My husband ignored his symptoms when he was in his early 40s. And when he was first diagnosed at the age of 43, we had no idea what colorectal cancer is. So with the help of so many people, especially the folks with Gardant, Fight CRC, and other organizations that are committed to preventing this disease from becoming any more of a crisis, we truly applaud their efforts and urge you to support this, because I do believe it will change the way that we screen for colorectal cancer. Our quest is to educate as many people as possible. We know so many people, so many Americans, so many Californians where I am are not up to date on their screening. In fact, only about 40% of them are up to date here in California, and I know the number is even larger in other places. With a test that could be done simply with a blood screening, I think many more people would be apt to really understand that this 100% preventable disease could be diagnosed much sooner and prevent families just like ours from going through what we did. So in my last few seconds, I want to applaud you for considering this, and please understand I am representing many families, and I know that you're going to do the right thing, because we really need some more support in this whole prevention world. Thank you for taking the time to listen. Thank you for hearing us, and please approve the Shield test.
Hi, my name is Dr. Robert Azrin. I am a family medicine doctor here in Southgate, California, and the other providers here at my clinic, along with myself, have been performing Guardant Shield tests since September, October of 2023. There have been several challenges over the years with colon cancer screening, but more so since the COVID-19 pandemic. Particularly, gastroenterologists are just completely backed up. On average, patients are waiting 4 to 6 months to get an appointment. That's just for their initial consultation. Sometimes you're waiting for 2 to 3 months after that just to have the colonoscopy. With FOBTs, patients often complain that they don't want to do it, or they just pile up the kits because they're too lazy to do them and bring them in, or they think collecting it is just gross. With that said, patients who have abnormal FOBTs are just left waiting and anxious because they can't get in to see the specialist. And they say, here I am, unable to have here I am with a positive FOBT test. You're telling me I might have colon cancer and I can't see the specialist for 6 months. Now with the Guardant Shield test, it's been a complete game changer for us. We have yet to have any patient tell us no when we offer them the exam. But the only barrier really has been that there's no FDA approval to do this test. So we have to do our due diligence and go by HEDIS guidelines, offer them colonoscopies, FOBTs, Cologuard screening first, give them adequate time to fail that or not complete those things, and then we can offer them the Guardant Shield test. As a personal experience, I do have an uncle who came in around December complaining of being unable to use the restroom. He had gone to the ER once, was discharged home, came to see me. He lives in Riverside County, we're here in LA County, and I referred him to a gastroenterologist and then did a Guardant Shield exam on him. He, shortly after seeing me, ended up back in the ER, was admitted, had a colonoscopy, and was found to have colorectal cancer. About 2 or 3 days after his hospital discharge, we got his Guardant Shield test back, and it was positive. We currently have about 4 or 5 other patients that are positive on Guardant Shield, but unfortunately, they are still waiting to have their initial GI consultation so that they can have a colonoscopy done. If Guardant Shield were to get approved, I think it would be a complete game changer in the way that we approach colorectal cancer screening, and I couldn't advocate for it more. Thank you.
I'm Jody Hoyos, CEO of the Prevent Cancer Foundation. I'm here to talk about the potential of blood-based screening options as a tool in the fight against colorectal cancer. I wish to disclose that the foundation has received funding from Gardant and other industry partners to support education and outreach on cancer prevention and early detection. We have not been compensated for these comments. When it comes to colorectal cancer, early detection equals better outcomes. Catching cancer early can mean more treatment options, less extensive treatment, and better chances of survival. Unfortunately, although we have really effective screening options for colorectal cancer, not enough people are getting screened and they are missing the chance to catch cancer early. In the Prevent Cancer Foundation's 2024 Early Detection Survey, 42% of US adults age 45 and older reported they were not up to date or were not sure if they were up to date on their routine colorectal cancer screening. And one of the top reasons reported or cited for why they are behind on their screening was nervousness about the screening examination. The early detection survey results also revealed nearly a third of people aged 45 and older who are not up to date or not sure if they were up to date said a different or less invasive screening test would make them more likely to prioritize their colorectal cancer screening. We want people to take charge of their health and get checked for cancer. And giving them more options has the potential to help us achieve that. Thank you very much.
My name is Patricia James. I'm 75 years old and live in Temecula, California with my husband. I am the proud mom of 1 daughter and an even prouder grandmother of 3 beautiful grandchildren. Today, I'm sharing my story with you as a Shield user because I feel lucky to have the chance to take the test, and I want to make sure many others get the same opportunity. I came to the United States from England when I was 9 years old, and soon after arriving, my mom died from cancer. Losing her at a really young age obviously shaped a big part of who I am today and prompted me to focus a lot on health and prevention throughout my life. I wanted to make sure I could be there for my own family for as long as possible and that I didn't miss out on all the things my mom unfortunately did. Despite being generally proactive when it comes to my health, I admittedly didn't start screening for colon cancer till I was 59. To put it simply, I kept putting it off because I just didn't want to do it. I didn't like the options that were available to me. And when I did finally go through with that first colonoscopy, I had a very bad experience with it, as I had suspected I would, and refused to do it again. In 2013, I was diagnosed with fallopian tube cancer. I'm relieved to say that I am now more than 10 years cancer-free, but again, this experience forced my doctors and me to pay closer attention to my body and to prioritize the importance of cancer screening and prevention. When my doctor called me and asked if I'd be open to Shield, I immediately said yes, which was a very different response compared to what I had given him when he asked about me getting another colonoscopy. I mean, after going through something like cancer, a needle in my arm is nothing. The best part was I was able to get it right after my mammogram. I didn't need an extra appointment. It was remarkably quick, and it actually took longer to drive there than it took to get the blood drawn. The level of convenience is a game changer for someone like me. Even though I'm tired, I hardly have a minute to spare during the day. I'm on the board of our homeowners association. I'm the chair of the landscape committee, and I regularly participate in Bible studies. I swim, read, and revel daily in afternoon walks. I'm a strong believer that being physically active is important to keeping my mind and body healthy. At the end of the day, I'm a busy woman, and Shield made it so that I could get screened for colon cancer. Without it, I wouldn't have been screened at all. On top of this, as someone who is 75 and is not only adverse to colonoscopies, but understands that colonoscopies can become more challenges to do as we age and may not even be recommended by doctors after a certain point. Shield is a great alternative to help me stay on top of my colon health for longer. It will end up helping me and others screen past the time we ordinarily would be able to do a colonoscopy alone. Look, I don't ever want to mess around with my health. I've lost people to cancer, and I battle it myself. I know how precious life is, and I'm someone who wants all the time in the world to keep living. I have sung the praises of this test to friends and look forward to getting it again. I hope the FDA sees the importance of Shield and having it as a screening option, especially for people like me who have busy lives and are uncomfortable with other screening options. Patients like having choices and playing a role in their health decisions. I know I do, and I think a blood test could enable 4 more people to get screened than are currently being screened today. More screening options will result in more screening getting done, which will hopefully transform into better health outcomes for so many. Thank you for allowing me the time to share my story and thoughts today and for considering my perspective. I really appreciate it.
Hi, my name is Thiago Biaki. I'm a medical oncologist here at Moffitt Cancer Center in Tampa, Florida. I've been practicing medical oncology for the last 12 years, only GI medical oncology for the last 12 years. I'm focused on colorectal cancer and liver cancer in my clinical practice and my research activities. And I'm here today to support a blood-based test for screening for colorectal cancer published this year at New England Journal of Medicine. As you know, colorectal cancer is a pandemic in this country. We have 150,000 new cases per year. And mortality for those patients older than 60s coming down, but unfortunately mortality for those young patients with colorectal cancer is coming up. We have seen this trend in clinic. I would say 20% of those patients coming for treatment now, they are younger than 50. And of course, as you know, screening is the most important step here to diagnose this disease early enough to be able to recommend curative treatments. Of course, the main barrier for colonoscopies is logistics. Actually, we know that the adherence for colonoscopies is around 50% in US, which is far from the ideal scenario. Actually, I don't believe that we have GI facilities to do colonoscopy in everybody who needs a colonoscopy. This is the first step, first point. Of course, fecal-based tests are helpful. Any non-invasive procedure can help to guide this surveillance, can help to raise awareness for colorectal cancer. Even fecal-based tests can cause some logistics issues, and I think the main benefit of a blood-based test is incorporating this test in the annual physical and for any adult. It caught my attention and the study that sensitivity was really high, mainly for those patients with stage 2, 3, and 4 colorectal cancer. Of course, for pre-malignant lesions, this is not ideal. And of course, this kind of a test is not going to substitute a colonoscopy, which is the only exam that can remove polyps and can locate the tumor. Of course, this is important for patients with localized disease. But I think this is I truly believe that this is the first step towards, I would say, a broad blood-based test for screening, not only for colorectal cancer, but maybe for other tumors in the future. And I think this test is going to be really helpful if we can incorporate this in practice. That's it.
Hello, and thank you for allowing me the opportunity to provide comments today. My name is Andrew Spiegel, and I am the CEO of the Global Colon Cancer Association. We are a nonprofit umbrella organization of all colon cancer groups around the world. I've been in the colon cancer space for more than 25 years. Back in the 1990s, along with a group of other people, I helped co-found the first colon cancer patient advocacy group in the United States, now called the Colorectal Cancer Alliance. And I work across all countries now in trying to get more patient groups formed and make sure that colon cancer patients are at the center of policy decisions being made around the world. The idea of introducing a new screening method, blood-based testing, has been a dream of mine for decades. And that is because I have witnessed firsthand what it was like when there was no screening at all happening in the United States compared to now where you have screening rates in the 70s. But that still means there's a long way to go. We know that there's tens of millions of Americans who still have not been screened for colon cancer, despite all of the widespread availability of all the different tests that are out there and all of the work that's been done to raise the awareness. Still tens of millions of people who should be getting screened for colon cancer are not. And we know that the reason for that is that the barriers to the current screening methods prevent people from getting screened. You simply have people, many of them in underserved communities. We have very high rates of colon cancer in the Black and brown communities, in the Hispanic communities, and that's because they refuse to get a colonoscopy. They don't want to handle their feces in the stool-based testing. And imagine if you could introduce blood-based testing where they simply go to the healthcare provider who draws a vial of blood and they can know whether they have colon cancer at an earlier stage. We know that the majority of people who are diagnosed with colon cancer in the United States and around the world have not kept up with their colon cancer screening. This is an unbelievable dream come true for me as an advocate for colon cancer, that we would be able to introduce a screening method that is so easy and so non-objectionable to people that really there would be no more excuses for skipping colon cancer screening. This type of screening has not only the ability to save millions of lives in the United States, but as everyone knows, other regulators around the world look to the FDA and rely on the FDA for approvals in their own country. So imagine in other countries that don't have the ability to provide colon cancer screening in a cost-effective way, where they can't do mass colon cancer screening, they certainly could afford to do blood testing and analysis. So I really hope that we can move this innovative procedure forward and we can get this test on the market to people who need it, and we can start really getting the colon cancer screening rates to where they need to be. And thank you very much for allowing me the opportunity to provide my comments.
Hi, I'm Trevor Maxwell. I'm from Cape Elizabeth, Maine, and I've been living with stage 4 colon cancer since 2018. I was 41 years old when I was diagnosed. I'm the founder of Man Up to Cancer, a nonprofit organization that inspires men to connect and avoid isolation during the cancer journey. For full disclosure, Man Up to Cancer receives grant funding from Guardant Health to support our annual men's cancer retreat. I'm not being compensated for this testimony, and I would never receive compensation for doing this. I speak to you today as a young onset colon cancer patient on behalf of my nonprofit community of more than 2,500 people, and most importantly, on behalf of the thousands of people who die each year from CRC and their grieving families. I'm here to advocate for the approval of the Gardant Shield blood-based screening test for colorectal cancer. I'm sure you know the statistics related to the rise in young onset CRC. It's already the leading cause of cancer death for men under 50 and the second leading cause of cancer death for women under 50. This has prompted the USPSTF, ACS, and others to lower the recommended screening age to 45. However, millions of people between 45 and 50 are not aware of these trends, or they do not follow through with having a colonoscopy, or a colonoscopy is not offered to them. According to Fight CRC, 80% of Americans between 45 and 50 are not up to date on screening. This is where a blood test can make a huge difference. For those who don't want a colonoscopy or face significant delays in getting one, they can take a blood test. Imagine a 45-year-old man, father of 3 kids, who declines colonoscopy and a cancer is already developing and he doesn't know it. Without any screening, this cancer will continue to grow and spread until he develops symptoms and most likely presents at an emergency room with stage 4 CRC and a terminal diagnosis. Imagine how different that scenario gets with a blood test option. Instead of doing nothing, he takes the blood test, which identifies a stage 1 or 2 colon cancer, and he is cured with surgery, keeping him alive and with his family for years to come. CRC is one of the cancers that can be identified and cured in early stages and is almost always fatal in stage 4. We need to identify this cancer when it's curable. I also want to touch briefly on male behavior and cancer screening. We know adherence rates are low for colonoscopy. One contributing factor is that some men don't want to go through procedures involving the anus or rectum, such as colonoscopy or sigmoidoscopy, based on masculinity norms. For men, I expect the adherence for a blood test will be far superior to currently available screening methods. That will bring down the overall mortality rate from CRC. To sum up my thoughts, there's no question that a blood-based screening test will save lives that are being lost to colorectal cancer. Thank you for the opportunity to testify.
My name is John Gormley. I'm 76 years old. Live in Newport Beach, California, with my wife, and we have 2 grown children. I've worked with Guardant Health on patient education efforts in the past, and today I'm here to tell you my story. So about 3 years ago, I was diagnosed with stage 2 colon cancer. And despite my diagnosis, I feel myself very fortunate because we caught the colon cancer early. We caught the colon cancer early, in fact, early enough that I wasn't experiencing any symptoms. I actually went to see my primary care doctor, Dr. Greg Robertson, for an unrelated issue. While I was there, he looked at my chart and said, you know, it's time for you to get a colonoscopy. You haven't done that since 2003. So he gave me an option. Either I could get a colonoscopy or I could do the Shield blood test right there in his office. I knew a colonoscopy was an invasive procedure that would require me to see a different doctor, take time off work, go under anesthesia, have someone else drive me around. The Shield blood test, on the other hand, would take about 30 seconds. I felt it was an easy choice. Dr. Robertson called me a few days later with the results, which came back abnormal. Soon after a colon cancer diagnosis was confirmed, my doctor immediately scheduled me for surgery. And because the cancer had been caught early, the recovery was quick. I was back to work in a week or 10 days. As scary as it was to be diagnosed with cancer, I knew I was lucky because I happened to go to the doctor that day and was fortunate to have someone like Dr. Robertson who was aware of this screening process. I was lucky to have access to a test that was so simple to do and caught my cancer. I'm here today because I believe other patients should have access to this type of technology. I know that the case of the Shield blood test played an early role in my diagnosis, and it's part of the reason I'm able to go on with my life. Thank you for allowing me this time today and considering my perspective.
Hello, I'm Dr. Len Lichtenfeld, a medical oncologist and strategic advisor based in Atlanta, Georgia. And I would like to note at the beginning that I have served as an advisor for Guardant Shield and have been compensated for my services. In the past, I have managed the American Cancer Society Cancer Control Science Department, which was involved in the prevention and early detection of cancer, including the guidelines you're well familiar with. I also was Deputy Chief Medical Officer and Chief Medical Officer at the Society before I left several years ago. We're here today to talk about colorectal cancer screening, and I'm a firm believer that, obviously, in the value of prevention and early detection to reduce the mortality and burden of colorectal and other cancers. I'm also a firm believer
in the concept that the best test is the one that you're willing to get. And the problems that we have today, and we again are all familiar with that, is that notwithstanding the substantial progress we've made in reducing the burden of colorectal cancer in this country, the reality is that we still have a long way to go. There are a lot of reasons why people don't get screened for colorectal cancer. Some of them are personal. Some people don't want to get the tests that are available. Some people don't like certain aspects of certain tests. Certain people can't access a test. Certain people, for equity reasons, don't have that access, which is an unfortunate feature of our healthcare system in this country. I personally have had experience with issues, and I lived in a rural area, even getting my own screening colonoscopy. So whatever the reasons may be, the reality is that even with the tests we have available today, we have not been as successful as we want to be, and we're not as successful as we can be in reducing the burden of colorectal cancer. And therefore, having other tests such as Guardant Shield could potentially improve opportunities and options for clinicians to discuss with patients and for patients to take advantage of. And particularly a test that is available, including on an opportunistic basis, which is difficult to do today as we're having this conversation, could really reduce the mortality. And an interesting fact is that we've increased the number of people eligible for screening in this country. And we've had difficulty with resource allocation. We don't have sufficient GI folks in adequate locations around the country to do screening. And we've got to be attentive to that. And in fact, the mortality curves for some groups are beginning to flatten. So I, for one, do believe that we need to have tests that patients will accept, that they'll incorporate, the clinicians will incorporate into the care process to discuss with patients in the hope of continuing decreasing the burden of colorectal cancer in this country. And I do believe that Guardant Shield helps meet this need. Thank you, and I appreciate your attention and your time today.
Hi, my name is Winora Johnson, and I'm a stage 3B colorectal cancer survivor. While I have no evidence of disease to date, reoccurrence poses a real threat to me due to my Lynch syndrome status. While I have worked with Guardant Health in the past, I have not been compensated to provide my comments here. I share them today as someone who has battled colorectal cancer, lost family members to colorectal cancer, and someone who knows the toll that this disease can take on the lives of those it impacts. I'm sharing my comments today as someone who wants desperately to see progress in our fight against it so that more lives can be saved. Colorectal cancer disproportionately affects communities of color, particularly Black Americans. And some of this is due to lower screening rates. For instance, routine colonoscopies screenings are as low as 38% among Black Americans. This is due in part to a variety of different factors that include cultural barriers, stigmas around colonoscopies, transportation challenges, limited schedule availability, and social determinants of health. The prospect of Guardant Health's Shield test being a new screening option for patients makes me excited and hopeful. A more accessible screening modality that could easily be ordered as part of a standard routine blood draw has the potential to reach patients who would otherwise remain unscreened. Blood-based screening could help get more patients engaged in the screening process, especially those patients who have trouble accessing a colonoscopy or who don't follow through on their doctor's recommendations to get one. It can conveniently reach people where they are in a way that works better for them. And when you know better as a patient, you do better as a patient, as knowledge is power. If you have a screening test that works, and that people are willing to use. It has the potential to empower physicians to push harder for their patients to screen and significantly improve upon colorectal cancer screening rates. After all, as they say, the best test is the one that gets done. More screening options can result in more screening getting completed and better health outcomes. So blood-based screening could be a valuable tool in the fight against colorectal cancer. And no one knows this better than me.
Hello, my name is Dr. Gregory Robertson. I'm a board certified internal medicine physician in California. I've worked with Guardant Health on patient education efforts in the past, but I'm not being compensated for sharing my thoughts today. For background, I'm not only a prescriber of the Shield, but a user of it as well. And I'm thrilled to have the opportunity to tell you about the positive impact it's had on my practice in just a short time that it has been available. Before the introduction of Shield, the number of my patients that weren't being properly screened for colorectal cancer was knocking on probably 50%. People are just anxious to go through a colon cancer screening, so anxious that some of my patients are nearly 15 to 20 years overdue per guidelines period. And I assure you that's not for a lack of trying with them. Put very simply, it's just difficult to get patients to do a colonoscopy. Mainly requires multiple steps. You have to see another physician, take time off work because of the anesthesia required. You need to do a bowel prep, and someone else needs to drive you to and from the procedure. So it's a lot of rigmarole to go through. And when patients are juggling jobs, families, and multiple competing priorities, something like a colonoscopy can very frequently fall to the wayside, especially when they have no initial complaints anyway. The stool tests, while an available option, aren't as straightforward as one might hope. Most of my patients don't want to physically collect and package their stool, let alone think about doing so. It is not an easy sell. And yet, as a doctor, I know how deadly colon cancer can be if not caught early, and I know how important screening is. So seeing my patients refuse it and knowing the implications of their health has kept me up at night. That's why when the Shield test came out, it was a no-brainer. I needed an alternative option that would be more appealing to my patients. It was highly sensitive, simple to do, and met all the excellent criteria for a screening examination. Because of its convenience, my patients have felt the same. In fact, when I give my patients the choice, the uptake of doing the Guardant Shield test is virtually 100%. I don't think I've had anyone say no to it in the last 2 years. Given what my screening rates looked like just a few years ago, this type of change in my practice is remarkable. And the best part is the test has not just improved screening adherence in my practice, it's actually helped detect cancer in early stages on a few of my patients. Patients who I'm not sure would have completed another screening offering and who still might be walking around today without knowing they had colon cancer, if not for Shield. Thankfully, all those patients have gone on to get their cancer diagnosed and quickly treated, and they're doing well. It's a testimony to Shield and Shield alone, and to the power it has to reach people who may have otherwise remained unscreened, wake them up to the problem that needs to be addressed, and get them more quickly back on track with their health. To be able to play a role in finding something early on for a patient that you can correct and cure is very gratifying as a physician. Every patient and provider deserves to have Shield in their arsenal when it comes to colon cancer screening. It is an option that people would choose, an option that people will like, and an option that has the potential to save their lives. Thank you for listening and considering my perspective.
Hello, my name is Michael Sapienza. I am the CEO of the Colorectal Cancer Alliance. The Alliance is the world's largest nonprofit advocacy organization dedicated to ending colorectal cancer in our lifetime. In 2009, on Mother's Day, my mom died of this disease. As you know, it is the second leading cause of cancer-related deaths for both men and women in this country, and it was so in 2009 from cancer. Unfortunately, there's three main groups in this country that are still not getting screened. One, people of color, underserved individuals, right? Two, people that live in rural areas. And three, people that are ages 45 to 59. The age group 45 to 49 is only about a 25% screening rate. 50 to 54, only about a 45% screening rate. So tests like the Guardant Shield blood test will provide additional access to those specific groups that can't get other available tests. The way that the Colorectal Cancer Alliance looks at screening is not just around data. So if you think about data, you have sensitivity, you have specificity, you have precancerous adenomas, etc. But we look at it, yes, the data. Data number one, important. But two, adherence. Who is actually going to do the test? Will they do it? And then three, we look at access. Who actually has access to the test? So again, the Colorectal Cancer Alliance, the largest voice in colorectal cancer across the country and across the world, really thinks that Guardant Health's Shield test will provide new opportunities for both adherence and access to colorectal cancer screening.
Hi, I'm Chris Evans, president of the Colon Cancer Coalition. At the Coalition, we work to educate people about colorectal cancer and to knock down any barriers to screening. We know that screening saves lives, yet millions remain unscreened. There are many barriers to getting people screened, and those are the ones we address. We also know that presenting patients with options does help them make educated health decisions. The introduction of a blood-based choice for colorectal cancer screening will increase screening rates and ultimately save lives. So we're very, very excited about this new possibility for our patients.
That was our last recorder's presenter. And I now pronounce the open public hearing to be officially closed. We will continue with panel deliberations. We don't have any legal questions from the morning, either from the sponsor of the FDA. So we will continue with some of the discussions. But before we do that, does any panel member have any questions or comment for the sponsor of the FDA? Dr. Abrouche?
Yes, thank you. I'm wondering if on the epiprocolon, on the package insert, was there a warning to the patient and the doctor that they would require a colonoscopy if the blood test was positive?
Will anybody from the FDA be able to respond to that?
Yes, I'd like to invite my colleague, Dr. Pathak, to answer that question.
Yes, hello. I believe that the intended use for epiprocolon would have indicated, and labeling would have indicated, that a positive result should be followed up by a colonoscopy.
Any other questions for the sponsor of the FDA? Dr. Hewitt?
Yes, this is Dr. Hewitt. With reference to the analysis of the data, as I recall, determinations of the performance with reference to adenomas were based on the highest score or largest adenoma. Was any interim analysis carried out with reference to the numbers of adenomas and the performance of the assay? Basically, what I'm asking is it size or is it the number of adenomas present that was driving the detection?
We could start with the sponsor.
This is Victoria Raymond from Guardant Health. In regards to the questions about performance of the different categories of advanced adenoma, I'll invite the PI, Dr. Chung.
Daniel Chung. So with regard to how we measured the sensitivity for the adenomas, we did look primarily at the highest grade of dysplasia in terms of either whether there's high-grade dysplasia, whether there was a villus component, and we also looked at size, but we did not look at the multiplicity of adenomas.
Any follow-up from the FDA?
Well, I will say that we did do a lot of analysis. I don't know that we have the actual analysis that you are looking for, but we did break down a lot of data based on lesion size, the type of the histology. If you're interested in that data, I can invite my colleagues to bring those backup slides up.
No, actually, what I'm getting at is the direct question of, is the detection of adenomas based on the volume of the adenomas necessarily maybe disconnected from the severity of the adenomas? There are some fundamental scientific questions about the ability to detect primarily altered methylation and or fragmentation, and the question of about the sensitivity of the assay with reference to, is it present in the blood, is it below the detection limits, or is it leaking into the blood? Thank you.
Well, I will say that in the analytical validation studies, we do look at the limit of detection, and we are talking about very low concentrations that can affect the performance of the assay. However, the overarching clinical study is very supportive of the detection of stage 2, 3, and 4 colorectal cancers. In terms of the volume, I do not believe we have that data.
Thank you. Dr. Borowski?
Yes, hi, Sandy Borowski from UC Davis. My question is about the stage 1 difference, the 55% versus the 45% that weren't detected. And I guess my question is about whether there was a further analysis of that. Simple things like grade and phenotype and maybe more complicated things would be possible. Sort of related to Dr. Hewitt's question about what are the features of very early lesions that render them detectable in the blood. And so it strikes at the biology and biological differences between a colonoscopy screen versus a blood screen.
Will the sponsor have data to provide?
Yes, so one of the things that I will note when you look at the stage 1 performance of Shield, that stage 1 performance is actually within range of what we see for other non-invasive testing being used in the clinic today. And if we can, I'll show a slide here that shows the stage 1 performance for FIT testing ranging from 50 to 65.5%, and the stage 1 performance for Shield at 55%. And additionally, Shield has 100% sensitivity for stage 2 and 3 colorectal cancer. I think we don't have any data at this time to share with additional grade of tumor, but I invite our Chief Technology Officer, Daria Chudova, to comment further.
Thank you, Daria Chudova at Guardant Health. Hopefully the slide came through that Victoria was referring to with comparison of 50 to 65% in stage 1 for FIT and blood at 55%. To your comment about what are the features of the lesions that are detectable versus not detectable, there are two things to consider. One is the variety of underlying genomic states of the tumors, and we've looked very carefully to design the assay in a manner that captures variety of CRC cancers. There's no indication from our internal data or the literature that the signature we define is specific to a particular subtype. So we'd expect that signature to have capability for capturing different molecular subtypes of the tumor. Then the second question is potentially what you were alluding to, which is limit of detection of the system and how low can you go in terms of the amount of shedding that occurs. And we believe that that's the biggest determinant for the detection capability of the system, sort of maybe in reference to your volume questions, at what level do we start to see traces of the tumor. The data from our analytical validation and clinical validation is supportive of each other with this regard. And so we believe just the shedding level is probably the most significant variable here, and the performance of the test aligns with those assumptions.
Thank you. I guess I'm also interested in sort of an optimistic possibility, which is that some stage 1 colorectal cancers really will remain such for long periods of time, maybe well beyond the patient's lifetime. And those, therefore, would be the subject of potential, what we call overdiagnosis and overtreatment. And it would be certainly advantageous to know if a blood test was able to distinguish those early stage cancers that did have more consequential potential for progression. And so again, optimistically, there may be features in that stage 1 data set that might add specificity for consequential cancers to your blood-based test?
These are very interesting hypotheses, definitely, and I would suggest that the data supporting higher level of detection of advanced adenomas that have higher malignant potential as high-grade dysplasias and lesions with villous components kind of goes with your hypothesis. So clearly, very excited to see in the future what we can learn from that experience.
Do you have, you're going to be following up for 1 year. Do you have any intention to continue to follow up some of these patients? Because that will be able to answer some of these questions.
Hi, Victoria Raymond from Gardant. So as mentioned, we are following the patients enrolled in Eclipse for up to 2 years to understand those long-term clinical outcomes. And I'll invite our Chief Medical Officer, Craig Eagle, to talk about plans for longer data collection.
Craig Eagle. So in terms of the stage 1 and the actual tissue samples, we don't have the tissue samples and access to follow up with patients in the Eclipse study. So there is no doubt opportunity for future studies. For the long-term study, we're following up the patients in Eclipse out to the 2-year mark for other cancers, as well as if there's any colorectal cancers detected.
Any comments from the FDA?
No, I do not believe we have any additional comments.
Great, thank you. Dr. McLeod?
Thank you, Mariel McLeod again, patient advocate. I wanted to ask, I'm not sure this is more a question for the FDA or Guardant Health, but in recent years with the increase of young onset colon cancer, colorectal cancer, and leading to the access of additional screenings in lieu of colonoscopy, would you ever consider, I know the benchmark for screening is now at 45, but will you ever consider lowering the screening age and access to this type of exam, especially as young onsets are usually diagnosed at a later stage?
So I will say that we follow the recommendations of the clinical community and the guidelines that are set forth. At this time, the guidelines are 45. If they were reduced to 40, we would certainly take that into consideration as part of the benefit risk assessment. I would also add that we would, if a sponsor came in with data supporting down to a screening test for 40 and above, we would certainly accept that data for review and include it as part of the benefit risk assessment.
Thank you. I appreciate your response.
Anything from the sponsor to add it?
So similar to what Donna just mentioned, we too follow the guidance of the clinical guidelines and our clinical stakeholders, and look forward to paying close attention to this space and the alarming rise in young onset colorectal cancer as we move forward.
Thank you. Dr. Rajapal?
Dr. Sheila Rajagopal. So I was wondering, this is for the sponsor, while the population was intended as average risk, do you have any information available regarding incidental capture of microsatellite instability via the circulating DNA component that might prompt additional referral for clinical testing at this time?
Would the sponsor please address?
Yes, Victoria Raymond from Gardant. I will invite Dr. Chudova to comment on that information.
Daria Chudova, I will take maybe the second part of your question regarding MSI status and any information the assay provides about this. The results we return to patients are limited to qualitative result of abnormal signal detected or not detected, so that status is not assessed as part of the testing. As part of our internal data development programs, we have evaluated the capability of the assay to detect CRCs that have MSI phenotype versus don't, and data supports similar detection capability for these types of lesions, but that data is not returned to the patient. And maybe back to Victoria Raymond, our moderator, for a question about average risk population as intended use.
So thank you, Victoria Raymond. So we do know that guidelines do recommend assessment of MSI status and in newly diagnosed colorectal cancers. We actually did not collect that information as part of Eclipse, so I cannot answer that question for you today.
Thank you. Dr. Hewitt?
Yes, this is Dr. Hewitt. I have a question for the FDA. Does the FDA have definition of the difference or statement of the difference between a screening test and a diagnostic test?
In general, a screening test is that which is used in the asymptomatic population, whereas a diagnostic test is used in a population of people who are symptomatic.
Thank you. Dr. Bruce?
Yes, I wonder if I could ask the FDA as well as the sponsor, Gardant, about how do they feel about having some sort of statement in the package insert about the requirement for the patient to have a colonoscopy if the blood test is positive.
So I would like to start off answering that if you don't mind, because I do believe this was your earlier question. We may not have adequately addressed it. And the intended use statement does refer to patients should be followed up by colonoscopies. So that is a recommendation that is in the package insert.
So it's the wording is should.
Right. So
it's not.
I have to. Maybe we could pull up the intended use, but there's a lot of labeling, and there's a section in that occurs in the review of these submissions and in the authorization of these tests. And there's a section entitled limitations that's for physicians so that they understand the limitations of the test and what should be done. However, the FDA does not get into the practice of medicine, and it would be the clinician's decision to decide how to follow up. So I would, if Gardant would like to bring up the IU, or if you think the IU is necessary to bring up, we can do that. But yes, this information is in the labeling.
Thank you. Would the sponsor want to add anything?
Yes, I can pull up the proposed indication for use here. And as you mentioned in the first bullet, it outlines that patients with a quote-unquote abnormal signal detected may have colorectal cancer or advanced adenoma and should be referred for colonoscopy evaluation.
Thank you very much. Dr. Borowski?
I think it said may have.
Sorry, Mr. Bruce?
I think in the slide that was just up, I think the wording was the patient may have a colonoscopy.
No, can I have the indication for use slide again, please? Patients with an abnormal signal detected may have colorectal cancer or advanced adenoma and should be referred for colonoscopy evaluation. So the may have is referring to this is a screening test.
It went by so fast. I can't understand. But I just want to make sure you guys appreciate the importance of a colonoscopy. If the patient has a positive blood test and they don't have a colonoscopy, then there's nothing that can be done.
Correct. Thank you, Mr. Bruce. Dr. Borowski?
Yeah, Sandy Borowski, UC Davis. I was interested in thinking a little bit more about the receiver operating curve and the choice of threshold. Given that the result of a positive test would be a colonoscopy, which is already in some ways considered the gold standard for screening, although admittedly with difficulties in access and acceptability. Would it make sense to choose a different point on that curve for higher sensitivity and lower specificity to get more people sort of into the colonoscopy side of things?
Will the sponsor respond?
Yes, a lot of thought went into defining that threshold. And so I'll invite Dr. Chudova to comment on kind of the inputs and thoughts about that threshold.
Daria Chudova, Guardant Health. So the target of 90% specificity was defined earlier in our development program, and it was aligned to the existing guideline recommendations for screening tests as an acceptable targets. And as you refer to, the acceptable benefit risk trade-off is related to the number of follow-up colonoscopies that will be done versus the sensitivity assessment. There's obviously from a development standpoint, different points in the ROC space that could be explored, but we're really guiding it based on the clinical recommendations of experts that were part of the study design. And maybe I could ask our clinicians to comment on this because I feel that could add to the perspective here.
Daniel Chung. And so, as was pointed out, 90% specificity was identified as the target really for an ideal screening test. And I think the number comes about because you're trying to find the right balance between too many or too few false positives. And I think that as we've discussed and has been sort of, I think, proposed by numerous organizations, and guidelines, that 90% is the target that is appropriate for this screening test.
Thank you. Dr. Hoover?
Yes, I have a question for the sponsor. It appears from the data presented that unlike histology, which has no parameter for age is considered in the logistics regression cutoff points in the algorithm. Is that correct?
I'll ask Dr. Chudova to comment.
The question was whether the age is part of the logistic regression for the analysis? Yes. No, it's not an explicit covariate in the model.
Is it a covariate at all in the model?
It's not a covariate in the model. We explored that very, very deeply, I would say, in development, knowing the sensitivity of methylation assessment with age. What we learned from that experience of trying different models to extract the best signal-to-noise ratio from the data is that the models already take into account these two factors that are ongoing in the same time. One is decrease in specificity with age, and two, increase in the prevalence of cancer with age. And so when the models are presented with sufficient amount of training data that reflects these characteristics based on the nature of the samples, it learns that dependence in an optimal way based on the sort of many attempts to inform the model explicitly about the age. So it was tried and didn't yield better performance than the current models we have.
So it's encompassed, but it's not a variable. That's very good.
Thank you.
Any addition from the FDA?
No additional comments.
Dr. Singh?
Yeah, hi, Vikesh Singh. I have a little bit more of a general question because I don't really know what the answer of this is, but when patients undergo a non-invasive screening test, whether that be through blood or stool, and they have a positive result, what proportion of those patients go on to get a colonoscopy? Because, certainly a lot of the members of the public who also were, had come up to the mic and talking about improving, access, for communities that don't have good access, whether they be persons of color or sites of the country that are very rural in geography. I don't understand that even if you have a positive test, if you can't easily get access to a colonoscopy, then the test is just a test, right? There's no confirmation or there's nothing done to actually reduce, the incidence of colorectal cancer, for that matter, or for that matter, treat it at a stage when it can be treated more effectively. So, I'm still trying to understand the connections here because I think that's very, very critical.
Will the sponsor respond?
You raise a critical point about the benefits and limitations of these non-invasive screening tests, and it's exactly true that when a patient decides to pursue one of these colonoscopy alternatives, it's critical that they understand that an abnormal signal by any of these tests requires that diagnostic follow-on colonoscopy is one of the reasons that it will be part of our educational materials, and also it's part of our indication for use. We know from data from stool-based testing that the follow-up diagnostic colonoscopy rates vary widely, mostly due to practice settings, but they can range anywhere from 30 to 80% at 6 months of post-positive test result. We have been following the patients that have a positive Shield through our commercial ordering to understand what our follow-on diagnostic colonoscopy rate, and we are seeing at 6 months very similar rates to what we're seeing in stool-based testing.
Would the FDA have anything else to add?
Well, I would just add that that challenge is going to be the same for any screening test.
I think that's a very, very important point too. Dr. Bauman.
Yeah, it's sort of a little bit of a follow-on on that. I mean, the sponsor did show some data that they don't think it's gonna, the availability of a blood-based test will not impact the use of colonoscopy as sort of the people that use it as their primary screening. However, we've been hearing the fact that people are having a hard time getting in for their screening colonoscopies because there just isn't availability. And with a 90% specificity, this test is going to generate a huge population of patients that have to go on and get colonoscopy. And they probably will be given priority over those that want to use colonoscopy for just screening that don't have any indication. And so I don't know if I really believe the statement that, you know, it will have no or little impact on the use of colonoscopy for screening. And that's going to have a big impact on prevention because that colonoscopy can do prevention, whereas this test cannot.
Will the sponsor respond?
Yes. So providers have been managing colonoscopy alternatives for a long time, and so we've thought a lot about the introduction of this test and what that means to the colorectal cancer screening landscape. And to further comment on your question, Dr. Ballman, I'd like to invite Dr. Eagle.
So I think there's a couple of things to think about, and whilst we recognise that choice of test is going to be something to consider, the reality of the data to date that we have suggests that colonoscopy is not impacted, and that's something we see from the data. And I'll shortly get an expert, Dr. Liang, to come and comment on some of that data. The other piece to remember with colonoscopy follow-up, as well as with colonoscopy after non-invasive testing, this helps prioritise, as we heard in some of the videos as well, the colonoscopy resources to those tests that are positive. So overall, if we're going to screen the entire population by using non-invasive testing like Shield and like other tests, it enables to prioritise and focus where the colonoscopy resources are being used. But I might get Dr. Liang just to go over some of the data where blood test was introduced.
Apologies, this is Daria Trudeau. I want to maybe quote one more performance indicator from Shield that speaks to the level of enrichment that we're going to see from positive Shield tests. So the performance data for Shield indicates that there's approximately 7 to 8-fold increase in risk of cancer for a positive test of a patient. So increasing the colonoscopy utilization for a group that has that much of an increase in risk is something that we should be considering and prioritizing resources based on these considerations. So just wanted to add that data piece.
Okay, thank you.
Yeah, just to follow up, this is Carla Ballman, but you did not weigh that against sort of the prevention that comes from people being able to get colonoscopy by when you prioritize colonoscopy scarce resources for those that have a false negative or false positive test, correct?
That is correct, yes.
Any comments from the FDA?
Well, I would just say that this is outside of the availability of colonoscopy, and the challenges with getting colonoscopy would be outside of our review and our benefit risk assessment. However, I would invite the panel to discuss amongst themselves these types of topics if you have any sorts of inputs that you think are valuable to share with each other.
Thank you very much. Dr. Borowski? You're muted, sir.
Apologies, now unmuted. Fanny Browski, UC Davis. I'm interested in the false positive group. The false positive group, I wonder if you have any data on, I know this was, Eclipse was a snapshot, but do you have any data yet on how many of those revert to a negative test? What the patient perception of that false positive test has turned out to be, how they think about it. And of course, also the group, if any, that convert from false positive eventually to true positive. Is this a detection problem?
It's sponsored?
Yes. So I will invite Dr. Chung to talk a bit about the data we have from the 1-year outcomes of individuals with a positive Shield. As we think about the landscape of a colorectal cancer screening test, when we think about the utilization of stool-based testing over many decades, patients and providers are used to having conversations about the benefits and limitations of these alternatives, so we don't anticipate any extenuating anxiety for those individuals who are false positive, given this is explained as a screening test and not a diagnostic test. And the false positive rate is within the range of the other stool-based tests that's being used today. Ongoing work will evaluate longitudinal testing in those individuals to understand subsequent test results, and then I will hand it off to Dr. Chung.
Dr. Chung. So just a couple of comments about the false positives. So number one, in Eclipse, the patients did not receive test results, so we don't really have any feedback from patients about their interpretation and feeling for those results. And in terms of those specific patients, as was pointed out, this is an observational study that we have not repeated tests on those individuals at subsequent time points. So this was just a one-time draw, and so we do not have that kind of follow-up data.
Thank you. Dr. Morgan?
Hi, Charity Morgan. Can you comment on the decision, this is questions for the sponsor, the decision not to do repeat testing in the 2-year follow-up period? And also, I'm assuming they are still going to have colonoscopies during the follow-up period, but please clarify that point as well.
Yes, thank you, Victoria Raymond. The goal of the study was to evaluate primarily colorectal cancer sensitivity and specificity in the average risk screening population. The study was designed in that way, and so repeat testing was not part of that study design. All of the individuals who were enrolled and were evaluated went on for colonoscopy and then were managed subsequently based on their colonoscopy findings, given, as Dr. Chung outlined, results were not returned to participants. Results of the Shield test were not returned to participants.
But they continue to have colonoscopies in the 2-year follow-up, depending on what their results were, and are those returned to will those be part of the study findings?
So I'll ask Dr. Chung to comment on management in various colonoscopy settings, but if additional colonoscopies were completed, that information would be collected on the 1- and 2-year outcomes that we're gathering.
Thank you.
Dan Chung. So just to clarify, the way the protocol was set up is that everyone who was enrolled and was evaluable did indeed have a colonoscopy at the starting point. So we have results from those colonoscopies, and we'll tell them whether it was normal, whether they had adenomas, polyps, whether they had cancer. And so their clinical follow-up will be mandated by what the findings of that colonoscopy were. Okay.
Thank you. Dr. Singh?
There is also, of course, the very important issue of false negatives, right? Because if this test failed to detect all colorectal cancers less than 1 centimeter, it also missed 17% of stage 1 colorectal cancers, and it missed 87% of advanced adenomas. That's the point at which colonoscopy has the most efficacy in actually altering outcomes, because you're finding disease and you're doing something about it, right? You're removing a polyp, you're removing an advanced adenoma, and you're also able to resect a stage 1 colorectal cancer. You may not be able to resect stage 2, stage 3, stage 4, right? So I think the false negative issue is critical. So I don't think the panel should only focus on false negative, but I'd be interested to hear others' perspectives.
Sponsor?
So Shield is a colorectal cancer screening test, and it has its benefits and it has its limitations. And you're right that in any patient who is willing to discuss colorectal cancer screening, colonoscopy should be prioritized as part of that discussion, because as you mentioned, it has that ability to not only detect early stage cancer, but also to prevent the development of cancer with the removal of advanced adenomas. Unfortunately, what we know is that the majority of people actually decide not to pursue colonoscopy, and that's where non-invasive options actually play a critical role, given their proven ability to reduce colorectal cancer mortality. And when we think about the false negative rate in Shield, it is within range of what we see with other non-invasive screening tests. And to further comment on this clinical perspective, I'd like to invite Dr. Eagle.
Craig Eagle, I think one of the things we've got to keep in mind as we think about the impact of these sorts of tests is this is a single point in time. And whilst clearly colonoscopy is the best test, as you mentioned, for both adenoma detection and also for colon cancer, and ultimately therapeutic and interventional as well. So there's a lot of advantages over others. But we've got to keep in mind that that early cancer detection you bring up, that false negative rate, is currently occurring right now with FIT. The second point is that if you only have...
Can I interrupt you? But what about the stool-based DNA test? That actually had a sensitivity of almost 90%, right? So yours actually was what performed at a lower rate for the stage 1 colorectal cancer.
Yeah, so you're quite correct. And what I'd like to just remind is that it's not one single stool test that's being used at the moment to screen for cancer. And then we need to focus on actually completing the test on multiple repeat intervals. And if we think about colonoscopy and the adherence to repeat colonoscopy, and we think about the adherence to stool-based testing, this is something that the panel should consider as well as you think about the false negative rates as you're comparing devices versus comparing with a one single time point measurement.
The FDA has any comment?
Well, I would just add that the false negative rate is the reason for the panel. And so I appreciate you bringing it up and welcome your discussion on this topic, given and in light of the fact that it is a blood-based test, which is expected to improve compliance with colorectal cancer screening. So I appreciate you bringing that up and believe that that is the critical question here today.
Dr. Baumann?
Yes, Carla Baumann. I just want to follow up on that last statement about, yes, I understand this is a single time point test, but do you have data on adherence? Because this test would have to be done much more often than would a colonoscopy that came up negative. And that means people have to come into the office like on a yearly basis. If it's yearly, you don't even, I haven't even heard a time frame. So could you tell us what data you have for better longitudinal adherence other than you think it's going to be better?
Yes. Yes, thank you. If we may answer, I'll invite Dr. Eagle to comment.
Yeah, so maybe I can just start with, this is an innovative test, and so like anything innovative, we're still working through collecting the data. And I just would remind that at this point in time, we do have a single point adherence in the range of 88 to 90%. For the blood test compared to well-established adherences at a single time point with the other tests that are in the range of the 30 to 60 range. I acknowledge we don't have longitudinal data, but as an innovative test, that's part of our commitment to follow up with studies. And I really look forward to actually the discussion with the panel about how we can design those studies to really look at longitudinal adherence and what sorts of measures they would need. But certainly this is a very valid point.
Okay, and we'll take the last question before we start deliberations. And at what point we can still ask questions for the sponsor of the FDA, but I would like to start with some of the deliberation. Dr. Hayward?
And it seems like we're being asked to make some decisions on what is a screening test in a prophetic manner. These are prophetic claims that it will improve outcomes, when in fact we don't have the data on serial performance of this assay. And in the absence of detection of adenomas, which are really the driver of being able to intervene early before symptoms and prevent advanced disease, it doesn't matter if you've got performance at stage 2, stage 3, and stage 4, which require advanced interventions. It is not meeting the fit-for-purpose space in the early stages in which it can be stopped. Colonoscopy is obviously the gold standard. However, fecal-based assays are providing critical information that are filling this space and preventing more advanced disease. In the absence of serial data to suggest that it's going to change that trajectory, it's a very difficult position.
Thank you very much for that comment, Dr. Hu. And I think that is one of the critical points for some of the discussions and advice back from the FDA. Will the sponsor respond?
Yes, thank you. Victoria Raymond. When we think about the evaluation of other colorectal cancer screening tests, the data that we have available today is similar to the data that were available about 10 years ago when these other tests were reviewed. And to comment on our plans for gathering the critical additional data that you've mentioned, I'd like to invite Dr. Craig Eagle.
Craig Eagle, I appreciate that the panel is deliberating on some areas that need further elaboration, and I think I'd just like to double down a little bit on the comments made before. Now, as we know, colorectal cancer is the second leading cause of cancer death, and that's with the availability of, as you point out, a very powerful test like colonoscopy, but also with the non-invasive tests. And so at the moment we're dealing with a situation that still remains the case even 10 years after the last deliberation of the panel. And so I just encourage to think about that at the moment in the US, about 85% of people get an annual physical checkup. And so the ability to slot Shield into that annual checkup and to screen much larger groups of the patient population or the adult population in the US than any test before it is actually a very positive opportunity for physicians to deploy should they need to for those that are unscreened. The other thing to keep in mind is 75% today of people dying with cancer are not up to date with their screening. So they don't do any device. And so whilst we focus and acknowledge that colonoscopy is certainly the most important screening test, unfortunately it's not deployed as much as we would like, and we still have that second leading cause of cancer death. And I really look forward to committing with the panel what sort of data we need to build out that unknown that we're talking about in the Shield test to really demonstrate some of these advantages.
Thank you very much. So as we continue with the deliberations, the portion is open to public observers. Public attendees may not participate except at a specific request of the panel chair. Additionally, we request that all persons who are asked to speak identify themselves each time, as this helps the transcriptions identify the speaker. So we will continue. At this point, anybody else had a question? We have some time, so any additional questions for deliberation?
I would just like to add one comment, a clarifying comment, if I may. Earlier, I was asked about the epiprocolon IU, and I just want to say that that was compared to fit within that study. It was a head-to-head comparison within that study and not compared to fit performance in literature.
So at this point, we will take a 15-minute break, and then we will continue back with the questions for the panel, if there are no other deliberations that need to be brought up at this time. Any other comments or any other? So then we will continue with answering the questions posed by the FDA to us. We'll be back in 15 minutes. Thank you very much. It is now 2:52 p.m. and I would like to call this meeting back to order. At this time, let us focus our discussions on the FDA questions to the panel. Panel members, copies of the questions have been sent to you electronically and posted online for the public. Please remember to identify yourself each time you speak, as this helps the transcriptionists identify the speakers. Please project the first question. So the first question is, Shield is intended for colorectal cancer screening. Well, can we go back to question 1? There we go. So Shield is intended for colorectal cancer screening in the individuals at average risk of the disease, age 45 years or older, as a primary screening option. The Guardian test demonstrated colorectal cancer sensitivity of 83.1%, advanced adenoma sensitivity of 13.2%, and advanced neoplasia a specificity of 89.6%. So the first question to this statement is, based on the clinical performance of this device, the benefits and risks of the device for colorectal screening, including considerations for the appropriate patient population and clinical scenario for this device. Does the clinical performance support the use of the Shield test as a primary screening option, similar to other non-invasive colorectal cancer screening options, or is more appropriate for specific populations, for example, patients who decline other colorectal cancer tests? So we are open for discussion. Anybody wants to start some? Yes, Dr. Bruce.
I'd like to just, point out something. I think I've heard that Gardant is advocating that their test will not interfere with the scheduled recommendations for colonoscopy. So, for example, if the patient undergoes a Gardant test at age 50 and it's negative, the official recommendation to the patient is to have a colonoscopy at age 50. And that recommendation should not change. So I want to know if that is correct.
Would the sponsor provide that information?
Hi, Victoria Raymond from Gardant. As we think about the opportunity of Shield to be used as a primary screening test, we would see this test being used in similar ways that stool-based testing is being used today. So for those individuals who are eligible for screening, the conversation would begin with offering colonoscopy, and should those individuals not decide to pursue colonoscopy, then be offered non-invasive alternatives that would be repeated at the frequency per test.
Thank you.
So the patient would no longer be in the schedule for colonoscopy.
With a negative Shield test, the negative predictive value for colorectal cancer is 99.9%. And so for that individual, provided Shield is approved as a primary screening option, which we believe it should be, given that the data supports has performance in line with other screening tests, then it would be repeated at the frequency of interval, which will most likely be between 1 to 3 years.
Thank you very much. Anything from the FDA?
Sorry, I was on mute. Sorry, I was on mute. I'll just add that prior colonoscopy was part of the exclusion criteria for the clinical trial. Subjects were not to have colonoscopy within 9 years in order to be enrolled in the trial.
Okay. Dr. Morgan?
Yes. I wanted to comment on the second bullet point about the primary screening option. My concern is, I think something Dr. Baldwin kind of alluded to earlier with her questions about this test winding up sort of replacing colonoscopy, or patients who opt for colonoscopy sort of being sort of put to the back of the line behind those who opt for the Shield. And without long-term data on whether or not this test would affect the use of colonoscopy as a primary option for screening, I think it makes more sense to have this as a second line, or saying patients who decline other screening decline colonoscopy or do not have access to colonoscopy. I think this is a good option for them, because I am sympathetic to the disparities that exist currently with access to colonoscopy. But I wouldn't want people who have access to colonoscopy and have the means to get it to opt for Shield instead. I think the data shows that colonoscopy is clearly superior, and I don't think Gardant would argue with that, but the issue is that it's better than nothing for people who are going to get nothing, but it's not better than colonoscopy.
Yeah, so I agree with you. And I think the importance here is not only the labeling, in where colonoscopy is still the recommendation as the screening test, and it is the recommended screening test, but also how the educational information from Gardant is presented to continue to harpen the idea that the colonoscopy is first, and that there still are, lower detection rates with these blood tests. But this is not different from the stool base that they have today. Dr. Winslow?
Yeah, thanks, Dr. Ferrer. Just a couple of questions, and maybe more clarifying for you, Dr. Ferrer, maybe FDA. With respect to the point that was just raised, it would be good to understand a bit the implications of such a recommendation, because I think I heard early on in the presentation the epicolon really isn't an option any longer. I know it's an FDA-approved test, but are the implications of a recommendation or decision like that really influencing practice of medicine as opposed to allowing the option or a choice? So I would be curious if the sponsor, perhaps I know FDA couldn't comment, whether they have some information on kind of where we're at with epicolon as a result of how that decision was made. And the fact that this test seems to be, at least performance-wise, on par with the other tests that are out there in terms of non-interventional. The other consideration too I was going to ask about is with respect to the discussion that was happening at the end of the last session around some of the data we don't have and how we are supposed to be looking at this particular question, because I think the test itself, or the study itself, was focused on performance at a single time point, and that's what I think the approval or the label will be based upon, and there will be some post-marketing, and I think the sponsor's been committed to looking at those additional questions that might come up. So I just want to make sure that we understand what we're being asked to look at in this question, and we're not doing more than perhaps the FDA would be looking for in terms of our recommendations and what's been done in the past. So I don't know if Dr. Roscoe, that might be help clarifying that aspect of this question.
Well, so I would like to invite Dr. Pathak to provide a more thorough description of the current available FDA-approved colorectal cancer screening tests to go through that. But I will just say that the FDA is motivated to support patient health. And to the extent that there is an established program for colorectal cancer, which includes screening methods other than colonoscopies, this test is sort of falling in the middle with compared to the stool-based testing, various different types of stool-based testing. So, but there are advantages to this test. So that is the challenge that we're engaging the panel with to try and help figure out where this should fall in terms of the IU and the recommendations in the labeling. But at this time, I'll invite Dr. Pathak to bring up a slide to go through.
Can we go to slide 13?
Now, to add to this, sorry to interject, is that the second test that we're talking about, the indications were different because it was as a second line. And what we're talking here is as a first line. But even though that this been as a second line, I think there has been denial of reimbursement, which is beyond this group, and we can that they have stopped offering the test must have nothing to do with the performance, but actually with the payment for that. So, and this is not the purview of this committee.
Right, and I brought up about the epiprocolon and the reasons why that was given a second line claim was because it fell short of the existing tests at that time. So we're motivated to have continually improving tests and considering the benefit risks to all the features of these tests.
But it seems like if we're going to have it as a first line test, if we're going to have Shield as a first line test, it has to have operating characteristics that approach colonoscopy, or certainly better than having a false negative rate of 17%. That's terrible.
This is Stephen Human. Yeah, I look at it comparing the other first line test. You have the first line test of colonoscopy, which is going to find your adenomas, remove your adenomas, and prevent death. You have second line test, or you have another first line test, which is the molecular fecal test. And those assays have pretty good performance, not great, as good as colonoscopy, but they have good performance, and they're going to have a high likelihood of finding your adenomas. You're going to get a colonoscopy if you follow the guidance. They're going to remove your risk. And if they don't find it in your first pass, they should, in the recommendations currently at 3 years, find it in the second pass. But that's at a 42% sensitivity for adenoma. In this instance, this assay from Gardant has a 17% sensitivity for adenoma. So you're only going to get a fraction of the adenomas that are going to get referred to colonoscopy in the first pass, and remain, even in this instance where we don't know the frequency, at a fraction of them in the second or third pass. You're kicking the stone down the road. To be honest, it's got nice performance as a cancer detection assay, especially in stage 2, stage 3, stage 4. And yes, there is a blind spot in colonoscopy, especially at the stage 1, stage 2 diagnosis, but it's not necessarily beating colonoscopy at stage 1, but the failure to be able to remove lesions that have a propensity to move forward is concerning.
Dr. Winslow?
Sorry, I didn't put my hand down.
Okay. Will the sponsor want to respond to that?
Yes, thank you. Victoria Raymond, Guardant Health. One of the reasons why the data support Shield being a primary screening option to be offered alongside non-invasive options. If we take colonoscopy off, as we've talked about, colonoscopy should be offered first. And then for those individuals who decide not to pursue colonoscopy, then that's the conversation around non-invasive options, which currently includes a wide menu of stool-based testing. Is it possible that I can please share a slide? Okay, if I can briefly share a slide. When you look at the current primary screening tests, these include not only the multi-target stool DNA or Cologuard, but also FIT and FOBT, which are used by millions of patients and providers on an annual basis, what you see is the performance of Shield is within range of these primary screening options. And we know that tests, including FOBT, that have an advanced adenoma performance as low as 11%, actually have been shown to reduce incidence and mortality from colorectal cancer. And so for those reasons, the data do support that Shield should be a primary screening option to be offered when you're offering other non-invasive options.
Thank you. Dr. Spencer?
Hi, Sean Spencer, Stanford University. I was wondering if it's appropriate to discuss recent modeling papers that have been published in academic journals.
Yeah.
There was a preprint in Gastro, and I think the real question here is, at a population level, will this replace colonoscopies or will this be used alongside of it? And they had some very helpful modeling here, because I think what we're dealing with in the hypothetical current state that they had 40% of individuals available were getting a colonoscopy, and 10% were getting FIT and 10% molecular stool, so 40% remained unscreened. And they had a model in which that it exclusively substituted current tests, in which you reduced stool-based testing by half to 10% and colonoscopy from 40 to 30%. And then in that instance, colorectal cancer deaths actually went up. But when they created a more real-world model in which the rate of the test was substituted for some testing, so stool-based test went down about 25% and colonoscopy went down by 5% from 40 to 35%. The modeling actually suggested that colorectal cancer deaths would decrease. And I think it's important to recognize that this will be used alongside. And I think it's really important. And I think in their briefing documents, they had a nice, reasonable consideration of how this should be discussed. And I think ideally, as someone who provides colonoscopies, we want to ensure access. And I think the access issue is something that we're actively working on. But for me, this modeling was fairly reassuring in terms of, I think the goal is to get more people getting colonoscopies. And I think this modeling suggests that even if you have a decrease in other testing, you would still see an overall mortality benefit in colorectal cancer. And I also thought it was worth noting I thought this was nice that the FDA presented this in this fashion, the percent positive Shield results with different age groups. You know, it's pretty striking that in the 50 to 59, 7% of Shield results are positive, and 60 to 69, 11% of Shield results are positive. And I think when stated in that way, I mean, I would be, the hopeful side of this is that is a large volume of patients that are very hesitant to engage in the colonoscopy screening system that might be looped into the fold. I would think that most people, the hope is that most people getting the Shield test, would otherwise not consider a colonoscopy. And I think that is the crux of the argument. And this modeling, given the performance characteristics, I thought was helpful in engaging in kind of the real world aspects of this.
One of the concerns I have is that this test will bias the individuals that don't want to have a colonoscopy to have a blood versus the stool test too, because it's a lot easier, not really understanding the differences in the earlier stages for that. Dr. Borowski? You're muted, sir.
Getting the mute button. Okay. Dr. Nebraska at UC Davis. I raised this morning as a question to the sponsor, the question about whether it was more important to catch early invasive cancers or to remove all adenomas whenever they arise in terms of mortality benefit. And I think that there may be expertise on the panel that I don't have, but from what I've heard so far, the answer is we don't know. There may be both. I think that the sponsor did a nice job of showing that the data for this blood-based test falls well within the range of the existing approved stool-based tests. And I think that we all agree that the important thing is getting more people screened in some fashion. And so this comes to my question that I asked later about the receiver operating curve and choosing the sensitivity limits. I think that, as Dr. Spencer just stated, we're trying to get people engaged in their own health and screening. And if we can use a blood-based test as the gateway drug to get them interested in having a colonoscopy, that's a good thing. And so it comes back to my question about where we should optimally find that sensitivity limits. And maybe we should think about this not just as an alternative secondary screen, but as a primary screen, which is recognized to be an imperfect primary screen that would allow us to do sort of a risk analysis of who ought to be prioritized for colonoscopy. So I'm interested in both the sponsors and the FDA's comments to those concepts.
I think that's a very important question, and it'll be very interesting to answer because that goes to the crux of some of the discussions that we're having. The sponsor?
I think one of the things that, and this is Victoria Raymond Gardant, I think one of the things that we do think is exciting is the data that we've showing that improved adherence in those individuals. I think this is a nice opportunity to consider a non-invasive option to bring those individuals up to date with screening. 42% of age-eligible individuals are not up to date. So seeing this test introduced will answer a lot of questions about how we actually close that gap on CRC screening and really significantly reduce that colorectal cancer screening mortality. Still the second leading cause of cancer related to death today.
FDA?
Well, I would just say that I interpret Dr. Borowski's question as being one about how the FDA views the selection of the threshold for sensitivity and specificity performance. And I will just say that this comes down to, we allow the sponsor to make those decisions, and then we review the data and consider the benefit risk. So certainly with a colorectal cancer where false positive has a relatively safe follow-up procedure, such as colonoscopies, that there is more tolerance for, we would consider there to be more tolerance for a high false positive rate. We would consider the risk of, for example, perforated bowels in that setting by looking at the adverse events that the sponsor had from the clinical trial. And in this case, Gardant, I believe, did not see any or did not attribute them to their particular clinical trial. And then aside from that, if we determine a favorable benefit risk ratio, at that point, it is the discretion of the clinical community to determine whether or not it is a test that they would like to use for their patient setting.
Yeah, but it's still a concern in the false negatives. Even though you might screen a lot more people, that maybe you might still having a lot more people that have early stages that are not detected. So that's still a concern.
Yes.
Padma?
Padma Rajagopal. In sort of listening to the discussions, and sort of with partly with an epidemiology background, I very much can appreciate sort of a two-tier move towards testing, where you might have one that encompasses more people to move towards a clearer test result, but that may have more leeway in terms of false positives and false negatives, which is sort of part of what we're trying to determine what makes sense from a primary versus secondary standpoint. But I think one of the things that stood out to me as a potential risk, just from actually hearing all of the patient and public weigh-ins, was that it seemed like there was a lot of conflation, both among providers and among patients, about the application of the Shield test. And that, I think, really speaks to the heart of the labeling needing to be very clear and the educational materials to be very clear. Because even people who were excited about this were excited about reasons related not always related to sort of this discussion about primary versus secondary or targeting populations that may not otherwise be able to get access, but sort of in ways that aren't part of the discussion we're talking about. So I think that's a risk that should be addressed, however we choose to proceed with this.
Thank you. Mr. Losspring?
You're on mute.
You're on mute.
Mr. Losspring, you're mute, sir.
Sorry, I'm Edward Lofspring, coming from a consumer standpoint, and I agree with the previous speaker. The labeling has to be clear that this is not a first-line test, and I agree. She took my words out of my mouth almost, what I was going to say. So, just gonna, we should piggyback on that.
Dr. Bauman?
Hi, Carla Bauman. I just want to speak to, that cutoff for sensitivity, specificity that was brought up, and the potential that this should be sort of used as a first stage going to a second stage. If that were the case, we would lose all the benefit from cancer prevention, because we know right now that Shield is not going to be very good at detecting adenomas, as we know that colorectal screening is. So I would be against sort of using this as a first tier to see who then needs to go on to colorectal just globally.
Fatma, you have a question, or you just forgot to? Okay. Dr. Huey?
So Dr. Bauman basically said a good portion of what I was going to say, and that is this really undermines the concept of cancer prevention. And we see that in the public commentary. None of the speakers actually address the complexity of cancer prevention. Cancer prevention is an incredibly difficult space. We all know that. I wanted to come back to some of Dr. Spencer's comments about the models, because the models are really complex and I haven't had an opportunity to dig into them. But one of the big challenges, there's two elements to this, is one, the advancing number of adenomas as people age. So that is a real complexity, and you don't know if all the models are treating risk in a stratified basis or not at age, one. And then the other one is really at the bottom end of this assay, and that is the 45. The fact that we've moved our screening age from 50 to 45, because we're trying to use the surgeon models of how long it takes to progress. But if you look at some of the real biomolecular data, it says that some of those younger patients probably have got mutations that are making their tumors or their adenomas and everything else advance faster. I think that it's too easy to skip over and say, oh, well, this is the average. If you look at the surgeon model, looked at table 2 and Knudsen in depth, what you saw was that the variance on those models was enormous, and they were taking the averages. And then when you start looking at the total variance, the risk of missing an event that you might have caught using any of the other tests was concerning.
I do agree with your statement. The concern I see is as continued aging population and the number of individuals that need to be screened with colonoscopy. What is the capacity to see all those patients? So the idea will be not so much preventative, but identifying those individuals with colorectal cancer. At the same time, I'm very concerned, and might be that to the literature that has to be provided, that there might be a misunderstanding of what a negative result is, and then these false negatives are undermined or not fully understood to see what you follow up the patient with. Dr. Gilger?
Thank you, Mark Gilger, Baylor, Houston. Really, really wonderful discussion. I think our goal is increased colon cancer screening. You then offer that patient a test. The test preference is colonoscopy, because not only can you see it, you can remove it if you see something there. That makes great sense. Problem is, not everybody wants a colonoscopy. That's an issue. And so then you need to offer them, how can I improve my odds of making that patient actually follow up and get a colonoscopy? So then you're going to do a second test or an alternative test. Stool testing may be a little bit better, but rather cumbersome. Done it myself. I think the blood test simply offers you an alternative. And if that's positive, I think the patient is going to be very, very inspired to have a colonoscopy. That makes sense to me. And if it's negative, 99.9% negative predicted value, that's pretty damn good. And if it's a false negative, that's a very real concern. But the recommendation will be it gets repeated 1 year to 3 years down the road. It still gets repeated. We repeat colonoscopy every 10 years. So I think this test fits in reasonable and makes some sense.
Thank you very much. Dr. Morgan?
Yeah, one thing I want to say about the negative predictive value, that is dependent on the disease prevalence. So it's going to be, negative value is going to be high for a rare disease, no matter how good a screening test is in its specificity. So I wouldn't rely only on that as a marker of how you evaluate the test. One thing that I was struck by when thinking about the fact that this test has such poor sensitivity for the advanced adenomas, and this does speak a little bit to discussion question too, so I hope I'm not getting too ahead of the discussion. We had that nice figure showing the timeline of the development of colorectal cancer and how long it takes to actually turn to the deadliest distant form. But the Shield test really isn't going to take advantage of that long lead time before the development of the cancer. There is that long period where there are the adenomas that are growing. And the nice thing about colonoscopy is that we get to take advantage of the long sort of dormant period before these cancers turn deadly. And so, like Dr. Rajagopal was saying, it was troubling to hear so many people in the public suggest that this could be used in place of colonoscopy, saying things like, I'd rather get this than a colonoscopy. I understand that, people who don't have access to it, this is better than nothing, but I don't want to downplay the issue that this test is going to miss a lot of cancers and advanced adenomas, and the negative predictive value doesn't really capture that that's what's going to happen.
Very interesting comment, Dr. Morgan. Thank you very much. Dr. Spencer?
Yeah, hi, Sean Spencer. I wanted to get a sense, it seems like we're comparing a lot of our discussion to the true advantage of colonoscopy, which is prevention. And I wanted to get a sense in our decision, how much should we weigh in prevention versus diagnostic? Because the question posed about it being a screening is just an asymptomatic individual and diagnosing cancer. And it seems like a lot of our discussion is revolving around prevention. And I wanted to get a sense of how much that should weigh into our ultimate decision.
That's an excellent point to bring back to the discussion, because that's the intended use of the test, to specifically detect colorectal cancer in a particular population. I think that's important, but still, there's all these other advantages for the early detection in early stages. Dr. Bauman?
Yeah, this is Carla Bauman. Another concern I have, which isn't on this slide here, is that the detection or the sensitivity for stage 1 was like 54% or somewhere around there. And when I asked like what the sojourn time is to like grade 3 or something, no one really knows. And so I feel like we have a real lack of data as to the utility of this for cancer detection. If they're not detecting the early stage, okay, forget sort of doing the prevention. Let's get it as early as we can, i.e. stage 1, and can do things. And without knowing what the frequency of the test is, let's say they missed a stage 1, but next year they test again, but we don't know they're going to test next year. There's no data at this point as to what the frequency of the testing is, or what it should be. So I'm at sort of a loss of how to handle this.
And I'll be interested or curious to see what the plans are by Gardant, and then you see your hand up. So if you can respond to this question of what your plans are for the future.
Yes, Victoria Raymond, Gardant. The stage 1 sensitivity of about 55% that we observed in Eclipse is actually within range of what we saw for FIT-based testing, which is estimated to be somewhere between 50 and 65.5%. So it is, again, within range of the primary screening test that's being used today, and FIT is the leading colonoscopy alternative non-invasive screening test that's being used.
I'm sorry, this is Carla Baumann. Is FIT FDA approved for colorectal cancer screening?
No.
It does not have that indication. Is that correct?
I'd like to clarify. FIT is authorized for detection of blood in stool. There are some FIT tests that had clinical data to support a CRC screening claim, but it is obviously recommended as a CRC screening test and used that way.
That's a very important point. Dr. Spencer?
Yeah, it seems like test substitution is really the key question here. And we haven't been talking much about the data that the sponsor showed on slide 68. It appears that there have been 2 studies done that looked at this, Liang 2023. And it seems like when they offer Shield, that's obviously not a large study, but colonoscopy rates increased and there didn't seem to be much test substitution. And I'm just, that hasn't been a major focus of discussion so far. And I just wanted to bring that up, that there seems to have been an assessment outside of the Eclipse trial of this.
Gardant? Gardant, do you have a feedback for them?
Apologies. Victoria Raymond from Gardant. Yes, we understand test substitution is a critical concern, and through our literature review of understanding how primary care providers are sequencing these tests, how stool-based testing has been used, we haven't seen that to date, but we have explored in prospective randomized trials what this looks like in the real world, and I would like to invite Dr. Liang, who was author on one of these papers, to walk through that data. And if we can share a slide, that would be great to illustrate this discussion.
Hi, Peter Liang. So with regard to the concern for test substitution, there are 2 randomized control trials, one of which I led, which showed there's no test substitution. So I think we're waiting for the slide to pop up here, but there is a 2023 randomized control trial that Dr. Spencer referenced that evaluated individuals who were overdue for screening. The control group was offered FIT or colonoscopy, and the intervention group was also offered different blood tests, a septa-9 test, if they declined FIT and colonoscopy. And compared to the control group, individuals in the screening group, intervention group, the screening uptake increased by 1.8-fold. Importantly, uptake of colonoscopy and FIT in the intervention group was similar to the control group. There was also a different study from 2024 randomized controlled trial that also evaluated individuals who were overdue for screening. And in that control group, screening options included FIT and colonoscopy. The intervention group was also offered the Shield blood test. So similar to the first study, there was a 2.4-fold increase in screening completion in the intervention group compared to the control group. And again, importantly, there was no statistically significant test substitution. So I think the key here is that many people are not willing to undergo screening with the current modalities that we have. And these studies show that a blood-based test increases overall screening uptake and can help the large number of Americans who are not today with screening, which currently stand at 50 million. So these studies support the potential of adding Shield to current screening options and show that there's increased adherence without significant test substitution.
Interesting. Thank you very much. Dr. Hewitt?
Yeah, I'm concerned that the comparator remains fit and not the molecular test, the molecular fecal test, which is really the test that is the FDA-approved antecedent for comparator. And so it is a bit of an apples and oranges situation. I'm aware of the hesitancy of people to engage in fecal testing. I'm a little bit perplexed by it. We've all had children, and many of us have pets. It's not like it's a big problem, but I do acknowledge that people do seem put off by it. I do worry that patient education, both on the complexity of performing the test, as well as what the test can and cannot detect and accomplish with reference to prevention specifically, is critical. The opportunity to have a colonoscopy after positive fecal assay, or with a colonoscopy that is able to treat adenoma and stage 1 malignancy, is a critical element of this discussion.
Thank you. Dr. Birch?
I think the critical thing here, as we keep going around and round about this, is whether the Shield test is going to be a substitute for colonoscopy, or is it going to be additive to colonoscopy? It seems to me it has very poor operating characteristics compared to colonoscopy, so I don't understand how it could replace colonoscopy. So I think it should be offered first line and be maybe a first screening test and added to colonoscopy.
I don't think the sponsor is trying to replace colonoscopy. Is what they're saying is, as the patients refuse to have colonoscopy, that these could be a first line after that to screening, for example, versus the molecular stool versus and this one, and then the blood. So that's what the scenario is at this point. So I don't think the sponsor is trying to eliminate or confuse or confine the colonoscopy as the primary, as a tool for screening. Dr. Morgan?
Yeah, Charity Morgan, I do need clarification because that wasn't my understanding from the presentation. I thought they were proposing Shield as a primary line option, meaning that patients wouldn't have to decline colonoscopy in order to be offered Shield. If Gardant could clarify what their proposed indication is.
Yes, thank you. Victoria Raymond from Gardant. To be clear, colonoscopy should be the prioritized option. And for those individuals aged 45 years and older that are at average risk for colorectal cancer, that are engaging in a conversation about CRC screening options, they should be offered colonoscopy. Unfortunately, we know that there are millions of people who decide not to pursue colonoscopy. And for those individuals, non-invasive options are critical to get people screened today and have been shown to reduce CRC mortality. And the performance demonstrated today with Shield shows that performance, both in terms of CRC sensitivity, advanced neoplasia specificity, and advanced adenoma sensitivity, is within range of other non-invasive screening options. And therefore, the data support that it should be offered alongside, which we mean in a similar manner to non-invasive tests that are available today.
Thank you. That's very clear. Dr. Hewitt?
So I agree with everything the sponsor said in terms of what the science says. But when you listen to the public speak and you address the reality of the situation, they aren't focused on prevention, and they are not aware of the trade-off of difference between this assay and colonoscopy or fecal molecular detection of presence of DNA.
I agree.
As a result, you have a major problem in the education of the population. And that is, I'm wearing the uniform. That's my duty, is to protect and promote the health of the United States. And if they are confused, then there is a duty that we ensure that they are guided towards the correct test that is going to provide them with what they need. If they don't understand that this is not helping them from the perspective of prevention, it's a concern. I'm not saying this assay can't accomplish that in the future, but what I am saying is we don't have the data.
Dr. Spencer?
Yeah, I wholeheartedly agree with Dr. Hewitt. And as someone who has run a primary care clinic and is now a gastroenterologist, I think it's really important to acknowledge that we need to rely on physicians to have shared decision making with patients. And I think education and communication is really the crux here in that. And I was glad to see that very well discussed in the sponsor materials, and that the conversation that needs to be had with patients is, you are due for colon cancer screening, you need a colonoscopy, and there are other tests available that may be involved in the possible suite of screening. But I think what I'm hearing the sponsor saying is that is the discussion that they envision primary care doctors having with patients, is that colonoscopy, although this technically will be considered as a primary screening option, that in the medical shared decision making in the context of a primary care office, that ideally colonoscopy will be offered and the preventative aspects of colonoscopy will be stressed. And I guess I don't want to jump ahead to the third question, but I think it will be important for us to discuss kind of the post potential monitoring of this, because I do think if it is monitored, the colonoscopy rate starts to decrease. I think that would become a major issue. But I think, in terms of the plausibility of that happening versus increasing, I can't predict either way, but I do think that it would be important to proactively monitor, because if we do see the release of this product and colonoscopy rates start to decrease, I think that would be concerning.
Thank you very much. Dr. Bruce, you still have your hand up, but that's...
So sorry.
Okay, so Dr. Borowski.
Remembering the mute button this time. Yeah, I agree with Dr. Hewitt and Spencer. We have to come back to, I think, first principles. First principle is for a screening test to be valuable to a population, it has to reduce mortality. That's undemonstrated in this assay, admittedly, probably also undemonstrated in the stool DNA assay. There's a widespread belief that these methods, if adopted in the population that currently is unscreened, will contribute to a decrease in mortality, but again, unproven. And as has been discussed, the primary benefit to mortality may be the prevention that's effected through removal of adenomas, even if those adenomas do not have potential much of the time to progress. Some do, and removing those ones that do offers a preventative benefit and a mortality benefit. I do think that the onus is on the sponsor to continue to study this, because there is the possibility that, for example, even the very low rate of advanced adenoma detection is still detecting those adenomas that are the important ones. And so it may be that there's a mortality benefit very comparable to colonoscopy, but again, unstudied, so the data's not there yet. So I think that's what we're all talking about. I think the public assumes there is a mortality benefit, and many of them articulated that. And so I think they've been misinformed. And we have to be very careful about that. It is complicated. Cancer is not easy. Not all cancers are created equal. Some are benign, actually, and some are quite lethal. And the public is beginning to understand that, but maybe not well enough. So those are my comments.
Yes, and I completely agree with all these comments, but it's still in the back of my mind is these 5 million US individuals that decide not to have a colonoscopy even today without having a blood test. So what do we do with these individuals? Is this better than nothing? And I don't think we could have some of these conversations, is this better than nothing? But at the same time, is the reality that we're faced with. And also access to colonoscopy in the rural areas and other areas. So that's from a public health point of view, of the number of individuals that get any type of screening, and it's a concern too. Dr. Hewitt?
So Dr. Gonzalez, I acknowledge the limitations in rural health with reference to access to colonoscopy and everything else. I do worry that the public is looking at this assay as well. It tells me I don't have colon cancer. I can move on. And although they keep talking about the number of patients who are, following up, that is a select population. These are people that have gone out and are seeking this. These are the so-called early adopter effects. And so the concern is that the cart gets before the horse on these patients, and that the long-term benefits are, I had it, I don't know, because we don't know the follow-up time. And that's where this really gets to the issue. Does the assay have potential? Yes. But we're missing a lot of wires on this assay that in 1 or 2 years' time, we might be able to have much clearer decision-making than we do today.
Thank you. Dr. Pathak?
Yes, if we could go to slide 13, I'd just like to clarify a few things. Okay, so, first of all, Gardant has been saying that colonoscopy should be offered first, right? But that would potentially, alter the claim that they're seeking, because they're seeking a first-line claim. And the way the USPSTF, colorectal cancer screening guidelines have been set up is that you can choose from a variety of testing options specified by the task force. And these have been roughly been seen, at least in their 2016 publication, to be roughly equivalent, right? Including the high sensitivity FOBT FIT offered every year, the stool DNA FIT like Cologuard offered every 1 to 3 years, the flexible sigmoidoscopy every 5 years, and the colonoscopy every 10 years, and so on and so forth. So we need to be clear about the terminology we use when we're talking about the use of the Gardant test, right? Like, how can you have a first-line claim if you're saying at the same time that colonoscopy should be offered first? I mean, maybe that could be worked into the labeling, but what this list of tests represents is a list of first-line testing options that's deemed acceptable by the task force. There is, obvious variability in performance, but if you could go to slide 51, I can cover some of the highlights of the FDA approved tests, right? Now, Cologuard is an FDA approved first line test for colorectal cancer, and has a 92% sensitivity for colorectal cancer and an 89% sensitivity for stage 1 colorectal cancer. It also has a 42% sensitivity for advanced adenoma and 86.6% specificity. EpiProColon was the FDA approved test that was approved as a second line only after all recommended screening options by the USPSTF had been declined. And this test only had a 68.2% sensitivity for colorectal cancer and 22% sensitivity for advanced adenoma and 78.8% specificity. Now, this Shield test does better with colorectal cancer sensitivity, right? It's significantly better than the EpiProColon at 83%. I mean, that's statistically significant, but also the advanced adenoma sensitivity is only 13%, which is lower than the EpiProColon test had. However, the Shield test has a much higher specificity at 90%. And so I think the sponsor has sort of tried to compare their test to FIT in particular, right? And the specifications for FIT are like 79% sensitivity for colorectal cancer, 94% specificity. And in terms of advanced adenoma, the FIT sensitivity is about 22%. So we should take all of these, all of this information into consideration when determining whether the Gardant test, which is non-invasive, which is only a blood test, whether it should be granted a first-line claim or a second-line claim. And yes, the discussions that have been going on about what population will get this test and whether there will be replacement and so on and so forth, that has been very valuable for us to hear from the panel. And we'd like to hear more from the panel about where this test falls in terms of being first line or second line, and the thoughts around any implications on any segments of the CRC screening population. Yeah, so that's all I had to say.
Is that question number 2? I can't remember.
No, question number 2 is advanced adenoma performance. I was just reflecting back to.
Yeah. I think we have even in 3. Okay, very good. Thank you. Dr. Singh?
Yeah, hi, Vikesh Singh. Can you actually pull up Dr. Pathak's previous slide? Just the one before this? I think it's really important. There's kind of what's out there in terms of guidelines, and then I think there's the second and more important component is actually what are the majority of practitioners using. So, I think it's important to note that, as a practicing gastroenterologist, and there are certainly many on this call, most of these tests are really not being used as screening tests for colorectal cancer, especially flexible sigmoidoscopy. All the studies that supported that are relatively antiquated at this point. I think when you start to talk about what people are doing day to day, I don't even know when the last time was when I screened somebody and said, look, can you please bring out the flexible sigmoidoscope because that's what this patient wants. CT colonography is used largely in patients who can't undergo a colonoscopy safely because they're at sedation risk, they have comorbidities, et cetera. So really what we're largely dropping down to is really the non-invasive tests like stool DNA and the FIT test and colonoscopy. So the real question is, where does the Shield test fit into that armamentarium? And I appreciate we got to pull this up because this even continues to be on the board certification exam, but most GI doctors laugh at it because they're like, I really don't do much of this anymore. I don't really understand what the value of looking at half the colon is, especially if the cancer's on the other side. So, as nice as those studies were from the 1980s and early 90s, they really have no value in today's clinical practice. So, I think that's what we really need to speak of. And then the question becomes and I think the second part of this again is, what's the goal here? If we want to find things early and prevent colorectal cancer, this is not a great test. If this is a test that convinces somebody to go on to get the second gold standard test, that's a somewhat different sort of value proposition. So I think we have to contextualize this before we decide on what we want to adopt, because again, this also adds cumbersomeness to an already cumbersome screening process, right? So let's start to think about that. Now, I appreciate blood may be better than stool, but I'm not so sure that this test is actually showing us that.
Thank you very much. Dr. Hewitt?
Dr. Singh, I'm glad to hear somebody else has got boards that are outdated. I'm glad I got a laugh over that. Okay, so going to slide 51 that was shown previously, we're looking at the Cologuard test. Yeah, its performance at stage 1 is not quite as strong as we're seeing with the Gardant test. On the other hand, its performance with adenomas is substantially better. And if you take the number of adenomas that detected here in this, let's just use these fractions. What you find is that, it found 42.4, and you look at the number there that's shown, it's 321. Well, if you divide that number by 20, by 20, which is 5% progression, you end up with 20 tumors basically. And so what you're discovering is that this assay may give up a little bit in one space, but is actually more effective early on in detecting those adenomas that are going to become cancers. So, it is a trade-off of prevention versus detection at a later stage. And I think we all want to prefer prevention. And that is the challenge. I mean, when I look at the performance of this test, it looks a great deal more like the epiprocolon. So that was, quote, a second line test. And, it's not unreasonable to think that one could have a second line test that with additional data gathered over a period of time could be restaged and moved up to a first line test. But again, we're being asked to approve a test for whom we really don't understand the performance over time in serial sampling.
Dr. Pathak, you had your hand up?
Oh, I just wanted to clarify that Cologuard had 89.7% sensitivity for stage 1 colorectal cancer. They picked up detected 26 out of 29.
So I was even off.
Thank you very
So
much.
it's significantly higher performance.
Okay. So yeah, it's performing where you want it to perform, which is those events that do not require abdominal surgery, chemotherapy, or additional advanced therapy. And that's the burdensome place for patients. Now we heard from patients, they don't want to have big surgeries and chemo and everything else. I mean, that's what our public talked about. But the problem is that you need to be able to detect stage 1 and adenomas to accomplish that goal, prevention.
Thank you. Dr. Morgan?
Yes, this is a question for the FDA. Do we have data on what the stage 1 sensitivity was for epiprocolon?
You're muted, Donna.
Sorry, I'm on mute again. If you give me 1 second, I can look it up right here. Stage 1. I'm not seeing it here. I will ask one of my colleagues, do you have the data for stage 1 cancer detection in the epiprocolon?
Point of clarification.
Yes, the epiprocolon picked up 7 out of 17 or 41% of stage 1 colorectal cancers.
What year was epiprocolon
31%?
approved?
No, 41%.
41%.
What year was that assay approved?
And it was approved as a second line.
Yeah.
So I think what we're seeing here is that the operating characteristics of Shield, to me, they seem closer to epiprocolon, both on the sensitivity for advanced adenomas and the stage 1 sensitivity for colorectal cancer. But it does, Shield is outperforming epiprocolon and doing better, closer to Cologuard on the specificity and the overall sensitivity for colorectal cancer, just to sort of add more to the picture.
Yeah. This is
Dr.
Steve.
Do you have a response to that, Dr. Hewitt, to Dr. Morgan?
No, I was going to comment that what it appears is that epiprocolon is a bit like the Flex Sig that Dr. Singh was referring to. It's an assay that was approved but has fallen out of favor, no longer available in the market because it did not meet the clinical need.
Yes, but the approval we have to look at is safety and effectiveness. How it goes into the clinical need, I think it is kind of, but that's what we need to look at. Dr. Winslow?
Yeah, just building on a number of, this is a great conversation, building on some of the comments that have been made. So keeping in mind that, as I understand it, it's not a replacement for colonoscopy, but really the focus is on patients who forego colonoscopy, right? And if we think about it in that context, what's the benefit there? The idea is that it increases access, it increases adherence. How best to do that? Do we place controls on it, or do we, through education and through post-market studies that are able to further inform us, whether it's on intervals or otherwise, allow that to be the position that it takes in the market initially, because it's alongside those other options. And therefore, the physicians and the patients, through education, the appropriate materials, are able to make those informed decisions. Because I think that's a lot of where the conversation is around. So I'd be interested in exploring that a little bit more in terms of whether that's something that could be, feasible from some of the benefit risk discussions that we're having.
Dr. Burge?
Can I get clarification? Can we all get clarification from the FDA or other people on the difference between first line and second line screening tests for colon cancer?
What are the differences? Are you asking that?
Yes.
Yes.
So I want to stress that epiprocolon got what is called a second line, which is that the IU actually specifies that the patient must decline. And this question
decline colonoscopy.
Decline other screening tests, all other screening tests, because the performance was substandard to fit. And that is not an expectation that the FDA would have, generally moving forward with new tests. However, FDA recognized the benefit, as did the panel, of a blood-based test. So that is why it got the second claim.
Can I ask a quick question about
while being approved? I'll just say while being approved.
clarifying question about that to Donna.
Yes, go ahead.
If Shield were approved saying that patients had to decline colonoscopy, but did not have to decline other screening tests, will that still be considered a second line claim?
So I think that that's an excellent point, and I would just like to point out what I think a lot of the esteemed panel members have raised, and that is that the intention of alternatives to these tests, and that is a discussion that physicians have with their patients. Cologuard is not as good as the colonoscopies, yet they do not have a second line claim or have a requirement for documentation that the patient has refused colonoscopy or is not there. It's a discussion that physicians have with their patients about alternatives when they are recognizing that they're not compliant with their colonoscopies, excuse me. And I think that Dr. Hewitt raised a very good point when he said that, and also someone else in the panel raised a good point about making sure that patients understand the purpose of colorectal cancer being for prevention, the purpose of the education, so that they're really well informed about the decisions that they're making when they make these tests. So these tests are all part of a screening program that are used in lieu of colonoscopy.
So the sponsor's statement that this test is not intended to replace colonoscopy, that's not contradictory of going after a first line claim.
No, well, this is a personal opinion. This is what we've engaged you to discuss, but this is a clinical discussion, and everyone acknowledges, and presumably clinicians acknowledge, colonoscopy is the gold standard. When colonoscopy is not being done or not being done for 10 years, what are the alternatives for screening? And so that is these alternatives in a colorectal cancer screening program include Cologuard, FIT, and the other tests that were on slide 13.
Okay, thank
and
you.
Now we're discussing where and how Gardant might fit into this scenario.
Dr. Bruges?
Well, just a quick comment there. Sorry to keep going over this over and over again, but still, the crucial question here is whether Shield is going to replace or add to testing with colonoscopy.
Right. So this is the concern. This is part of the benefit risk assessment, is acknowledging that the potential benefit of this test is that it will provide more patients with knowledge about their colorectal status. The risk is that it will deter people from more effective testing. That's the same risk for any of these other alternative screening tests. And in consideration of the performance, we're asking the panel's opinion in this regard. And appreciate that you've been weighing in this regard.
So if we can go back to question number 1. So with regard to question number 1, I think the committee is in some agreement that the test is adequate for screening more advanced colorectal cancer stage 2, 3, and 4, and that there is any benefit of having a blood test done for the compliance and accessibility in different areas. But the committee has still a lot of concerns of the lack of sensitivity in early stages, and then the number of false negatives that could occur, and how that could be viewed or understood in the community. Does the FDA needs anything more, Donna?
I really appreciate all of your very valuable input. It's been extremely helpful. I would ask one quick question. I'm not sure if anyone is prepared to answer it, but we would be interested to have you expand briefly on the mention of educating patients to the value of prevention, and in those panel members' experience in the clinical setting, any comments they might have about successfully ensuring that patients are informed would be valuable. I don't know, it does not have to be done at this point, but we would appreciate that expansion of that discussion at some point during the questions.
So yeah, but it's also interesting to see that as in an area when there are recommendations in addition to genetic testing for screening, for example, carrier screening for cystic fibrosis, it took about 5 to 10 years for those recommendations to go into effect or even really get widespread. So that's something to have in mind too, that even though we're seeing that education is very important, not only the public, but also the primary cares, that is going to take some time.
We've made some headway in breast cancer in this kind of education, and it involves usually infographics because it's hard to talk about statistics without sort of a picture, but discussing of X number of women screened, how many will have a false positive, what does a false positive mean? It means a biopsy, and waiting to figure out what that biopsy is, and so forth. And of the women for whom cancer is detected, what proportion are we actually able to evoke a better outcome? And it's a minority, so there's common belief that the mammogram saved my life, but the reality is obviously much more complicated, with many of those women having detection of lesions like DCIS that maybe are not really lethal and could have been left alone, and some invasive cancers that fit in the same category. And then, of course, there's the possibility that even when we detect a consequential cancer and we detect it at an early stage, its biology is such that it's still lethal. And so that's the kind of education we're doing in breast cancer. I think colon cancer and colon cancer screening is a bit different, but those are the things that we really need our patient populations to understand.
Very good.
Thank you.
I would actually, so I'm a physician who sees both patients with breast cancer and a hereditary geneticist who sees patients who have cancer syndromes, but aren't affected by cancer. And in this type of setting, there are a lot of variations and flavors of tests across the spectrum there. And to that end, this type of testing is something where for different reasons, I've discussed with patients the possibility of having testing that can identify an existing cancer, but not be able to intervene on something that is pre-malignant or could become a cancer. And the language can be that straightforward.
Thank you. Dr. Hewitt?
Yeah, this is Dr. Hewitt. I think that the education issue is very challenging. Americans have become very familiar with blood tests, But what we discover is they imbue them in powers far beyond their scope on a regular basis. They really are looking for answers and don't understand the limitations. People speak to me as a pathologist about their test constantly. And what you hear is the trend and the theme, and again, back to molecular as well, is that they assume an answer is a finite answer. And I think that's one of the big challenges here is I had a blood test, I don't have colon cancer, move on. We've had to work very hard to finesse our communications on the blood test for prostate cancer. And in fact, I don't think we're accomplishing what we need to. As Dr. Orowski pointed out, with breast cancer, we've had the same challenge about the significance of lesions and how many of them are probably not that aggressive and everything else. And so you end up with major risk of overtreatment. You end up with other risk. And so it is something that, honestly, once you're dealing with patients who are over 50, their thought processes are locked in on this. You need to be educating them about prevention at my son who's 14 and my daughter who's 21's age, probably my 14-year-olds, not my 21-year-olds.
Yeah, and then we have also an additional comment from Dr. Singh to the whole panel that it says that if that in negative result might falsely reassure the patient that doesn't have cancer. Thank you very much. So we will go to question number 2. Patients with AA have a high risk of developing colorectal cancer. The Guardant Eclipse study demonstrated that 83.1% sensitivity for colorectal cancer, but only 13.2% sensitivity for the detection of AA. Please discuss the benefits and risks of colorectal cancer screening tests with 13.2% sensitivity for AA. And I think we have alluded a lot of these already. If the risks are present, please discuss whether there are potential mitigations which might be deployed to ensure physicians and patients are able to make informed choices regarding screening test options to mitigate clinical risk of the Shield test AA sensitivity. Dr. Morgan?
Hi, Charity Morgan. So yes, we've already kind of gone over this quite a bit. I just would want to say, based on sort of the materials provided by both the sponsor and the FDA, comparing the performance of other approved non-invasive tests, I think that the sensitivity for AA of Shield is not at the level of these other approved non-invasive tests. So Cologuard is at 42%. FIT FIT's not approved for colorectal screening, but it's at 23%. Even EpiProColon, which is second line, is 22%. So this 13% is, I think, substantially less than what we're seeing for other approved non-invasive tests. So I think if Shield were to be approved as a first-line treatment, I think that it would be important for the labeling to really make it clear that this test is really only going to be able to detect stage 2 and later CRC with any real reliability, and that advanced adenomas at stage 1 are a real limitation for this test. So I think that's a possible mitigation, is very clear labeling for the product.
And I think, there is a concern about this sensitivity for AA, but maybe further studies in follow-up to the different time for repeated testing might be so very important, because we're talking about maybe 1 every 3 years, but again, we don't have the data to reflect if it actually advantageous or not, because it might take even more than 3 years to develop them at that point. But I think we've gone over some of this. I think with the committee, I understand that there's a risk, with the sensitivity for AA in this patient population. But is it in the book can recommend that could mitigate the deployment to ensure the physicians and patients are able to make informed choices? Dr. Hewitt?
I remain perplexed and concerned that patients don't necessarily understand up front that they're drawing 4 tubes, 40 ml of blood for this test. And if you're confronted with 4 tubes of blood, which may require multiple venipunctures, versus a fecal test, there needs to be clarity about what is going on. I also remain concerned that there's a population of patients for whom 4 tubes of blood may not be adequate. Sponsor noted that they're only using probably on the order of 7 to 10 mils of the blood, but the kit is asking for 4 tubes. So I'm a little bit perplexed on this as well. But it's an important factor in terms of adoption and patient selection. You have an elderly patient. This test is tested up to age 85. You have plenty of patients in their 70s who are relatively frail, for whom a 4-tube collection may really be a little bit much.
I was just curious, has the sponsor thought about less requirements? I know the clinical trial might be 4 tubes of blood, as Dr. Hughes said, but you don't require that. Even 2, you could still have leftover for repeats.
Yes, Victoria Raymond, Gardant. Yes, we do know that we only need about 2 tubes of blood to run the assay, and actually our assay failure rate for various reasons, including not enough blood sample, is very low within the lab, less than 2%. So that doesn't appear to be an issue going forward, but that's the data we have now.
Okay.
However, this is Dr. Hewitt. The documents provided to the committee were that 4 tubes of blood. So that is currently what the assay is specified for.
That's what's requested in the blood collection kit, but we don't, that's not what's required to actually run the sample.
But is that in the labeling or not? That, as a tester and a laboratorian, what is in the labeling and what is requested are critical issues towards ensuring that the test is the person receiving the test, drawing the blood, needs to understand exactly that. That's a specification.
I will ask Dr. Chudova to address your question.
Thank you, Daria Chudova, Guardant Health. So the test kit contains 4 tubes, and we request to collect up to 4. A single run of a test in the lab requires up to 2 tubes of blood. In the instances of QC failures of the first aliquot, we do go to the second aliquot, and that's what the 3rd and 4th tubes are used for in a typical process.
Okay, so then you do require for repeats the extra 2 tubes.
That's why the kit contains 4, but if it gets shipped as 2 and to return successful analysis result on the first analysis, we will report that result.
Okay. It's a lot of blood. Dr. Ballman?
Yeah, thank you. This is Carla Ballman. It is a lot of blood, and I wonder what that's going to do to adherence rates in a longitudinal study after someone's gone through sort of drawing 4 and not finding it pleasant.
Dr. Bruce? Yes, Dr. Bruce.
I wanted to clarify the indication in the package insert for this. Is it for screening of colon cancer or for polyps or both? Obviously, I think it should be screening for colon cancer, not polyps.
Gordon?
Yes, if we can please pull up the indication for use slide, which I believe is in the FDA's deck, or we're happy to pull it up as well. If we can share, okay, great. So we will go ahead and share if that's okay. And so what the proposed indication for use is for the detection of colorectal cancer. And it's for colorectal cancer screening in individuals at average risk of the disease, age 45 years or older.
Thank you very much. Does the FDA has everything they need?
Yes, thank you.
Okay, very good. So we will be moving. So the committee still has concerns about the lack of sensitivity for detection of adenocarcinomas, and the educational piece will be critical for making sure that not only the providers, healthcare provider, but mainly the public understand some of these limitations, but they're still concerned that might not be understood. Question number 3. If the device is determined to be safe and effective based on existing data, please discuss whether a post-approval study to gather additional information about benefits and risk of programmatic colorectal cancer screening, such as repeated testing over an established period of time, would be beneficial. Please discuss the types of information that will be important to collect during such study. And I think we have discussed among already all of us that post-market or further studies on longitudinal will be extremely important for this sensitivity of AA, but at the same time on the schedule, how often you have to test it to give you maybe improved sensitivity for AA. Dr. Rajapodal?
Padma Sheila Rajagopal. Yeah, one of the points I was going to make was on frequency of retesting, but another point I'd made that I'd alluded to with the prior question was on incidental findings. So, as the sponsor alluded to, they do capture points with their cfDNA, such as microsatellite instability that could prompt further clinical testing, but they aren't sharing that data with patients, aren't validated within this range to do so, but it would be important to clarify sort of a recommendation that can be provided to patients about referral for additional testing, given that would be a clinically relevant finding. So how that would be incorporated into post-marketing would be relevant.
Dr. Morgan?
Yeah, I just had a clarifying question to the FDA. This post-approval study, are you asking about one that be conducted by the sponsor? Because it does talk about programmatic colorectal cancer screening. I didn't know if you wanted sort of an idea about studies about the entire landscape, or you're speaking specifically about post-approval studies about Shield.
Post-approval studies to mitigate the risks of Shield.
And so with that in mind, I think things that would be helpful to collect in that would be more information about those false positives that they were seeing, and more long-term data for those subjects.
Dr. Spencer?
I think the Eclipse study that we mentioned is a patient population that was already engaged in and willing to get a colonoscopy. I'm very curious in a population where programmatic colon cancer screening is performed, so more in a primary care setting, what the operating characteristics are in that setting, because I think that is ultimately where we envision this being used. I think to gather the information that we've discussed today, the Eclipse study was necessary to be designed the way that it was, but I think it's very important to understand how in a real world setting this is operating. And, I could predict it either dramatically improving performance characteristics or keeping the same or decreasing. And I think also integrating within that study, any replacement statistics, I think will be important, especially in the context of global programmatic colorectal cancer screening to understand.
Thank you. Does the FDA has everything that they need for this question?
I believe so. I would like to acknowledge that I would like to confirm that the panel believes that evaluating the performance of the test to support interval testing would be beneficial. I don't think we actually tapped into that.
Yes. I think there was when we were discussing question number 1, that there was a consensus that that there was still not data on the value or how often do they testing, and then what the improvements to the outcome will be. Dr. Hewitt? You're muted, sir.
Thank you. This is Dr. Hewitt. I want to amplify on Dr. Spencer's comments. I think a real-world testing environment in which different interval periods are evaluated and that it's not just one interval. If you have a patient that you are going to test at a yearly interval, you probably need to test them for 3 times, 2 years. If it's a 2-year interval, you may need to test them at the initial at 2 and at 4. I'm not so certain about 3, but a fixed interval one time is not going to provide the resolution of information that's going to be required to really understand the performance of the assay and how it impacts public health. Probably both need to test different intervals and more than one interval.
Thank you.
Thank you.
Dr. Rajapahul?
Yeah, I just wanted to add that given how much we've emphasized education as a panel, it would be very helpful to have some degree of study as to whether patients and providers understood the educational materials and seem to have an accurate understanding.
That's very valuable. Thank you very much. So at this time, the panel will hear submissions, comments, or clarifications from the sponsor. And I'm sorry, FDA first. You have 10 minutes. FDA first.
We do not have any additional questions at this time.
Great, thank you. Now we'll hear submissions, comments, or clarifications from the sponsor. You have 10 minutes.
Thank you. Victoria Raymond from Gardant. First, I really want to thank everybody for the discussion today. We really value your expert feedback and advice. We completely agree that colonoscopy is the best test and should be offered as the first option for patients. We know that for patients who prefer a non-invasive alternative, current options, which are really limited to stool-based tests, play a critical role in reducing preventable colorectal cancer mortality. Today, millions of patients utilize tests like FOBT as their primary screening option, and millions of patients are utilizing FIT as their primary screening option. In fact, today, annual use of FIT is greater than the multi-target stool DNA, and all of these options are offered to patients and physicians as first-line screening options. What's important here is even with those options, we still have 42% of people who are not up to date with screening. Can I please share a slide from our presentation on the screen? Data from Eclipse demonstrate that Shield's performance for colorectal cancer detection is in the range of these non-invasive stool-based tests. This performance for colorectal cancer detection is within range is in line with our proposed intended use statement. Again, we want to reinforce that we do not believe in any way that Shield should replace colonoscopy for those patients who are willing to undergo it. We're absolutely committed to making it very clear in our educational materials that colonoscopy is the best test and should be offered to patients as the first option. Rather than replacing colonoscopy, it is our position that the data support Shield should be offered as a choice for patients, similar to those guideline recommended and FDA approved non-invasive stool-based screening tests. The underlying principle of colorectal cancer screening is to reduce colorectal cancer mortality. In this regard, our goal is to get more people screened, because if we get more people screened, we will have a greater chance at reducing the number of preventable colorectal cancer deaths. Shield offers an important choice for patients and physicians who are at average risk for colorectal cancer. Thank you so much for your thoughtful discussions today.
Thank you. So before we proceed to the panel vote, I would like to ask our non-voting members, Mr. Lofspring, Nathan Winslow, and Angela McLeod, if you have any additional comments. Ms. Bromelt?
So there's a typo. It should be Lofspring goes first.
Oh, okay. So Mr. Lofspring?
No, I have no additional comments at this time. No.
Okay. Nathan Winslow?
Yeah, just briefly from an industry perspective, we do really see this as an important advancement in cancer screening. It has a real potential to increase patient access and adherence, and I think that's what the focus a lot of the focus of the discussion has been and what the studies have shown. The sponsor has designed a study that it was based on what guidelines and other non-interventional options are performing around, and their data shows that they're in line with what those tests are showing in terms of performance. So I think it's important just from an industry perspective to look and see how this can be an option or a choice, as was phrased, alongside those other tests to really yield the potential of this from an access and an adherence perspective. And if there are ways to balance the benefit risk through patient education, labeling, and more information in the post-market setting, I think those are all things that from an industry perspective would make sense.
Thank you. Ms. McCloy?
Angela and Marilyn Cloud, no additional questions or comments on my behalf.
Thank you so much for your thoughtful comments, everybody. So we are going to be now moving to a vote. We're now ready to vote on the panel's recommendations to the FDA for the Guardant Blood Collection Kit. The panel is expected to respond to 3 questions relating to safety, effectiveness, and benefits versus risk. Mr. Swing will now read the 2 definitions to assist you in the voting process.
The medical device amendments to the Federal Food, Drug, and Cosmetic Act, as amended by the Safe Medical Devices Act of 1990, allow the Food and Drug Administration to obtain a recommendation from an expert advisory panel on designated medical device pre-market applications that are filed with the agency. The PMA must stand on its own merits, and your recommendation must be supported by safety and effectiveness data in the application or by applicable publicly available information. The definitions of safety and effectiveness are as follows. Safety, as defined in 21 CFR section 860.7D1. There is a reasonable assurance that a device is safe when it can be determined based upon valid scientific evidence that the probable benefits to health from use of the device for its intended uses and conditions of use, when accompanied by adequate directions and warnings against unsafe use, outweigh any probable risks. Effectiveness is defined in 21 CFR section 860.7E1. There is reasonable assurance that a device is effective when it can be determined based upon valid scientific evidence that in a significant portion of the target population, the use of the device for its intended uses and conditions of use, when accompanied by adequate directions for use and warnings against unsafe use, will provide clinically significant results. The proposed indication for use submitted by the sponsor, as stated in the PMA, is as follows. The SHIELD test is a qualitative in vitro diagnostic test intended to detect colorectal cancer-derived alterations in cell-free DNA from blood collected in a Guardant Blood Collection Kit. SHIELD is intended for colorectal cancer screening in individuals at average risk of the disease, age 45 years or older. Patients with an abnormal signal detected may have colorectal cancer or advanced adenomas and should be referred to for colonoscopy evaluation. SHIELD is not a replacement for a diagnostic colonoscopy or for surveillance colonoscopy in high-risk individuals. The test is performed at Guardant Health Incorporated. Panel members, we will now begin the voting process. Each voting member has received a voting link. Please remember to add your name to the ballot. Once I read all 3 questions, we will take a short break to verify the votes, and then I will read the votes into the record. Voting question 1. Is there reasonable assurance that the SHIELD test is safe for use in patients who meet the criteria specified in the proposed indication? Please vote either yes, no, or abstain. Question 2. Is there reasonable assurance that the SHIELD test is effective for use in patients who meet the criteria specified in the proposed indication? Please vote either yes, no, or abstain. Question 3. Do the benefits of the SHIELD test outweigh the risk for use in the patients who meet the criteria specified in the proposed indication? Please vote either yes, no, or abstain. At this time, please give us a moment as we tally and verify the official votes. Thank you.
Okay. Dr. Gonzalez, we may want to go to a break now so we can tally up the votes. If you could take us to a break.
Can you remind me how to find this voting link?
I sent it in your chat.
Okay.
We'll go ahead and take a break and we'll tally up votes. So Dr. Gonzalez, can you take us to break?
We'll go to break for how long? 5-10 minutes?
Yeah, something like that.
Okay, 5 minutes.
Thank you.
We're back. We tally the votes. On question 1, the panel voted 8 yes, 1 no, no abstentions. The data shows reasonable assurance that the Shield test is safe for use in patients who meet the criteria specified in the proposed indication. On question 2, the panel voted 6 yes, 3 no, no abstentions. That there is reasonable assurance that the Shield test is effective for use in patients who meet the criteria specified in the proposed indication. And the final question, the panel voted 7 yes, 2 no, and no abstentions, that the benefits of the Shield test outweigh the risk for use in patients who meet the criteria specified in the proposed indications. The 3 voting questions are now complete. I'll now turn the meeting back over to Dr. Gonzalez. Thank you.
Thank you, Mr. Swin. We will now take 5 minutes break to allow we just did that, I'm sorry. Welcome, everybody. The votes have been received and have been read. I will now ask the panel members to discuss their votes. If you answer no to any question, please state whether changes to labeling, restrictions on use, or other controls will make a difference on your answer. For the record, please state your name and how you voted for each question. We can start with the panel. Dr. Borowski?
Yeah, I voted yes on all 3 questions. I do think there's a responsibility going forward for education and proper labeling, and a further expectation that ongoing studies will specifically address eventual mortality impacts and maybe intermediate to that, biologic features of the differences between screen detected and undetected lesions when they do arise. Thank you.
Dr. Raja Gopal?
Yeah, I voted yes to questions 1 and 3 and no to question 2, and my vote would be influenced by labeling. The reason that I'd voted no for anticipated efficacy, I think that's the question, was because of the questions surrounding adenoma and stage 1 cancer, which were part of the indicated use. So it's the same as what Dr. Borowski is saying, and I think other panelists has discussed at length. If there were refinements to that indicated use or refinements in the way that providers and patients were educated, I would change my vote to a yes.
Thank you. Dr. Hewitt?
I voted no for all 3 for exactly the reasons stated above. Reason the first one, the safety I'd have voted a no is it puts patients directly at risk for the development of cancer because we do not have the data on the follow-on assay. That is a stricter interpretation, but it involves the impact of the population. So with additional data, I do think that the vote may be different, but with the information we have at hand, no.
Thank you.
Not fit for purpose.
Thank you. Dr. Spencer?
I voted yes on all 3, with the interpretation, with the indication that this is to be used in asymptomatic individuals for the detection of colon cancer. I did feel like it met that indication and thought so. I think in the labeling, it should clearly indicate that this is not to detect adenomas and is not designed as a preventative strategy. I think for its definition of a screening test to detect colon cancer, I did feel like it met the criteria. And I think the studies, as we've all mentioned, that need to be done are to understand it in the context of its intended use in a colon cancer screening program, particularly in a primary care setting where these tests are instituted.
Thank you. Dr. Morgan?
Charity Morgan, I voted yes on all 3. My reasoning was very similar to Dr. Spencer's. The indication is for detection of colorectal cancer. If the indication had said something about advanced adenomas, that would have changed my vote. I was on the fence about the effectiveness because of the limited sensitivity for stage 1. I think if the labeling was very clear that this test is strongest for detecting stages 2, 3, and 4, that would turn my vote from a weak yes to a strong yes. But ultimately, I concluded that they did show effectiveness as well as safety and a good risk-benefit profile.
Dr. Bruges?
Yes, I voted no on 2 and 3, and yes on 1. My primary concern here is that I don't think SHIELD is a particularly good screening test for colon cancer. And I think that many of the other existing tests, including stool-based tests, are better than the blood test.
Dr. Baumann?
I voted yes on all 3 questions, and I echo pretty much what others have said, especially Dr. Morgan. I'm hoping that in the label it does. I mean, I do feel like it is a good colon cancer screening test, but for later stage colon cancer, not as good for stage 1, and definitely not good for advanced adenomas.
Thank you. Dr. Singh?
I voted actually yes for all 3. That's primarily because I think there is the adherence component. I know the stool test is a little bit better, but it's much more cumbersome for the patient. And I think in many instances, I've seen them not get done, whereas this is something I think patients can adhere to a little easier. But I do share the same issues around labeling. I do think it should be somewhere in the label that should say that this is a screening test for asymptomatic colorectal cancer detection and not polyp and adenoma detection. Something to that nature, I think, is going to be really important. And I think it's also important to make it clear that a negative result does not reassure one that you don't have a finding that is going to require a colonoscopy. So I think that's sort of where the problem in some ways lies, right? Is really that negative result patient, or is it good enough to go home and say, look, I don't have a problem, I don't need a colonoscopy? I'm not so sure I can provide that reassurance at this moment in time. The other thing that I don't know if anyone really brought up, and I don't know how common this is, but you almost wonder whether these kinds of tests, as they proliferate, whether there should be a need for securing consent from the patient before they undergo testing or screening, whatever you want to call it. Because, I think a lot of people might just be like, well, I'm getting this test, so just like I get a CBC or a comprehensive or a TSH, and if the result is normal, or in this case negative, I'm good to go. And I'm not so sure that's sort of what they should walk away with. I think if a physician sat down and said, look, I'm happy to order this test, but you need to be clearly consented and sort of express, so I can express to you what are the risks and benefits and the performance characteristics of this test. I think that would add a lot of value, but I don't know if that's something that the FDA has jurisdiction over.
Thank you. Dr. Gilger?
I voted yes on all three. I would echo Dr. Singh's erudite comments, very much so. I think there's a very, very strong post-marketing surveillance, specifically to look at the impact on colonoscopy, the impact on adherence, and the impact on colon cancer mortality. Thank you.
Dr. Roscoe, do you have any final remarks from the FDA?
Can you repeat that? Did you call my name?
Yes. Do you have any final remarks from the FDA?
No, just to say thank you very much. I'd like to express our appreciation to you, the chair, Dr. Ferrer Gonzalez, and the rest of the panel. You've been exceptionally helpful to us. You've provided us a lot of expert testimony and input, which will certainly guide our decision making. It's not only valuable for this test, but obviously for a lot of colorectal cancer screening. It informs a lot of information about our approach to colorectal cancer screening and screening in general. So huge appreciation, huge thank you for your time today, because it is obviously a very intensive process that you have to invest in, and I appreciate that, as does the rest of my colleagues. I would also like to thank the public for sharing their stories with us and their journeys through this challenging clinical context and diseased. And we appreciate that, because we do take patient positions and their perspectives very seriously at the center, and appreciate their time in that and energies in providing us with that information. Of course, I would also like to thank Gardant for their time and presentation today. And finally, I would like to thank my colleagues at the FDA, who have been working extremely hard and diligent, not only on the review of this submission, but also on the panel itself, because ultimately at the end of the day, it's about the patients, and we all are invested in the best outcome for the patients. So thank you so much for your time today.
I would like to thank the panel members, Guardant Health representative, and the FDA for their contribution to today's panel meeting. This meeting of the Molecular and Clinical Genetics Panel meeting is now adjourned. Thank you so much.